Good morning, everyone. I request everyone assembled here and joining us online to kindly mute your cellular devices. All participants who have been allocated to display their posters today, kindly do so at the earliest, if not done yet. Please note that there will be a group photograph at 2 p.m. before lunch at the amphitheater just outside the auditorium. We will start the program in 5 minutes. Internet access details are all across the auditorium for the ease of login.
So given the weather situation, there is a possibility that we might have the group photograph a little early than we have planned. So let's see when there is a little bit of light so that we can get, because otherwise I think we are going to lose a lot of important people by today evening. So we don't want them to be missing in the group photograph. So let's try to see how best we can get this arranged. Thank you.
On the behalf of the Symposium Organizing Committee of Translational Health Science and Technology Institute, THSTI Faridabad, I, Rohit Kashwaha, extend a warm welcome to all of you to the second day of the 3-day symposium on Towards End TB, Achievements, Challenges, and Future Directions. The main theme of this symposium is Shortening TB Therapy. Today is 24th of March, and 140 years ago, that is on 24th March, 1882, Dr. Robert Koch announced to the world the discovery of Mycobacterium tuberculosis. Since understanding the pathogen also involves delineating the TB pathogenesis, I take this opportunity to warmly welcome Dr. Audrey Stein to deliver the keynote lecture titled Studying Human Tuberculosis Lesions Across the Spectrum of Infection and Disease, Implications for Drug and Vaccine Discovery. Dr. Stein holds parallel appointments at the Africa Health Research Institute, AHRI, in Durban, South Africa, and at the University of Alabama at Birmingham, USA. At AHRI, which is a Wellcome Trust and HHMI funded, he leads the Human Lung Project efforts. He's a leading authority on bioenergetics and immunometabolism of Mycobacterium tuberculosis infected host cells and the role of nitric oxide, carbon monoxide, oxygen, and hydrogen sulfide in TB pathogenesis. I urge him to enlighten us with his valuable insights. Following the keynote address, there will be a 5-minute Q&A. In the interest of time, I request audience to please keep their questions brief so that there's an opportunity for others. Dr. Stein.
To use this or this one down arrow. Okay. Well, thank you very much for the very kind introduction. And I would like to thank the organizers for inviting me to give this talk. I'm really, it's an honor to be here. I have very fond memories of India in 2003 when I became a young assistant professor at UAB. Soon after that, I got invited to I think the first or maybe second ICGB conference, which was an absolutely amazing experience. And since then, I've been visiting India quite frequently, and I really love the country. So let me start immediately and give you a quick outline of the talk. Firstly, many of you might have wondered, why do we see the mass pathology, for example, human TB lesions and a few bacilli? It's not like every lesion that you see, a human TB lesion that you see is teamed with bacilli. And as a matter of fact, most of them are paucibacillary or abacillary. You don't really see the bacilli. This certainly must have diagnostic and prognostic implications, and also implications for understanding how TB causes disease, because of the very simple fact, if you don't see the bacterium, how do you know what the mechanism of disease really is? So the second part of my talk, I will be asking the question, how can knowledge on human TB disease help understand drug mode of action and drug or / vaccine development? I'm not going to go into much depth. There, I'm going to emphasize the micro and macro environment of the TB lung and why it is important, and why we must improve understanding of the cellular and pathological basis of active and TB lesions. So the talk is not very complicated. It should be very simple to follow. It's geared towards the students and the postdocs. So this represents the spectrum of TB infection disease. Obviously, there's a distinction between infection and disease. Initially, the infection can be cleared, or if it's not cleared, it might end up in the so-called latent TB infection stage, which could then further progress towards the subclinical TB disease stage, where you may have an abnormal chest X-ray, you may be sputum positive or negative, but you feel fine, similar to latent TB. And subclinical TB disease can further progress towards active TB disease, which we know a bit about. So I'll be focusing on active TB as well as latent TB, the disease pathology aspects of these two different disease states. So as we all know, I think we would agree that new methods for detecting TB are urgently needed. And again, I'm just making the point here, those white blobs that you see there, that's the tubercles. And again, if you look on H&E pathology, what you'll see is that there are very few bacilli. Okay, so these are the white blobs, right? When you do an H&E or a Ziel-Nielsen stain, there's very few bacilli. So why is that the case? Is it because of the lack of good detection methods? Most probably so, because the Ziel-Nielsen method was initially developed in the 1890s by more or less 5 people who developed the assay, and that in itself, that's more than 100 years ago. So we haven't really moved beyond that. So that is a gap in our knowledge, or a challenge. So interestingly, decades ago, it was shown a TB colony, and this was well known in the 1940s and 50s, that a TB colony shows characteristic stratification, which can be differentiated by 3 layers, which is the non-acid fast, weakly acid fast, and strongly acid fast bacilli, right? And this is page 1 in a book written by Kane and Pagel, 1948. That was the common knowledge at that point in time. They fully recognized it is for what it is. I think today we would like to ignore those type of facts because it just complicates things, right? So the diagnostic and pathophysiological implications of this biological property has largely been underappreciated and is rarely cited, right? But I think many of us, we're aware of that fact, but it's an inconvenient truth. Most necrotic TB lesions, like I mentioned before, is paucibacillary. This is a good example, and I've shown this quite frequently, of a tuberculoma, necrotic TB lesion. And right in the middle there, you have this area filled with bacilli. So why that's the case, we have no idea, but this is very rare. I've only seen a few of them after looking at really thousands of specimens. You don't see a lot of necrotic TB lesions filled with bacilli like this. And cavities, yeah, that's a different story. So what I'll be talking in the next couple of slides is the application of RNAscope for the detection of remnants of Mycobacterium tuberculosis. To give you a little bit of background on RNAscope, basically it targets TB RNA, right? And you have up to 20 pairs of probes that covers about 1 kb of your target gene of interest. And these probes bind in pairs. And only when the probes bind as a pair do you actually generate a signal. That signal gets amplified, and ultimately what you end up with is a punctuated dot, which is an indication of a single transcript. So 1 spot is a single transcript. So we attempted to evaluate this approach for the detection of TB, but as we all know, TB has a waxy cell wall, and the question was, is the probe actually going to penetrate if you want to stain whole bacilli, or, do we have free-floating TB RNA in tissue specimens? So we investigated that, and after a lot of optimization, really careful optimization by some of the technicians and postdocs in the lab. Oh yeah, it's very, very sensitive and very specific. So I have to point out also, it's not an indication of the strength of expression. It's just simply a single transcript. And a more intense signal means that there's more probes binding to the target. Less intense, there's less probes binding to the target. It's not an indication of expression. So we look at extrapulmonary TB. This is a testicular TB sample. On the left-hand side here, you have a Ziehl-Neelsen stain. This is the seminiferous vesicles with the spermies, and this is just loaded with MTB. And on the right-hand side, here you can see the RNA scope signal, right? There's clear differences between the two signals. Both identify the bacillary forms, right? Specifically the Ziehl-Neelsen, you only see the bacillary structure. In the RNA scope images, what we see is we see the bacillary shapes, and then we see halos around the bacilli, and then we just see little spots, right? And I'll come back to that a little bit later. This is a higher magnification. Again, on the left-hand side here, you have Ziehl-Neelsen stain. You can clearly see the rods, and here you have RNA scope, and this is the blown-up picture. You can see there's diffuse halos here, solid states, and here we have a spectrum of this type of signals that we can expect. So this is in the plane of view here, and this probably is because the bacilli is vertical and cut through that plane. That's why you see the little halo. The cause of the halo is, we're speculating that it might be RNA that's leaching out of the intact bacilli, because sometimes you see a loss of this and you get these little blobs, which we believe is a disintegrated bacilli. And then you get the ultimate signals, the true signals, and I'll come back to that in a moment, which is these punctuated dots. So we tested it against an active TB case. So this is an open book cut of a TB lung. You can see the cavities there on top. You see these multiple tubercles. And when we stain that specific block, we can clearly see, well, there's intracellular. You can see the intracellular as well as extracellular bacilli. There's again intracellular. But when we look, and that's easy to diagnose, the feedback to the pathologist is very clear cut. However, when we look at a different tissue block from the same patient, right, it turned out to be Ziehl-Neelsen negative, right? And then appraisal becomes a problem, and the feedback of the pathologist to the treating clinician would be, oh, well, this is high incidence country, this is granulomatous inflammation, necrotic lesions, consistent with TB. And that's most of the pathology reports that we get. It's consistent with TB, and it's automatically assumed it is because of TB. And in this particular case, it is. This is an example. Again, Ziehl-Neelsen negative. I'm just going to focus on this one area. And if you look at it, you see all these punctuated dots, right? Thousands of them. Some, not all granulomas have these punctuated dots. Some granulomas contains absolutely nothing. And in addition to that, we also found that the RNA, for some reason, actually localized to the cytoplasm, it's perinuclear, right around the nucleus. And this was a very interesting observation. Not all cells have them, but some cells, I would say maybe less than 1% of the cells do contain them in the apparently normalish airways, as well as in the consolidated airways, including necrotic lesions, granulomas. And this is reminiscent of a study published in 2000 by, was it Lancet by Graham Rook, where they showed the same thing happens to TB DNA. TB DNA accumulates in phagocytic and non-phagocytic cells. In this particular case, we show that the TB RNA, for whatever reason, is accumulating. We couldn't distinguish or identify clear evidence of a bacillus inside the cell. And we find the presence of these RNA in epithelial as well as in phagocytic cells. We also show that TB RNA is confined to very specific microenvironments. Here you have the bronchial epithelial layer, RNA scope is just like loaded with it. But again, not every airway is loaded with TB RNA as expected, right? The moment you have a signal everywhere, then that becomes a problem in itself. So this was in this one particular airway, it was just loaded. And when we stain with antigen 85, we get positive signal for antigen 85 as well. So that's good independent evidence that TB at some point in time resides there. We don't see actually bacilli in the bronchial epithelial layer, but we do see a TB RNA. And we've 2 years ago, we already also published and actually show that TB can cross the bronchial epithelial layer. This is the upper airways, right? You can see them inside the lumen, then crossing the bronchial epithelial layer into the interstitium. So it's not impossible. Pathologists typically don't look at the upper airways. When we inspect this a little bit, we look at it a little bit closer. Again, this is the seminiferous vesicle. This is the Angin 85 stain. So we develop a dual Ziehl-Neelsen Angin 85 stain. So Angin 85 is a loosely cell wall associated major secreted antigen or cell surface excreted antigen. What we immediately notice, we notice the pink cells, right? And here you can see the brown bacilli, which stain positive for Angin 85, but there's a lot of negative cells. And we've done this over and over. So there's basically two populations that exist, two phenotypically distinct populations. Why some of them are positive for Angin 85 and why some of them are negative, we don't know. It's certainly not because of differential gene expression. Sometimes you have two bacilli across each other, and the one is positive and the one is negative. Whether that's an indication of live cells or dead cells, we also have no idea. I'd like to hear the audience's feedback on that. When we look at the cells closer, here's clear evidence that you have a positive Angin 85 as well as negative. There you have a couple of negative cells, there's a positive. And consistent with previous studies, we actually can see that the cytoplasm is actually stained positive for Angin 85. And again, several other papers have shown the same thing, right? So here we have a positive bacilli and positive host cell cytoplasm. Here is positive cytoplasm, but there you have a negative cell. So you get mixtures of cells. Mixtures of infected cells, Angin positive as well as Angin 85 negative. What that means, again, we don't know. This is about the only example that I could find where most of the cell was Angin 85 negative, uses Ziehl-Neelsen positive, and the one pole was Angin 85 positive. You either get them, they're either positive or they're negative. And again, whether that's an indication of a live or a dead cell, we don't know. So next we ask the question, well, what's the diagnostic or prognostic relevance of a technique like RNAscope? And we have this very interesting case from Alabama as a 61-year-old female. And she was, the doctors really couldn't diagnose her properly. At some point in time, they thought she had cancer, right? But 360 days, as well as 11 days prior to hospitalization, they did a CT-guided lymph node biopsy. And when they look at this biopsy, and there's the biopsy, well, there was clear evidence of granulomatous inflammation, and it was acid-fast negative, right? So she was eventually hospitalized, and a few days after hospitalization, a concentrated Bell smear was made, and that turned out to be negative in the Bell cells. The Bell cells were then also platelet out, right? Unfortunately, the day after they collected the Bell fluid, she died, and 3 days later, a post-mortem was performed. And then it turned out to be that she suffered from disseminated TB. TB in the brain, lung, liver, spleen, kidneys, basically everywhere, bone marrow, urethra, everywhere. And not surprisingly, 43 days later, these plates turn out to be positive for MTB. So then we ask the question, well, okay, is this an example where the healthcare system sort of failed the patient, but in all fairness to the healthcare system, there was no classic symptoms of TB, right? There was no reason to pursue this case further, but they did the standard assays, for example, staining for acid-fast bacilli. So this is a needle biopsy, turned out negative, Ziehl-Neelsen, for acid-fast bacilli, and then we apply RNAscope to the antemortem sample. These specimens, these needle biopsies that was taken while she was still alive, right, and compare them to the ones at the post-mortem tissue specimens. And it turned out to be, these are giant cells, and you can just see these beautiful speckles of TB RNAs basically everywhere. So if we applied RNAscope at that point in time to that patient, she would have been treated and most likely will be still alive today. So that was a surprise. I mean, it wasn't select areas. This whole needle biopsy was just, it looks like a Christmas tree light up. So we also independently confirmed this with Antigen 85. You can see the giant cells, these little red arrows there, that's actually bacilli, these like bacillary spots, Antigen 85 positive. You can see this giant cell is sort of weakly positive, but still positive. This one is strongly positive, and then you have the surrounding cells that's also strongly positive. This is true also for the bone marrow, very strongly Antigen 85 positive. And again, if you look at a postmortem specimen, as expected, they were also strongly Antigen 85 positive. We also confirmed this with eSTAT6 and CFP10 and some uncharacterized commercially available cell surface proteins. There is an advantage of using Antigen 85, but to detect TB, the actual bacillary shape using Antigen 85, I wouldn't say that's the best sort of approach. For some reason, you don't always get the nice clean, crisp bacillary shapes. You get some sort of blotches somewhere, and then you're not really sure how to interpret that. So the conclusions of this part of the talk is we believe RNAscope has prognostic and diagnostic potential as it detects bacillary shape and TB RNA in Ziel-Nielsen negative tissue specimens. The identification of TB RNA as Ziel-Nielsen negative antemortem biopsies provide evidence that RNAscope can guide therapeutic intervention. Now, RNAscope is not that cheap. It's about $450 to design the probe, and it's about $1,000 to design and generate the probe. But I mean, just think about next-gen sequencing, right? 10, 15 years ago, it was very expensive, and today it's $200. The dual Ziel-Nielsen Antigen 85 findings show that phenotypic distinct subpopulations of bacilli can exist in vivo, and accumulation of TB RNA is reminiscent of the previous study that I mentioned, published in Lancet, where they show an accumulation of TB DNA. Yeah, and the TB RNA and secreted antigens are within macrophages and non-professional macrophages. So the second part of my talk is studying active TB in human TB in whole lungs. And I would like to emphasize whole lungs because you might ask the question, well, why? So you can spread a lung over a tennis court. And imagine you take 1 centimeter by 1 centimeter, 4 microns. You look at only that piece in the lung, and you base your diagnosis or interpretation on that very small section. You lose context, right? So there's value in focusing and looking at TB and studying TB disease on the level of pathology, on the cellular level. But the disadvantage of that is, again, you lose context. In the context of the whole lung, that would be very difficult. Macroscopic approaches like X-ray and PET-CT scans is limited resolution and yield little cellular information, but it provides you an overview of disease progression. So there's pros and cons to both. And this is some of the published studies that we've shown a couple of years ago. We made use of micro and nano computer tomography. And these spots, these yellow regions, those are actually necrotic regions. We confirm that they are necrotic regions. And they're actually the TB granulomas, and it turned out that the granulomas, which we perceive as oval or spherical, turn out to be cylindrical. It's just because if you flip this F figure towards you and you cut it, you'll see a round circle. We also made the observations that the smaller the lesions, necrotic lesions are, the more oval and spherical it is. So it's not that all lesions are non-spherical. The bigger they become, the more cylindrical they become. Here we have other structures embedded inside the lesion. These are actually airways, and this is hemorrhage. So we really successfully apply this imaging tool to understand active TB lesions. We expanded it to a lung slice here. You can see this tubercles here. These are S, and just to make the point that context do matter. And this is the CT scan here. These red arrows, these are the necrotic lesions, and that nice structure that we see, this is this one here, with the white blobs, that is blood vessels, and again, these dark spots, those are airways. But I mean, if we perform a 3D segmentation of this image, we ended up with a diverse granulomatous structures, these lesions just demonstrating the diversity in granulomas, but in the context of that whole slice. So the red here, this is the blood vessels, the blue is the airways, and the yellow, that's the actual granulomas, right? So where there's a lack of airways, this area you see a preponderance of lesions, and in this area where you see an abundance of airways, you see a reduced number of granulomas, just making the point which we demonstrated in the paper that the granuloma shape is being dictated by the airways. The airways help shape the granuloma. And this is just sort of a side view to make the point here that these granulomas actually continue. It's just because we had a 1 centimeter cut here. So they're actually far more complicated than what you see here. So how can TB disease pathology contribute to vaccine development? I just have 1 or 2 slides. I'm not a vaccine expert, but one thing is clear, and we get this from the old literature and also our own studies in Durban. There's a lack of understanding on the differences between primary and post-primary TB disease. These disease pathologies are distinctly different. They can be easily identified, right? For example, in the case of primary TB, that's the first infection in immunocompetent individuals. And post-primary TB, it's a second infection. It was initially sensitized by primary TB infection, accounts for 80% of clinical cases and nearly 100% of transmission. Again, in primary TB, begins as a focus in any part of the lung, post-primary begins in the apex of the lung. And in the case of primary TB, it triggers modest inflammation, and it usually involves caseating lymph nodes. As a matter of fact, the old literature was very clear. There seems to be active replication in primary TB, but the immune system is capable of containing that infection. Not the case of post-primary TB. People die of post-primary, most people die of post-primary TB, right? Where you get infection triggers severe inflammation, caseating and necrotic lesions, and cavitation, and ultimately death. So there's major difference between primary and post-primary. And you think about the vaccines, BCG is effective against dissemination in children. Dissemination in children, right? So the question is, are we really, do we have the right guidelines for developing good vaccines? And it's my view that I think the differences between primary and post-primary can help guide vaccine development. And the pathology and radiology of early TB is also significantly different from post-primary TB. And this is just to illustrate, and we published this. This is a good example of post-primary TB, right? Massive cavity there, massive lung destruction there. This is the CT scans here. Whereas in the case of primary TB, you see this patchy or flocculent densities. You get calcification of the lymph nodes, which you may or may not detect. Hematogenous dissemination of bacilli via the vascular is quite characteristic. If it resolves, you may end up with a latent TB. So there's massive differences between primary and post-primary TB. And unfortunately, the study of TB pathology is very few people are studying it. So to conclude this part of the talk, micro-CT, computer tomography can characterize, do I have 2 minutes? Okay. Can characterize a wide range of lesion types. CT can reveal hidden information hidden from histology. Incorrect conclusions can be drawn from histology without knowing the 3D context. Lesions develop along the airways. The complex shape of the lesion will affect distribution of drugs and nutrients, and lesion heterogeneity likely triggers different immune responses, and it might have implications for aerosolized drug delivery. We believe so. So, oh geez, I'm not going to make it. Well, I'm going to cut some slides. I want to show a couple of slides on how to identify TB lesions. There were studies in the 1920s, 30s, and 40s. They were clear-cut evidence for latent lesions. Three lesion types were identified: fibrotic scars, fibrocaseous lesions, as well as necrotic lesions. And in the old times, and I'm talking about the 1920s or 30s, the only way to study latent TB lesions was through postmortem tissue. And close to between 80 and 89%, 88% of people in South Africa actually has latent TB, right? So it's a rich source of information. So again, how do we study it? Based on our CT findings of active TB lungs, we know that we can use calcification as a proxy for TB lesions. And this is a 27-year-old female with had a head injury, died from a head injury. Postmortem specimen, we use our CT-guided platforms. We identify lesions, and there we end up with a lesion, a fibrocaseous lesions with partial calcification that's very consistent with actually active TB. But this is in a perfectly healthy individual who died of causes other than TB. There you can see The lesion, the hemorrhage here is very consistent with the last gasp during death, mild fibrosis consistent with the age. And if you look at this distribution here, clear evidence of fibrosis, necrotic area as well as calcified areas. And I would just want to make the point here, active TB lesions is an indication of a failed immune response. We know quite a bit about that. Latent TB lesions actually a successful immune response. Okay, there's another lesion. I'm just going to skip over this. This is a lesion inside a blood vessel, actually pretty well described in the old literature, and you can see this epithelioid histiocytes there. And this is a very interesting case. We were hoping to find this lesion here, which turned out to be the latent TB lesion, but it got dislodged from the tissue, but we saw this signal. We weren't sure what this was. So I want to ask anybody in the audience, do we know what this is? Can you yell out? Turned out to be schisto or bilharzia. It's quite prevalent in Africa. And I sat on the plane and I actually count the number of schistos, it's very consistent. So this is an added value of using imaging to the study of TB. You end up actually with something like this. And we actually found some of these schisto eggs in TB granulomas as well. So I'm just going to cut my talk short. We believe that imaging can be used as an effective scouting tool to identify and characterize TB lesions. We can identify latent TB lesions in the current age. So far, all the post-mortem specimens that we look at contain latent lesions, and that's 20 lesions. Okay, and I'm going to just move forward, and I'm just going to skip that. Yes, I'm sure Anil would have appreciated the talk on bedaquiline, but we can talk later. I just want to thank the funders for paying the bills. Thank you very much.
My question relates to the first part of your talk. It was relating to the RNAscope. I had a couple of questions about that. That the RNA, as we all understand, is quite unstable. So you have to make a choice of the kind of target that you are going to be looking at. And secondly, would you see how that impacts the detection of antigen 85, which would be a protein and hopefully more stable, as compared to an RNA, which would have different stability parameters? And last third thing would be, would you see RNA in latent TB lesions?
Yes, oh, very good questions. And that's usually the first question I get, RNA stability, right? So inside a living cell, RNA stability becomes an issue because you have a single DNA copy and you might have 100,000 RNA transcripts, right? So the level of transcripts has to be regulated by RNases, right? That degrades RNA. And that is true inside a living cell. Outside a living cell, that's a different story. So making the point that extracellular RNA might not be that unstable simply because there's no RNases. And when we do the eukaryotic RNA scope, we sometimes see extracellular RNA as well. So that's a very good point. When it comes to antigen 85, I think antigen 85, I think they should complement each other. Antigen 85 complement the RNAscope study. This RNAscope target is actually DHFR, a sort of housekeeping gene, not the best target. We've generated more probes, the DOS Regulon probes, S and T. We've generated probes against that too, but that was the one that was commercially available, and they couldn't give us any particular good reason why it was DHFR, but it's a good housekeeping gene. So yeah, in a nutshell, RNA is unstable inside the cell, outside the cell, I'm not really sure. And again, it all depends how RNases degrade the RNA, because you can, if you have a single probe pair bind to RNA, that RNA can be very short. But again, this is entirely speculation.
Thank you.
Hi. So again, related to RNAscope, like you mentioned, you're using DHFR as one of the targets. So is it the detection is only for a single RNA, or it's detecting multiple RNAs?
No, it's just single DHFR RNA.
Okay. because the RNA that you're detecting outside and in the host cells could also be some of the small regulatory RNAs that have been shown to be secreted out of the cells and regulate the host transcription. So it all depends on what is the target that you're detecting. But if it's a single RNA that you're detecting,
Well, we have, what I didn't show is that we have a whole battery of positive and negative bacterial RNA controls as well as host RNA controls. So, and one of the features of the RNAscope, it is extremely sensitive and extremely specific. So we don't have any doubt about the specificity, if that answers your question, but we can have a conversation afterwards.
Hi, Idris. This is Sheetal from IGIB. So I wanted to make a comment about the Zeal-Nielsen stain. So acid-fast bacilli can also be detected by CyberGold, and this is work from Anne Linnett's group. And
By what, sorry?
So it's a DNA dye called CyberGold.
Yeah.
And that actually works very well. So Anne Linnet showed it in the Kramnik mouse model, and we've basically been able to replicate it. You can get beautiful fulminant necrotic granulomas with that. So wouldn't that be a cheaper approach than RNAscope? So have you compared that head on?
Yeah, well, we haven't done the comparison, and that requires a totally different study, but it's a very good question. And I know there's a couple of dyes and we've used some of them, but I mean, maybe some of my clinical colleagues can maybe comment on that. So the Zeal-Nielsen is sort of one of the standard approaches in most clinical laboratories, although despite its many deficiencies. Yeah, so that's basically my comment. I think in animal model systems, when you look at lesions, they typically filled with TB bacilli, right? But that's not the case in humans.
So not with Zeal-Nielsen stain. You can't detect them all with Zeal-Nielsen, but you can detect them with CyberGold. They're both based on acid-fast staining properties. It's just that you need a fluorescent microscope for one, and most labs would not have, diagnostic labs would not have.
Yeah,
so.
yeah.
But I just suggest that maybe compare it head on. It might be a cheaper option.
Yeah, no, I fully
Thanks.
agree.
Great.
No, thank you very much for that.
Okay, that's the last question.
Very brief point, Adri. So you mentioned about animal models. It's possible that we may be looking at these lesions in the animals 3 months after infection, not 3 or 30 years. So that may have something to do with it. But going back, I wanted to make a comment about RNA stability. I think granulomas, particularly evolved granulomas that have been around for years, are metabolically of low activity, and RNA may actually live longer in that environment.
That you're absolutely right, Deepak. And maybe let me just come to Jaya. I didn't answer your question fully right. So those latent lesions actually was confirmed. The tuberculous origin was confirmed with RNAscope and Angin 85. Thank you, Deepak.
Thank you very much, sir, for your far-reaching insights on how vaccine and drug discovery are significantly influenced by TB lesions and the underlying pathogenesis. I now request Dr. Medigeshi to felicitate Dr. Stein. Thank you, Dr. Stein and Dr. Medigeshi.
So he's chairing a different session, right? Okay. He's not the chair, right?
Moving to the session 3, understanding the biology of TB pathogen per se and how it operates in the host has a huge implications on how end TB could be achieved. In the next 2 sessions, that is 3 and 4, a total of 10 speakers will illuminate different facets of basic biology of TB pathogen. To chair session 3, we have Professor Anil Tyagi and Dr. Valakunja Nagaraja. I request both of you to be seated on the dais. Professor Tyagi is the former Vice Chancellor of Guru Gobind Singh Indraprasth University, New Delhi. At the University of Delhi, he was the first to characterize mycobacterial transcriptional signals in detail. He has developed several candidate vaccines for TB. During the span of last 3 decades, he is honored with multiple awards, including the Shanti Swarup Bhatnagar Award, the Ranbaxy Research Award, and the Vigyan Gaurav Samman Award. He is an elected fellow of all 3 Indian Science Academies. Professor Nagaraja is an honorary professor both at Indian Institute of Science and Jawaharlal Nehru for Advanced Scientific Research, JNASR Bengaluru, and has formerly served at JNASR Bengaluru as the president. His primary interests include understanding mycobacterial gene expression and their regulation and DNA protein interactions. He is a fellow of all 3 science academies of India and the World Academy of Sciences, TWAS. He is a recipient of several awards, including the Shanti Swarup Bhatnagar Award, Ranbaxy Award, J.C. Bose Fellowship, and Sir Vishveshwaraya Senior Scientist Lifetime Achievement Award. We have 5 talks in session 3, each for 20 minutes. Following the talks, there will be a brief Q&A session with all 5 speakers. I request the audience to hold their questions until then. I urge the speakers to please stick to their time. The buzzer will ring at 18 minutes. I request the speakers after their talks to be seated on the dais for the Q&A session. I request the session chairs to kindly lead session 3.
You can go ahead. So just one small announcement. Anybody who has yet to still put the poster on the poster boards, please do that right away. Thank you.
So we have next session on basic biology of TB pathogen. We have 5 speakers. Every speaker will have 20 minutes to speak and 5 minutes for question answers. But all the talks will be delivered first, and you are requested to hold on to the questions till the end of the session, when you can ask any question to all the speakers who will be there on the dais. So we start with the first speaker. Dr. Balasubramaniam Gopal is a professor at Division of Biological Sciences, Indian Institute of Science, Bangalore. His research interests include gene expression analysis in bacterial pathogens and more recently, synthetic microbiology. In addition to his academic affiliation, he has been associated either as a co-founder, advisor, or consultant to startup, mid-sized biotechnology and pharmaceutical industries involved in the production of biosimilar and active pharmaceutical ingredients. Dr. Gopal is a fellow of science academies in India and a recipient of prestigious Shanti Swarup Bhatnagar Prize of Government of India. I invited Dr. Gopal to deliver his talk.
Oh, great. So thank you, Anilji, and thank you, Professor Nagaraja, for and the audience to be around. I'll take about 12 to 14 minutes max. And so I've got 3 short stories to give you about what we've been into when we've been working with the transcription mechanism. This is something that we've been involved in over the past 2.5, 3 decades or so. And so this gives you a feel as to where things are. So this particular topic that I'm going to focus on today is about the primary sigma factor. And this in some sense is something that we got enthused to working on by one rather blunt question that Anilji probably would recognize that he asked me several years ago. Why are you working on all these ECF sigma factors, which are non-essential, which are orthogonal, and which have functional redundancies? And why don't you focus on something that's absolutely important for survival? So that got us to the first point, and which is why I began collaborating with Minay Nandikuri. And so this particular talk that I'm going to give you is actually work from 3 labs. mine, which is based in Bangalore, Vinay, who was earlier in NII and now in CCMB, and Sandeep Sundrial, who's a synthetic organic chemist and who's helped us move through this. So what am I talking about? I'm a structural biologist. We are rather simple-minded creatures. Huh? So we take enzymes or proteins and then talk about whether they are structured and whether they are not, and then try and see if we can understand some biology on the side. So in terms of SIGE, what are the questions? There's going to be parts which are structured. There are going to be parts which are unstructured. Does not having a structure means not being functionally relevant? And that's the focus. So I'll start. So this is the basis for where we go. I'm a physicist by training, and you can see it here. See, what we have been talking about is the RNA polymerase complex. There are about at least half a dozen, if not more, fully characterized structures. So then the question here is, what do we know now that we didn't before we saw all these structures? And what's it that's important? The first thing that comes across is the mycobacterial RNA polymerase is actually a structural biologist's dream or a nightmare, depending on the way you look at it. Why? There are way too many variations for you to make any generalities. The first, you've got an insertion, a sequence, a taxon-specific insertion in beta prime. It's not there in the other prokaryotes. The second, the complex by itself is inherently unstable. For people like you and me as enzymologists, what it means is that nature wanted several other factors to come in to build an allostery and regulatory mechanisms within a conserved system. The third, of course, is the omega subunit, which one hardly if ever sees in these structures, but that's important in the folding apparently. And then the fourth is of interacting proteins. And as Vinay's group promptly pointed out to us, there are several more that we had missed when we started this work. One part which I'd like you to think about. When you look at this complex, it now follows this particular way. It's now have the RNA polymerase and it goes and has to recognize an initiation factor. It forms the first complex and then comes the interacting proteins. You have CAR-D, which increases the stability by a factor of 10. You have RBPA, which increases the stability by a factor of 4. Put two of them together, you increase the stability by a factor of 25. So you now need to look at how all of these work in tandem. And then once we start off with this particular complex, I'll tell you what is it that got us excited. The first part, does not having a structure mean not having a function? So this was what excited us here. So when you look at the group 1 sigma factors, the primary sigma factors, these have a long N-terminal extension. Again, not conserved. Take E. coli, it's about 92. Take B. subtilis, it's about 87. Take M. smegmatis, it's 163. Take M. tuberculosis, 221. Why did nature want such a long polypeptide when it apparently couldn't be seen in any of the crystal structures or the cryo-EM? Was it important? And that's what started us going. And look at it in terms of sequence features. Now, this is something that as bioinformaticists and structural biologists, one begins to look at and then try and make some sense of things. And for several of the younger people in the audience, you might be enthused with this idea of AlphaFold and ChatGPT, which apparently makes these nice models. Are these there in any models? Again, no. One thing that comes in is something that we have begun referring to more recently as microdomains. These are the ones which are intrinsically disordered and apparently play a role in liquid-liquid phase separation. I'll come back to that if I can. So this is how Vinay started off with this dissection mechanism. You have 221 residues in the N-term. Try and chop it up into smaller bits and ask, do we need it? What was the basis? SIG-A alone has this extension. SIG-B, which is also principle-like, does not. Both of them are active. When you look at the transcriptome data, this again comes from Vinay's group, SIG-A and SIG-B typically have about 60% of RNA-seq data, which overlaps with each other. 40% is different. The third bit of interpretation. Take this tail, the 221, stitch it onto SIG-B. Does it work like SIG-A? Again, it doesn't. If you deplete SIG-A, does SIG-B take over its role? Nope. Again, you have issues with survival and you have issues with even with the way the pathogen behaves. So there was something unique about this N-terminal which we wanted to understand. Here's the initial set of experiments. And what you see out here is One of the variations that we've done over the past few years is to now begin adding all these other interacting proteins, because that's typically what makes all the difference. So one of the things that comes across is once we start making these small bits out of the N-terminal parts, chop it up into smaller half segments, and then ask a question, are they still active? It so happens that only one small part between residues 132 and 179, just remember the numbers for a minute, 132 and 179, mostly acidic, and if you remove that, you typically have a very sharp fall in activity. What does it mean? So one of the things that comes across for us when you look at structures or molecules per se, is look at these different domains. In terms of MTB, there's no sigma 1. That's very difficult to define. There's no sequence pattern. Are we actually staring at it? So what Vinay then did is then ask the same question from what he had inside the cell. Now, what this particular slide summarizes is about 3 years of Biplab Singh's work, where essentially he managed to achieve a SIG-A depletion phenotype, made SIG-A with different flag A, with different N-terminal parts which are depleted, and then ask the same question, what are the parts which are important? Now, one of the things which comes across is that the same small acidic patch, 132 to 179, that makes all the difference for SIG-A to be SIG-A and not be substituted by SIG-E. Does this make a difference in the way it interacts with the promoter? Now, this is again something which I thought was worth worrying about. Because in some sense, when you look at the structured part of a sigma, now this is something that all of us assumed was always going to be the case. You had SIG2, which interacted with the Pribno box. You had SIG4, which interacted with the -35. You had SIG3, which now had an interim region. And then sigma 1.1 and 1.2, which essentially also played the role of trying to remove the nascent RNA polymerase from making an unproductive complex. Textbook stuff. Was that indeed the case? The first thing that we've realized out here, you remove the N-terminal polypeptide, you see differences in terms of DNA binding and recognition. So probably there's a 1.1, 1.2 there. We don't know whether it's that for sure. The second part which comes in, just focus on this for a minute. Post-translational modifications. Now, Adri, who just spoke before me, spoke about antigen 85. Now, that's also a classical case of post-translational modifications altering the expression of genes, and for which we know very little. The important thing which comes in here, all the phosphorylation events happen in the same acidic patch that I spoke to you about, the one that's important. So that's serine 57, a threonine 69, and a threonine 71, all located here. Something that's never seen in structures, but apparently all the phosphorylation happens there. The second link that maybe that's important. Again, this data that comes in from our side and from Vinay's lab is not terribly clear. You make phosphoablative and phosphomimitic mutants. Do they make a difference? Marginal. You have about a fold increase in DNA binding, not great. What do they do in terms of the RNA-seq data inside the cell? Are they making all the difference? Again, the thing is out because it does make a slight difference, but not strong enough for us to say, yes, phosphorylation alone is a major governing factor. Then what is it that makes a difference? So then we said, if this particular N-terminal patch has all the post-translation modifications, remove it, you can't see transcription happening. Does it have a role, if at all, in terms of other interacting proteins? So this was another bit of work which Vinay got. And this is Biplab and Debashree's data. Debashree is out here in the audience, and you could probably ask her a little bit more about all of these. So this slide summarizes another 4 years of work. So there were pull down experiments, and then you make each one of these proteins and then ask whether it's bona fide. Here comes the catch. You have structured regions, unstructured. Remember, that's what I wanted to talk to you about. Can the unstructured parts also interact, or is it the structural regions alone? See, RNA polymerase as an enzyme has this interesting bit. So what it does is you have the C-terminal, which is there for the YB and the other proteins, the sigma-2, which is what makes a role in the RBPA, and the CAR-D now plays a role in the ternary complex. So what we now find is a couple of bits of data, which is still work in progress. One of the pull downs we get is of an IF3, an initiation factor, and then you have components of the ribosome, S6 and S9. What are they doing interacting with SIG-A? What we now figure out in the in the bits of data is, in effect, what they do is to dampen out the amount of free Siga in the cell. So what we always thought was a trait that was used in other systems, toxin, antitoxins, sigma, anti-sigmas, what we see are interacting proteins also playing a similar roles, even in the case of the primary sigma. So probably nature had something interesting. A last quick bit about the structured components. The unstructured parts, what have I said? What we've noted now is they are important, even if they are not seen in the structures. What about the structured components? Bioinformaticists like us, again, begin looking at the apparent needle in that haystack. Align 1,000 sequences. Can you see if something's sensible? So one of the things that comes in now that we have the benefit of so many structures is to ask, is there any similarities at all? After all, it's the same RNA polymerase apoenzyme. All of them have to bind to the sigmas. So is there something in the surfaces that could make some difference? And so when you look at some of these regions, we did find some of these sequences which are apparently conserved, structurally conserved components, and not obviously apparent when you can look at the sequences alone. When you look at the structures, it does seem to make some kind of sense. They are involved in important salt bridges, and probably one could probably take it forward. Indeed, this is what we see. So this is Twinkle's data, and hopefully before I grow substantially older, we'll land up publishing it one day. So here what we've done is looking at all of these mutants, and we figure out that just making these point mutants essentially stops transcription. So this becomes a site, we argued, for making molecules that can modulate enzymatic activity. So structured part now land up being modulators. Then comes in Sandeep Sundariyal's work, where he's taken up this particular part, which is structured between the beta prime and the sigma. And what he's been able to do is synthesize some of these compounds, and he has a couple of posters and of course we are there to chat with you in case you are interested. And he's synthesized an entire library. At this point of time, we have about 19 odd compounds. And we found that a couple of them, and this is something which Anilji would particularly like, because if you remember, one of the meetings that I used to present earlier, and you'd ask, show me something that works, that might not be a bad idea. So there's something which looks like it might work. Of course, there are caveats, as Nishit would agree, is we have intracellular permeability, which is still an issue. Caveat emptor. But the fact remains that you can use this as a strategy for non-competitive modulators. I wouldn't call it inhibitors yet. A summary. So what have I learned in this last 5-6 year journey with just SIGA alone? The unstructured components matter, which means that even if you had the structures, even if you had 7, 8, 9 cryo-EM and crystallographic structures, there are still substantial unstructured bits which remain out for open. What's also obvious, this is actually a beautiful synthetic biology toolkit that nature had so many different sigmas, all optimized for a different function. Just use it, plug it into an RNA polymerase, and you have set to go. So that's the first thing. The second is, this is work which is in progress, and I'll agree, and I'll be the first one to admit that we do not know the full part of all this phosphorylation part. Why is it happening in the intrinsically unfolded regions? Does it have a role in terms of getting an unproductive holo RNA polymerase complex to move away? Does it have any other significance in protein-protein interactions? The answer is still, we are still looking at it. Protein interacting to the principal sigma factors. Now, are they moonlighting anti-sigmas? That's probably the simplest explanation. Do they have a role in transcription translational coupling? Again, the jury is out. The fact that you have initiation factors, the fact that you have RNA ribosome components, that's still work that needs to be looked at. And then, of course, is something which we find particularly exciting, and that's about sequence structure patches that essentially land up being critical components or for the design of modulators. And so this is something that would probably keep us active probably for a few more years. So I'll end now. What we've been doing is started off with a Wellcome Trust grants way back a couple of decades ago, and it's been funded mostly by the SERB and the DBT. And this is work which Twinkle did for her PhD. Hopefully we'll publish, and Amit. Debashri is here in the audience, and she's been the person working primarily on the intrinsically disordered part and some parts on the phase separation, which I didn't talk to you about. And Biplab and Abhishek are from Vinay's lab, and I believe they might have a poster too. And our collaborators are Rajesh Gokhale, with whom we worked on SICK, and that's when we got into the antigen 85 and the other bits, Vinay Nandikuri and Sandeep Sundrial. And of course, whatever I did because I work at IISc. Thank you for your attention.
Thank you, Gopal. Please come. Our second speaker, Dr. Smriti Mehra, is an associate professor at the Disease Intervention and Prevention Program at Texas Biomedical Research Institute at San Antonio, Texas, USA. She also holds the position of Research Support Core Co-Lead and Core Scientist at the SNPRC Texas Biomedical Research Center and is an adjunct professor in the Department of Microbiology, Immunology, and Molecular Genetics at UT Health San Antonio, Texas, USA. She focuses on her research on the testing of a prime boost vaccine against TB non-human primate models, investigating the role of endolamine dioxygenase in tryptophan metabolism and modulation of the Th1 response, identifying M. tuberculosis genes that are essential for granulomatous survival and persistence, and identifying transcriptomic and epigenetic signatures that define protective versus non-functional granuloma and aid in biomarker discovery. Dr. Mehra has published more than 70 research articles in peer-reviewed reputed journals. Throughout her career, Dr. Mehra has been a member of several professional societies and national panels. Thank you. Please, Smriti Mehra. Yeah.
Good morning, everyone. I would like to begin by thanking the organizers for giving me this opportunity to present my work. So my lab is focused on TB granuloma. Granuloma, as you heard before, is a pathological lesion formed in response to MTB infection of lung macrophages. This leads to recruitment of neutrophils, which are the initial cells that surround the granuloma, that comes to the lung. And then there is chemokine cytokine. So then there is a chemokine cytokine storm, which leads to recruitment of first innate and then adaptive immune cells. These cells then surround the infected macrophages in a circular manner, forming solid granuloma. The central region starts becoming necrotic, and over course of time, it becomes caseous. How well this granuloma contains MTB will determine if the MTB will become latent or will it become, the granuloma will cavitate and disseminate MTB into the lung. So over the last decade, my lab has used NHP model of TB to look at endolamine dioxygenase or IDO. IDO is a key enzyme in tryptophan catabolism pathway. Tryptophan, as you all know, is an essential amino acid. So what we have seen in the publications that we published, that IDO is expressed, IDO mRNA is expressed at 100-fold higher level in animals with active TB, in the lungs of animals with active TB as compared to control animals. So this is a confocal image of a granuloma from our active TB animal, showing IDO expression, shown here in magenta color, at the periphery of the granuloma, which is outer to the internal core of the granuloma, but is still internal to the lymphocytic layer, shown here in blue. We have also shown that IDO expression occurs across multiple species in an MTB-dependent manner. So we have shown that in both our murine model of TB and macaque model of TB, IDO is expressed in active TB animals. And also in a recent publication, we showed that IDO expression occurs on interstitial macrophages and myeloid-derived suppressor cells. So as I said previously, IDO is a key enzyme in tryptophan catabolism pathway. It's a known immunosuppressor. IDO expression is induced by interferon gamma on antigen-presenting cells, results in breakdown of tryptophan to kynurenine, and it stops T cell maturation either directly or via maturation of M2 macrophages. In a seminal study conducted in the lab, we wanted to see the effect of D1-methyltryptophan, which is a known IDO inhibitor, on MTB progression in our NHP model of TB. So we use D1-methyltryptophan. It's a structural analog of tryptophan and competes with it to bind to IDO and prevents its activity. So we infected macaques with MTB, divided them into two groups. The control group, as shown here in gray color, and the second group received D1-methyltryptophan starting from week 1 post-MTB infection. It's represented here in burnt orange. To see the effect of IDO inhibition, we did kynurenine tryptophan ELISA on the plasma samples from both group of animals. And as you can see here, at week 3, control animals had significantly higher KT ratio as compared to the animals that received D1-methyltryptophan inhibitor. So this is week 3 of post-MTB infection, 2 weeks post-D1-MT treatment. We also stained BAL cells for kynurenine. So as you can see here, in our D1-MT treated animals, there are very few kynurenine positive cells as compared to control animals. At clinical level, the animals that received D1-methyl treatment did much better as compared to control animals. So they had significantly less bacterial burden in the lungs at time of necropsy and less involvement of lung in lesions, or there was less pathology in case of D1-MT treated animals as compared to control animals. We also saw higher recruitment of CD4 central memory T cells, which were more proliferative, as shown by Ki67 staining in the D1-MT treated animals as compared to control animals. And transcriptomic analysis showed that in case of IDO inhibited animals, there was higher expression of T cell stimulatory pathways and tissue remodeling pathways, while in case of control samples, there was higher T cell inhibitory pathways that were expressed. As I showed you earlier, IDO expression occurs in the granuloma, inner to the lymphocytic layer of the granuloma. So we wanted to see what is the effect of inhibiting IDO activity at the level of granuloma. And we hypothesized that IDO is acting as a barrier, preventing T cells which are present in the lymphocytic layer to enter the core of granuloma and kill the bacteria. So this is a pseudo image. We divided the granuloma in 3 layers. This is a pseudo image to show the layers clearly. So the inner necrotic center is in red, macrophage layer in blue, and the lymphocytic layer in green. We stained the granuloma from both group of animals for CD3 and IDO. And we saw that although the number of T cells present at the outer periphery in the lymphocytic layer were similar in both group of animals, but we saw higher penetration of T cells in the necrotic core in case of D1-MT treated animals. So this is a magnified image showing that in case of control, if there were 3 CD3 positive, Ki67 positive cells present in the core of the granuloma, in case of D1-MT treated animals, there were at least 20 CD3 positive, Ki67 positive cells. So not only did IDO inhibition lead to better clinical outcome, but also it led to reorganization of granuloma, and there was better penetration of T cells and better killing of MTB taking place. So we did another study where we wanted to see if we can combine D1-MT or IDO inhibition with TB therapy and uses a host-directed therapy approach. We infected macaques with TB, divided them into 3 groups. One group was control. Second group received moxifloxacin ethambutol or ME therapy, shown here in blue, starting from week 7 for 12 weeks. And the third group of animals received, along with ME, also D1-MT treatment for 4 weeks. Then we looked at the clinical parameters. And as you can see here, the animals that received D1MT and ME, shown here in pink line, did much better than the control animals. They did not lose as much body weight as compared to control animals represented by black line. And they also had significantly less involvement of lung in lesions and less bacterial load, as you can see here, as compared to control animals. We also looked at the sterile lobes, total granuloma lobes that were sterile in D1MT ME group and ME group only, and also the number of sterile granulomas in both group of animals. And as you can see, in case of D1MT ME groups, there were significantly more sterile lobe or significantly more sterile granulomas present as compared to ME alone group. So this study shows that we can use D1-methyl tryptophan or any IDO inhibitor in conjunction with TB therapy as a host-directed TB therapy approach. We recently characterized transcriptomic, lung cell transcriptomic at single cell level using our lung samples from our animals that were controlled or had uncontrolled TB. That means they had active TB or they had controlled TB or latent TB. We use canonical markers to identify various myeloid cell populations present in these 3 lung samples. Among the different families of genes, different families identified included alveolar macrophages. So the CD163 positive, MRC1 positive cell population. There were interstitial macrophages, negative population, and also a subset of interstitial macrophages that carry signature of interferon signaling. So these population had higher CXCL10, CXCL11 on IDO expression, as you can see here. So this confirms our previous observation that IDO is expressed on interstitial macrophages that recently have arrived from blood into the lung and carries markers of inflammation. Since we first showed the IDO expression in our active TB animals, others have also shown the relation between TB and IDO and have confirmed our observations. So in a study published by Shi et al., they showed that the plasma from patients that had drug-resistant TB as compared to patients that had drug-sensitive TB, and these patients were on TB drug therapy, had higher concentrations of kynurenine and other downstream metabolites in plasma. And in a study published by Collins et al., they also showed that the patients from South Africa, the plasma samples from patients from South Africa had higher KT ratio if they had active TB and HIV positive as compared to control patients that do not have active TB. So multiplex ion beam by time of flight, or MIBITOF, is an innovative technology that enables high-dimensional single-cell imaging in situ of tissue specimen. In MIBITOF, we label antibodies and then stain tissue sections with these metal label antibodies and read out their abundance and localization using secondary mass spec. So this results in a multi-dimensional image depicting subcellular location, expression, and localization of dozens of distinct proteins. Using MIBITOF, Dr. Michelangelo's group at Stanford saw that in human TB granulomas, IDO1 was one of the two of the highly expressed proteins present in human TB granulomas, along with PDL1. So they concluded from their studies that human TB granulomas are highly immunosuppressive. They have highly immunosuppressive environment, and there are active processes going on there that suppress T cell progression and T cell responses to MTB infection. We started a collaboration with their group, and we used our historical samples from D1MT treated animals and control groups and did MIBITOF analysis to look at IDO levels in both of these animals. And as you can see here, this violin plot shows that in D1MT treated animals, there was less IDO expression as compared to control groups. We also showed that there was no change in difference in CD4 T cell recruitment in lungs of both group of animals, but it was CD8 T cells that were recruited to higher number in the lungs of B1 empty-treated animals as compared to control animals. So this is a confocal image showing that there are more CD8 granzyme B positive cells present in the lungs of IDEO inhibited animals as compared to control animals. So these CD8 T cells were not only recruited to higher number, but they were more proliferative in nature, as shown by Ki67 staining in D1 empty treated animals, and there was more interferon gamma production. And this indicates that these were antigen specific, activated and proliferative cells. And this CD8 T cells were better able to penetrate inside the myeloid core of the granuloma in treated animals as compared to control animals. There was no change in CD4 T cell recruitment to the core of the granuloma, and they were better able to interact with macrophages also. So while NHP studies have advantages, but we cannot use them to do mechanistic studies. And a BLAC6 mouse phenotype, IDEO knockout mouse phenotype, failed to generate any TB specific phenotype. So we think it is because BLAC6 mice do not form any organized granulomas. We decided to generate IDEO knockout transgenic mouse in C3HEB FPJ or Kramnik mice model. We did a pilot study recently where we infected both Kramnik mouse, represented here in white, blue line, and the IDEO knockout Kramnik mice, represented here in pink color, with MTB. And as you can see here, we plated the lungs at different time points. As you can see here, there was at least 2 to 4 log reduction in bacterial burden in case of our IDEO knockout mice as compared to wild type mice. But there was no difference in granuloma formation in both groups of mice. We think it's because there is similar number of granulomas being formed in both group of mice, but there is better killing of MTB taking place inside the IDEO knockout mice granulomas. We are in the process of checking this hypothesis using antigen-specific flow cytometry and single-cell RNA-seq. So in conclusion, highly induced expression of IDEO in an MTB burden-dependent manner is a constant feature of TB granuloma in mice, non-human primates, and humans. Single-cell transcriptomic approach shows that IDEO expression mostly occurs on interferon-responsive inflammatory interstitial macrophages. IDEO induction in TB granulomas is highly suggestive of immunosuppressive function, which may allow MTB to persist. Inhibition of IDEO induction in TB granulomas in NHP leads to better control of TB and improve immune responses. Inhibition of IDEO induction in TB granulomas also allows CD8 T cells to access the granuloma core and increase their interaction with macrophages, as we saw from our MIBITOF data. Knockout of IDEO in mice with human-like granulomas reduces MTB burden, but not pathology. In the end, I would like to acknowledge the people who worked. Bindu Singh is a postdoc. Shivon was a graduate student in the lab. Dheeraj is a staff scientist, and Deepak Kaushal's lab and his lab members. SNPRC Imaging Core, Shashank Ganatra and Javier. Dr. Hall is veterinarian on the project, an ABSL3 tech group, and my collaborators in Washington University and Stanford University and the funding sources. Thank you.
We will move on. Our next speaker is Dr. Ashwani Kumar from IMTech. He's a senior principal scientist there. He's senior fellow of Wellcome Trust DBT India Alliance. and published several papers, 40 plus. His recent work, exciting work, is highlighting the contribution of biofilm formation in the lung and connecting it to virulence and drug tolerance in MTB. He has received many awards, including National Bioscience Awards, Swarna Jayanti Fellowship of Department of Science and Technology. Ashwani, over to you.
Yeah, thank you, Dr. Nagaraja, for that kind introduction. I also want to thank the organizers for giving me this opportunity. And to start with, I'm going to talk about Mycobacterium and where it resides in the macrophages, which seems like a very well-studied topic. However, I think it is not. I also want to start by thanking my students, Naveen and Shivani, who have done this work, and my collaborators, Mehak and Amit, shown here. Okay, so this is my lab. What do we do? Mostly sleep. Not really. We do work on a number of things. We, as Dr. Nagaraja has pointed out, we work on biofilms. We work a bit on microbiome and lung microbiome and how does it influence TB. We do a lot of work on redox sensing. We have published some work on autophagy and how does it help in killing mycobacterium. Unfortunately, we have been working on TB persisters. We have not been able to publish anything yet, and trafficking of MTB, and we have not published anything yet on this, but we believe we are doing some good work there. So as I said that TB, the localization of TB in macrophages is very well studied, and way back in 1970s, this is the observation that led to the textbook line that MTB is an obligate intracellular pathogen, showing that Mycobacterium tuberculosis is present in a phagosome, and they have studied a lot of electron micrographs and later on with the confocal microscopy. Mycobacterium tuberculosis is mostly an organelle bound. Later on, some literature appeared which suggested that it can escape phagosome also. Another interesting finding in early time was that live Mycobacterium tuberculosis, the virulent one, it resists the formation of fusion of phagosomes with the lysosome. And if you kill the mycobacterium, it is not able to resist that formation, the fusion of phagosomes with the lysosome. So this has been known for quite some time. And this is how we perceive the formation of phagolysosome happen. You have Mycobacterium tuberculosis, which is phagocytosed. And then there is an interaction with the early endosomes and then late endosomes. This leads to the fusion of the lysosomes with the phagosome containing mycobacteria, and then in the phagolysosome, mycobacteria may get killed and presented to the T cells. The antigens from mycobacterium can be presented to the T cells. But in the context of cell, where does this happen? That has not really been explored. Recently, a lot of literature has emerged on how the lysosomes, their function is modulated based on their subcellular localization. For example, the lysosomes, there can be two groups of lysosomes. One of them can be towards the cellular periphery, and these are the signaling hubs. They get signal, and there is localization of mTOR on these lysosomes. They get signals of growth, and they allow the cells to replicate and grow. Whenever there is a stress, these lysosomes can move towards the center of the cell, or towards the nucleus, and these are called perinuclear lysosomes. During this process, they lose mTOR, and then they come to inside and they perform this lytic function. So all the organelles has been thought, they are not really the same. Organelle may not have same function, but it depends on where in the space of the cell where it is. But similar thing with the phagosome is not known. How does the phagosome mature and where does it mature? That is not really known. And we were asking the question where mycobacteria localize. So mycobacteria in the macrophage cell looks like this. So there are two mycobacterial cells in this picture. This is the nucleus, one is close to the nucleus, one is slightly far away. So to say where the mycobacteria is in a cell, we can use the positioning of nucleus and give the relative positioning to the mycobacterial cells. And we can do this. It has been done for lysosomes. We can do this for mycobacteria by making these concentric contours. And you can keep on making them. And then you can say in which contour your mycobacterium is present and whether its presence there affects the TB pathogenesis or not. And the classical way of defining the spatial localization is to calculate the perinuclear index. Basically, you have this formula, it's very well published, and you can basically look for the presence of mycobacteria in each of the contour and then divide and figure out where it is. So if the perinuclear index is 1, it is very close to the nucleus. If it is -1, it is away from the nucleus. So we looked at mycobacterium, and this was an interesting experiment. These are THP1 cells, and you can see this is mycobacteria. This is the nucleus. And when you have heat-killed mycobacteria, this is mycobacteria very, very close to the nucleus. The live mycobacteria is away from the nucleus. We calculated the perinuclear index, and we found that the live bacteria tends to stay away from the nucleus, whereas the heat-killed mycobacteria quickly moves towards the nucleus. And if I analyze the same data in a different way, this is how it looks like. This is the nucleus boundary, and the live bacteria stays far away from the nucleus, whereas the heat-killed one moves close to the nucleus, suggesting that there is something that live mycobacteria does with respect to the spatial localization. And you all know this, there are 2 strains of mycobacterium that we often use in laboratory, H37RV, which is the virulent strain, and H37RA, which is the avirulent strain. And both the two has very different profile inside the macrophages. And we have also used the RD1 mutant strain. So RD1 secrete well-known ESET6 CFP10 virulence factors, and that also can modulate the phagolysosomal fusion. So we utilize these 3 strains, and as you can see here, the H37RV is in the M cherry, and it stays away from the nucleus, whereas H37RA or the avirulent strain is pushed towards the nucleus, and RD1 mutant is also pushed towards the nucleus. And you can see that in the perinuclear index calculation also. And we have also binned it in this way that you have 0 to 3 micron distance, and you will see that most of the avirulent strains here, H37RA and RD1 mutant, they move close to the nucleus, whereas the MTB is distributed away from the nucleus. And this also kind of suggests that the virulence of mycobacteria is really dictated by where the mycobacterial cells are localizing inside the macrophages. Then we asked the question whether if you infect with a single macrophage cell with both of the virulent and avirulent bacteria, what happens? Whether the mycobacteria which produce lipids, and these lipids are shed in the whole cell, whether it is influencing the localization of all the phagosomes, or it is changing only the particular phagosome in which it is residing. So as you can see here, H37RA again moved close to the nucleus. H37RV resists the movement of the phagosome towards the nucleus, and this is the distribution. The blue is the nucleus, green is H37RA, very close to the nucleus, whereas H37RV stays away from the nucleus. So these experiments suggest that Mycobacterium tuberculosis only modulate the phagosome in which it resides. It does not really change the positioning of all the phagosomes out in there. So we hypothesize that this is possible, that Mycobacteria enters the macrophages, and then Mycobacteria, the virulent one, which will resist the movement of the phagosome towards the nucleus, where there are a lot of lysosomes, there are perinuclear lysosomes. However, RA can move, and then the phagolysosomes fuse with it, and then it gets killed. So we asked if we inhibit this movement of H37RA or the avirulent strain towards the nucleus, does it really rescues it or not? And towards that, this is the machinery that is there for moving the cargoes. I am pretty sure that you all know this, but for the sake of the students, so we have polarized microtubules in the cells on which the cargoes move with the help of dynein motor. This is the dynein motor here, and the cargoes can have something like phagosome. It can also move the organelles. So we utilized dynein, siRNA, which is the motor protein, and inhibitor of the motor protein, cellubrevin. And as you can see that after inhibition of the dynein motor or knocking it down, the movement of the H37RA towards the nucleus was abolished. This abolishing of the movement led to decreased phagolysosomal fusion with the avirulent strain and also rescue of the killing of H37RA by Mycobacterium. So this kind of suggests that the virulence actually resides in controlling the spatial positioning of the phagosomes in which Mycobacteria resides. We did an opposite experiment. What if we can move the cargoes carrying Mycobacterium tuberculosis, the virulent strain, towards the nucleus and see what happens? So here you can see that we have used siRNA for ARL8. ARL8 is a protein which kind of is involved in anterograde transport, and it kind of pushes the cargoes towards the periphery of the cell. Knockdown of this has been shown that upon knockdown of this particular gene, there is a perinuclear clustering of lysosomes. So we reason that it may also be involved in the perinuclear clustering of phagosomes. And knockdown of ARL8B led to the movement of H37RV towards the nucleus. This is the perinuclear index. And this was beautiful movement because most of the mycobacterial cells were kind of localizing in the perinuclear region. And this leads to increased phagolysosomal fusion of virulent Mycobacterium. That led to increased killing of Mycobacterium. And trust me, this killing is better than what we get with interferon gamma. So just by moving or changing the spatial positioning of Mycobacteria, we were able to kill the mycobacterial cells. And now we know that you can have polarization of macrophages, especially with interferon gamma. If you polarize them to the M1 phenotype, then the blockade of phagolysosomal movement or the phagolysosomal positioning exerted by Mycobacteria, that can be overcome. So next we utilized interferon gamma and see whether that also changes the phagosome positioning in case of MTB infection. These are BMDMs, and this is the distribution of LAMP1. And upon interferon gamma treatment, most of the LAMP1 moved close to the nucleus, and this is the quantitation of the same. And we also asked this question with respect to MTB. So these are THP1 macrophages. Mycobacteria localizes away from the nucleus. The nucleus is here in this case, and Mycobacteria resides here, kind of far away from the nucleus. And when you treat the Mycobacteria infected macrophages with the interferon gamma, the Mycobacteria moves close to the nucleus. In this case, here is the nucleus. So the quantitation of that is given here. As you can see that without the interferon gamma treatment, Mycobacteria is primarily residing away more than 3 micron or so. But when you treat the macrophages with interferon gamma, most of the cargoes carrying Mycobacteria move towards the nucleus. And same is evident in this case, that if we inhibit the movement of the cargo with using the dynein siRNA, then the phagolysosomal fusion of cargo with lysosomes, the phagolysosomal formation is inhibited just by using dynein siRNA, which was not the case with the control siRNA. So these experiments suggest that interferon gamma also probably work through this mechanism of changing the spatial localization of Mycobacteria. And this is the quantitation of that, suggesting again that yes, interferon gamma work through this mechanism. So what is happening at the molecular level? So we have, so we know that TFEB is a gene which is a master regulator of lysosomal biogenesis. And we have in past shown that this gene is important for interferon gamma mediated regulation of TB killing. And this gene actually can move to the nucleus and it induces the lysosomal biogenesis. And as I said that we have earlier shown that interferon gamma induces a gene called as heme oxygenase 1, which produces carbon monoxide that leads to upregulation of the levels of intracellular calcium. That leads to movement of TFEB from the cytoplasm to the nucleus, leading to formation of autophagosomes and lysosomal biogenesis. So we asked whether this TFEB plays any role in this process, and this is what we found. So if you have control siRNA and you treat the cells with interferon gamma, MTB carrying cargo moves to the nucleus, whereas if you have TFEB siRNA and you treat the cells with interferon gamma, the mycobacterial cells, the cargo carrying mycobacterial cells does not move towards the nucleus, and killing also is rescued. And this we have shown earlier also in that autophagy paper, but the results were consistent. Then how does TFEB model conditioning? There was a recent paper which suggested that TFEB regulates an protein on the lysosomes called as TMEM55B, and TMEM55B interacts with the adapter protein called as GIP4, which loads this cargo, or lysosomes in this case, onto the dynein motor. We tested for many other pathways, but we focused on this because we got results with this. The canonical pathway for RILP1, RAB7, that does not work in this case. So I'm just describing the pathway that is most relevant in this case. So once the TMEM55B is overexpressed, it can put the cargo or lysosome onto the dynein motor, and then the dynein motor can move it towards the nucleus. So we tested this hypothesis, and we checked at the levels of TMEM55B using Western blot analysis and real-time PCR. And we found that interferon gamma leads to upregulation of TMEM55B expression in a TFEB dependent manner. Not only of TMEM55B, it also leads to upregulation of the anchoring protein, adapter protein called as GIP4, and it was confirmed. And then we employed the siRNA of GIP4 to look at what happens to the positioning. And here in this case, we are having THP1 cells infected with MTB, and we are utilizing the siRNA of GIP4. So you can see that in the control siRNA upon treatment of interferon gamma, the cells, mycobacterial cells, they move close to the nucleus here. But when you employ the GIP4 siRNA, they do not move close to the nucleus. And that is very much visible here in the quantitation of the data. And this also leads to, I'll be done in 2 minutes. And this also leads to rescue of the killing mediated by interferon gamma. So this way we are introducing a new pathway for regulating the subcellular localization of MTB, and that could regulate the TB pathogenesis. And this is the model that we propose, that in naive macrophages, the virulent mycobacteria shown in red here resist the movement of phagosome towards the nucleus where there are a lot of lysosomes present, whereas the avirulent one straight away moves close to the nucleus, gets fused with the lysosomes and gets killed. On the other hand, in activated macrophages, MTB, the blockade of this movement exerted by MTB is relieved by treatment of interferon gamma through this pathway of TFEB, GIP4, and TMEM, and it leads to the movement of the cargo carrying MTB towards the nucleus, where it fuses with the lysosome and MTB gets killed. And in the end, I would like to thank the funders. And I would also like to point out that initially this observation that some of the bacteria are close to the nucleus and some are away was made by my son when I was working at home. So yeah, I'm thankful to him. Thank you.
Thank you. Ashwini, why don't you come over here? Yeah, please come over here. So we'll move on to the next talk. That's by one of the 5 organizers who has been coming to stage quite often. You know who it is. And he's an associate professor in this place, works on understanding host MTB interactions and exploiting their findings towards development of novel vaccine candidates, therapeutic targets, and diagnostic tools. In addition, he is working on Acinetobacter baumannii and Klebsiella pneumoniae to better understand sepsis. He has demonstrated more recently the MTB work. Extracellular vesicles are virulent than being protective. MTB releases extracellular vesicles when infecting macrophages, and MTB vesicles can be engineered to express vaccine antigens of interest. But today he's going to talk something else. Yours.
Thank you very much, sir. I thank the organizers, though I'm an organizer, for giving me this opportunity. We'll try to rush through this because we are running a little late. Let me see how fast I can try to readjust the time. So this is with respect to a small amount of work that we are doing in order to understand what is the role of HubB. HubB is a nucleotide associating protein, which we find with respect to a few pieces of data that it is critical for the survival of the MTB, especially under the host-mediated stresses. And it actually, if you have HubB, there is a possibility of much more survival, especially for the exposure of the key line, the first line antibiotics. So we all know this, what MTB does and what the macrophage does. I'm not going to go into the details of it. So you have something coming out of the MTB and basically trying to modulate the macrophage environment, while at the same time the macrophage is trying to fight back and kick out the bacteria. So this is a very complex and a dynamic process which requires quite a few steps. One is after the pathogen enters, it basically alters its own and the host gene expression. It designs the molecular machines and the secretion systems in order to see whether it can deliver these particular proteins. And it also modulates the host cellular pathways by the proteins or the molecules that it is secreting, and scavenges the nutrients and prevents the phagosome lysosome maturation. It also fights the host-induced stresses, survives the immune onslaught, and multiplies to spread and establish. So there's a slew of events which basically have to take place. And there are a lot of players which come into picture. And one of the major players which basically is involved in most of the bacteria is nucleotide associating proteins. So when there is stress, which is seen especially during host pathogen interactions, these small proteins basically go and bind to specific regions of the genome and try to see how best to modulate the gene expression, which will promote either the survival or the death of that particular bacterium. What we found was, interestingly, when we looked at the transcriptome, the global transcriptome data on the TBDB, under several stresses, as well as in the presence of the exposure of several antibiotics. So there were a few proteins of MTB which were always coming up. And one of those proteins was the HubB. So it's quite surprising that different lab people, different groups across the world, when they were looking at this transcriptome data, the HubB was always coming up. So we thought, let's see what HubB is doing in terms of the stress. And here we can see that it's approximately 3 to 5-fold increase in the expression. So we thought, okay, let's see what does the literature say about this HubB? And as I said, it's a member of the nuclear associating protein. It's small, and majorly it has two domains. One is the N-terminal domain, which is the DNA binding domain. So you basically can split these domains. You just take the purified N-terminal domain, it will just bind to any DNA piece. It just has to be a double-stranded DNA. But if you fuse that with the C-terminus and have the full-length protein, it basically is able to fuse distinctly to only a few sequences, which indicates to a large extent that possibly the C-terminal domain is dictating where the N-terminal domain should go and sit. It is known for a long time that this is important for growth in vitro. Earlier, it was thought actually it is essential for in vitro growth. That's the reason several groups tried to see whether this could be one of the drug targets. But later on, other than the transposon insertion library data, so the data which was coming out from other labs indicated that this was more of an important for growth. So which means that it will still grow if you make a knockout, except that it struggles a lot. And of course, as I said, it is modulating the gene expression of the mycobacteria and different conditions. I'm not going to go details of it. But it is also one of the proteins which is amenable to a large set of post-translation modifications. And those modifications dictate what could be the function of this particular protein. In general, what is known from the literature is that if you were to have a post-translation modified protein of Hubby, it never binds to the DNA. It is sent to the periphery of the cell and possibly secreted out because it is considered to be unnecessarily coming in the way of the transcription. And so that's the N-terminal domain of the Hubby, because the C-terminal domain is so basic rich and it is unstructurable. Even by in silico, it becomes really difficult to predict a structure for that. So we just wanted to look at, given that the conditions under which the Hubby was coming up, so we thought, let us look at what is happening to the Hubby protein levels. First, at a simple growth situation, one in terms of looking at the rich media and in terms of looking at the minimal media. And you can see that both in the minimal and the rich media, it is coming up at the later stages of the growth in terms of the mid to late log as well as the stationary, indicating that there is a lot of protein. There seems to be some kind of a feedback, kind of a loop, which is basically controlling how much protein accumulates in the cell. I'm not going to go into the details of it. And then we looked at what is happening in terms of the protein levels of Hubby under different stresses. So you can see that in the presence of the hydrogen peroxide, it seems to be accumulating a little bit. And that literature sort of coincides with what others have published. And but of course, we consistently saw that as you reduce the pH, the bacteria struggles to survive, and basically it is unable to make the Hubby protein. And so that's the reason your nitric oxide experiment doesn't work really well, because the acidic pH is basically reducing the levels of the Hubby. And as I said, of course, we did the acidic pH because the mycobacterium controls that when it gets into the phagosome. And so again, we see that the pH basically as it goes down, you see that the Hubby levels go down. Then we looked at the antibiotics. Of course, we are looking at several other antibiotics, but I'm not showing the details because that work is still in progress. But what we see here is in the presence of INH, you see the protein accumulates. In the presence of EMB, ethambutol, it again accumulates, but in terms of the presence of the RIF, it goes down, possibly because of the transcription. But we do see that, I'm not showing here the GROEL2 protein. So the GROEL2 sort of stays similar in terms of the amount, is slightly less. So we do not know exactly what the rifampicin is doing here. It's quite possible that it is, there's some sort of a, because it is regulating a lot of transcription, rather inhibiting a lot of transcription, it's quite possible to predict that there could be downstream indirect events, which is leading to the reduction of the Hubby levels. What came as a surprising, but of course, this basically explains why people were thinking that this was essential, is that if you have the knockout, it grows very, very slow. In fact, we have extended this to almost 100 days, and we see that it still doesn't grow on the plate. Especially the growth phenotype is more prominent on the plates as compared to the broth culture. It's quite possible that there could be some different dynamics. There are other 6 nucleotide associating proteins of MTB. They probably are kicking in order to compensate for the loss of this particular Hubby. And as I said, there are 2 domains for this particular protein. And if you were to compare the domains of the virulent mycobacteria Hubby, as well as the avirulent, that is the Smegmatis HLP, you see that the N-terminal domains are basically, so as a full-length protein, you can see are approximately 80% identity. But when you look at the N-terminals, it is more prominently conserved as well as similar, identical in terms of 94%. But there is a huge difference with respect to the C-terminus, dictating possibly why this is different and having a different role in the virulent and the avirulent mycobacteria. So then we looked at what is possibly happening with this Hubby knockout in terms of the different conditions under which MTB sort of goes through when it is in the macrophage. As you reduce the pH, you can see that the knockout sort of gets killed very, very fast as compared to the wild type and the complemented. It happens during the nutrient depletion as well, and is highly susceptible to oxidative stress, nitrosity stress. And it also happens in the macrophages. So indicating that the loss of Hubby is making the Mycobacterium becoming susceptible to these stresses, indirectly indicating that the presence of Hubby is very important for making sure that it can fight these stresses. So then we also looked at the key first-line antibiotics. Of course, we are looking again the others, but I just want to give you a flavor of what happens when you look at the isoniazid, because this particular phenotype is very prominent. And you can see that you need a very high amount of the isoniazid for the wild type and the complemented, while the knockout is very susceptible to the INH. The same with rifampicin. In fact, in rifampicin, what happens is that you will see that it requires 14 days, while the knockout is gone within 7 days. And it is also requiring high amount of the rifampicin. So indicating that the loss of Hubby is making the cell susceptible to a smaller dose of the antibiotic. Also, the cell is getting killed much early on. So we again tested, we are also doing 2, 3, 4 days, but at this point of time, what we saw that this phenotype is also visible even at 5 days. We don't need to still go to 7 days. And what we see that also similar with rifampicin, and so indicating clearly these key line, the first line antibiotics are basically the dose of what is required is reduced when you have a loss of Hubby. So the question was, can we actually do the opposite? So we overexpressed the Hubby, and then we see that the MIC shifts for the INH. It is marginal for rifampicin, but we are also again testing the other antibiotics, whether this phenotype is very clearly only specific to these INH and rifampicin. That would be weird. But we do see slight differences with respect to how this pans out. And all the literature falls in place with the oxidative stress and the INH, indicating that there seems to be a tight interaction between those things. And then we took one of the small molecules that Dr. Nagaraja's lab had identified to see whether if you were to target this Hubby, whether that would lead to any difference in the susceptibility. And of course, in the broth cultures, the phenotype with the use of SD1 is slightly weak. But when we do the standing cultures, we do see that the inhibition is much more prominent. And when we take the SD1 and then look at whether in a combination with INH, what would happen, when you take the INH alone, you will see even after giving a high dose of INH, approximately 10 to the power of 4 cells still are alive after 12 days. But when you give even just a little bit of the SD1 and with the same dose of INH, all the cells are gone. So indicating that the MTB is getting more susceptible because of targeting Hubby. And then we did also some checkerboard analysis. This was in collaboration with Raman. And what we see is that when you use it as a combination, the MIC also reduces. It's not synergistic yet because of the values, but we do see that there is some sort of an additive effect. So because of that, we basically also tested in the macrophages and still see that as a combination, it works much better. And of course, if we use this molecule, one thing I want to make it clear before the questions come out on me, the SD1 is a very bad molecule in terms of the MIC. But it looks like it is able to at least bind to the Hubby. We are trying to do some in silico analysis and in vitro analysis in terms of screening libraries so as to make sure that we can find alternate molecules to the SD1, which basically have much more lesser MIC. But at least here, I want to indicate that you need to give a very high dose of the INH as well as the SD1, if at all you want to kill this macrophages. And then we took this particular knockout and just tested whether it was susceptible to the SDS, given that it was also susceptible to quite a few antibiotics. So then we see that it is highly sensitive to SDS. So when it is sensitive to SDS, it would indicate that there is a change on the surface of this bacteria. And so in order to look at there is a lot of literature indicating connections between this particular phenotype with susceptibility to SDS and looking at some of the polyketide synthesis. And then when we looked at these, then we see that the expression of these are down, and which basically reflects upon whether the PDM synthesis is going down. So PDM is known very well that it is one of the virulent proteins and is required for the survival of the bacterium. And then we see that by the lipid analysis of the polar lipids, that quite a few of these lipids are comparatively down in the knockout, and a couple of them are actually up. And in fact, if you want to look at these images consistently, we had to load 3 times more of the lipids that we get from the knockout as compared to with the wild type, indicating that overall that seems to be a difficulty in this particular knockout in terms of making sufficient amount of the lipids, which basically helps them to survive. We also looked at the PDMs. So you can see the PDM A and the PDM C are gone. You also can see that the family of the mycolic acid methyl esters are also gone. So indicating that there is probably global changes which is happening given that hubby is lost and given that hubby is required in order to trigger a set of genes. And Dr. Nagaraja's lab have shown recently with CHIP that it basically binds to several of those genes upstream of several genes. And of course, the functions of these and the relevance of particular genes being shut down needs to be still evaluated. So we also looked at, in addition to that, given that the PPSA to E locus downstream is immediately efflux pumps. So we tried to look at if these efflux pumps are also down, and we do see that the efflux pumps are down. It was almost gone in terms of the DRRA. And so DRRA is known to also indirectly is able to bring the PDMs onto the surface. So indicating that possibly there is some relation with respect to how this is going on. Maybe it is possible that because these efflux are down, the amount of the drug which is getting into the system is able to stay there and is able to kill the bacteria more efficiently. So the sort of model, this is still developing because we are still looking at a particular better strong phenotypes with respect to the other drugs as well. And what we see is that possibly the hubby is able to reduce the expression of the polyketide synthesis. It's known that when it binds, mostly it is reducing. Then this leads to the reduction of the PDM synthesis as well as the abundance of the polar and the apolar lipids, and it promotes the permeability to the drugs. This leads to the also there is a reduction in the expression of the flux pumps, which then increases the susceptibility to the INH and the rifampicin. So the speculation is that this is because it is on the cell surface, there is so much of changes. So the bacterium could be susceptible to more than just these first-line antibiotics. And we presume, we predict that this could be a promising target, especially for the combinatorial and shortening therapy. I'm not showing some data with the animals because we still are repeating them. If you just give the hubby knockout, it is not able to survive, but it's not that it doesn't survive. So we see only approximately 0.5 to 1 log difference. So we don't understand how it's able to survive given that it is sort of important for survival. But we think that there is an opportunity here that if you were to target the hubby, maybe it becomes more susceptible to INH. These animal experiments are going to be done soon. I'm skipping this because I've already mentioned about this. And I need to thank especially 2 of my students, Neeti Singh. So her poster is 2.338, I think it is today, as well as Lovely. Lovely is now following up on the work of Neeti, trying to look at some of the in silico screening of the libraries to see whether we can find something better than SD1. So the problem with the SD1 is it is binding to only the N-terminal domain because this was purified and targeted using that libraries. So what we think is that we need a molecule which is able to bind not just to the N-terminus, but to the C-terminus so as to prevent this to bind to the DNA. The question and the crucial step would be whether we would find a molecule that is able to dislodge what is already bound to the DNA. That would possibly be the key factor, and hopefully we'll find a small molecule in that direction. Thank you very much.
Please come on. Yeah, we'll move to the last talk of the session. This is from Dr. Raghunan Tirumalai from CCMB. Works on understanding complex interplay between tuberculosis and host with the goal of developing new and effective treatments. He has published around 30 articles in various journals of repute and received many awards and distinctions. He is also among the many in the audience, former student of Indian Institute of Science. Raghunath, floor is yours.
Thank you, Professor Nagaraja, and Professor Tyagi for chairing this session. Thank you for the introduction. And let me thank the organizers, Raman, Nishit, Krish, Amit, and Dinesh for organizing such a wonderful meeting. And my students and I are really excited to be here. It's a special feeling when you're amongst friends, associates, teachers, and I'm especially pleased that my mentors, Sandhya and Bill, are both here. It's a really special occasion for me to share some of the work that we've done here. So my lab at CCMB for the last 15 years, we've been interrogating several different aspects of the physiology and pathogenesis of MTB. And they can be kind of grouped under three basic headings here. Interactions at the host pathogen interface, antibiotic resistance, and pathophysiology. And today I'm going to be sharing with you one aspect of this work, and that is our work on studying host entry. And there are three mini stories as part of this. And I'll take you through this one by one. And it's fairly simple in the way it's been described, since there are lots of students here. And I hope you will be able to appreciate why we do this and where we are going with this. So this work started about 9 to 10 years ago when my colleague Amit Chattopadhyay came to us and said, we would like to collaborate with you to understand some aspects of pathogen entry. And that was the time they had done some work with Leishmania and found that entry of Leishmania into host cells was somehow being affected by perturbations in membrane components. So we know that now we know entry of intracellular pathogens involve interaction with the plasma membrane, and that's pretty obvious because pathogens have to go through the plasma membrane to gain entry into the internal milieu. We know that there are many candidate receptors for mycobacterial entry, and some of them are listed here, but the entry mechanisms haven't been very well characterized. And also, over the last 2.5 decades, Amit Chattopadhyay's lab has been working on the role of membrane lipids in receptor function, and that directly translated into something that we were interested in, and we wanted to investigate whether membrane lipids are involved in the entry of mycobacteria into host cells. This was about 9 to 10 years ago. So we've looked at three different components or three different factors that are associated with a membrane. And I'll take you through this one by one. The first was trying to look at host membrane cholesterol and try to ask whether this perturbing cholesterol levels in the membrane affect mycobacteria or not. So when we started this about 9 to 10 years ago, what did we know? So that was the time there was some very elegant work done by Amit Pandey in Chris Sassetti's lab, where he showed that host cholesterol is actually required for persistence. Plus, there was some interesting work from John Peters' lab published in the year 2000, where they showed that cholesterol was required for host cell entry. And the Peters lab used a chronic cholesterol depletion technique by using lovastatin, and they showed that entry of MTB into various kinds of cells, including HeLa cells and macrophages, was affected. So most of you know what cholesterol is. This is an essential membrane lipid present in higher eukaryotes. And what is important is that there's a non-random distribution of cholesterol in biological and model membranes. And we know that membrane cholesterol plays a vital role in the functioning of a large number of membrane proteins, including GPCRs. which Amit's lab has worked on for several years. So we decided to take not a chronic cholesterol depletion approach, but we used a compound called MBCD, which is also called methyl beta cyclodextrin. It's a cage-like compound to play around with the levels of membrane cholesterol. And Amit's lab has pioneered the use of this to take out cholesterol from membranes and put them back. So it was really cool. You can take out as much as you want, and you can also put back and enrich cholesterol in membranes. And here, if you measure cholesterol content, you can use MBCD to deplete cholesterol, and you can put back that cholesterol and see what happens. So the way we were looking at entry of mycobacteria was using Mycobacterium smegmatis. The reason for this is one, that you can get a quick readout, and it had already been shown around that time that the entry mechanisms of pathogenic or non-pathogenic mycobacteria are fairly conserved in terms of the entry itself. And we were using E. coli as a negative control here. So you can see from this slide here that when you deplete cholesterol, there is a reduction in the level of mycobacterial entry. And when you put cholesterol back, you can actually restore that entry, and there's no effect on E. coli. You can enrich cholesterol to levels that are excess of that of normal levels. And there you see, when you enrich cholesterol, there is now a decrease in entry as well. So if you put back more cholesterol than there are in membranes, there is a decrease in mycobacterial entry. And the third thing you can do is complex cholesterol. Use a compound that will bind to cholesterol and prevent its interaction with other things in the membrane by using a compound called amphotericin B. And many of you will know that amphotericin B is a well-known antifungal or antiparasitic molecule. So if you complex cholesterol, that shows here, then there's a reduction in mycobacterial entry. So what we concluded from all this work was that one needs an optimal level of cholesterol for mycobacterial entry. So that was our first finding. Next, we went to another lipid, which is part of the eukaryotic cell membrane. And this is a group of lipids called sphingolipids. So sphingolipids are essential components of the eukaryotic cell membrane, and they comprise about 10 to 20% of the lipids in the membrane. And we know that sphingolipids, this is what they look like. There's IPC, there's sphingomyelin, glycosylceramide. They have long fatty acid chains. They play a role in modulating the function and organization of membrane proteins. Plus, we knew around this time that they play a role in pathogenesis as well, and they've been implicated in the pathogenesis of chlamydia, Pseudomonas, Plasmodium, as well as Candida. And we also knew that mycobacteria utilize sphingomyelin as a nutrient source during persistence, but we were not interested in the use of these lipids as nutrient sources, but we're looking at their role in the membrane at the point of entry. So we asked, are sphingolipids involved in mycobacterial entry? And the way we asked this was by using an inhibitor of sphingolipid biosynthesis. And this inhibitor is called fumonisin B1. And you can see this is a sphingolipid biosynthesis pathway. If you use fumonisin B1, it interrupts this pathway at two different positions. And that leads to a decrease in sphingolipid biosynthesis. And you can actually characterize the sphingomyelin content as a function of fumonisin concentration. And let me qualify all of this by saying that whatever inhibitors we use, they're used at a concentration that are not cytotoxic. We have to do a range finding experiment before we do these entry experiments. And then we ensure that the concentration at which we are working are not cytotoxic to whatever cells we use. Either they are THP1 macrophages or they are J774 macrophages. So here you can see there's no decrease in macrophage viability, and you can see there's a dose-dependent decrease in sphingomyelin content when we use fumonisin B1. As earlier, we monitor mycobacterial entry, we're using smegmatis, and you can see there's a dose-dependent decrease. You see this on the, this pointer isn't working. Do we have another pointer? Okay, I'll use this. Okay, so this tells you that there's a dose-dependent decrease in, fantastic. So it's a dose-dependent decrease in mycobacterial entry when you use different concentrations of fumonisin, and there's no difference in E. coli entry. And we can also do confocal microscopy using DS-red labeled smegmatis and show that this is a real event. You can actually see a decrease in entry. So this led us to conclude that depletion of sphingolipids leads to a reduction in mycobacterial entry in host cells, and this effect is specific to mycobacteria. The third component which we have looked at is something called actin. You all know what actin is, and this needs no introduction. So we had Amit's lab around that time had discovered that messing with the levels of actin, or filamentous actin specifically, can lead to a decrease in Leishmania entry as well. So we wanted to see if that mirrors the entry of mycobacteria. So they showed that actin integrity is required for Leishmania entry. There was also some literature on the role of actin in mycobacterial entry into different kinds of host cells, and those are listed here. But in none of those papers was there any actin quantitation, and there was no data on macrophages, plus there was no information on mechanism. So we decided to see if we could fill this lacuna, and we used a compound called cytochalasin D, which many of you will be familiar with, and this inhibits F-actin polymerization. So you can use different concentrations of cytochalasin D and treat DHP1 macrophages with this compound. And you see that when you do that, there are changes in actin polymerization, although the overall morphology remains the same. And at the concentrations of cyto D that we use, our macrophages are perfectly healthy and there is no cytotoxicity. So one of the things that Amit Chattopadhyay's lab had standardized was how to quantitate the level of F-actin in the cell. And they did this by isosurface generation from confocal images. And they're able to determine the enclosed volume or projected area and actually quantitate the level of filamentous actin in response to cytochalasin D treatment. And you can see that there's a dose-dependent decrease in the volume of F-actin. So the question here is, does this actually lead to a reduction in mycobacterial entry? And the answer is yes. You can see a dose-dependent decrease, almost a 50% decrease in smegmatis entry, which is mirrored by the experiments using confocal microscopy. So we propose that the integrity of the actin cytoskeleton of host macrophages is necessary for mycobacterial entry. And this work was published last year. And we were really thrilled that they put some of our images on the cover page. So to summarize, this is what we found. There are three different elements of the plasma membrane that we have investigated in terms of mycobacterial entry. One is cholesterol. The other class is sphingolipids, and the third is actin. So the question that we are asking next is, yes, we found that these individually lead to a decrease in mycobacterial entry, but is there an interplay between them? Can you now inhibit each of these in pairs and see what happens? What may be the mechanisms of entry modulation? So we think that these may be because of either direct interaction of cholesterol or sphingolipids with entry receptors. And we know many receptors have either crack motifs, which are cholesterol recognizing signatures, amino acid signatures. Plus there are also sphingolipid binding domains in receptors. And these lipids might directly affect these receptor activity, or that they might affect the fluidity of the membrane, which now leads to a differential activity of these receptors. Actin probably is modulating phagocytosis. Intuitively, you know, this might be doing that. So this is something that we are now investigating, and Muskan in the lab is trying to look at some of these elements. We have now started using MTBH37RV in these experiments as well. So hopefully the next time I present, we will have some of that data. So the eventual goal is to see if we can use entry inhibitors to somehow modulate the pathogenic processes. And maybe sometime in the near future, someone in the audience can help us do an animal experiment to see if using some of these compounds will synergize with existing first-line therapy. So let me acknowledge the people who did this work, plus those who are in the lab at the current time, my bunch of thinkers. Let me thank the funding agencies who helped make this happen. And I want to make another statement. I want to sign off by quoting from a recent article that was written by Valerie Mizrahi, Digby Warner, etc. And this is an important article that some of you may have read. And if you haven't, please go and read this article, because it makes a lot of sense in terms of what a symposium like this is hoping to achieve. We say end TB, and we must understand what that actually means. So Valerie and others, as part of this article, there's a small paragraph towards the end that says we must address TB as a systemic challenge. And let me quote, she says, mirroring this complexity, the TB field has organized around specialist research thrusts that, albeit delivering important advances, have struggled to cohere in addressing TB as a systemic challenge. And what I'd like to highlight here is reframing the TB narrative while collapsing barriers among TB researchers will be critical if transformative progress is to be made towards ending the TB pandemic. And I said this 10 years ago in a meeting that Sandhya organized in ISC, is that we will only be able to make significant progress if we learn how to work together and bring our resources together, whether they're in terms of ideas or in terms of actual physical resource. And a conference like this is pretty much the best place where the seed of something like this can be sowed. So we need to come together as a community. Otherwise, we end up being in our silos doing what we love best. So let me make an appeal to everyone here, my colleagues, friends, associates across the world. If we are really serious about ending this, we need to work together and address larger problems. Thank you very much for your attention.
Thank you. Okay, with this, we come to the end of the session. And you are welcome to ask questions now. All the speakers are here, but we are running extremely late in terms of time. So you are requested to keep the questions to the minimum. Of course, you can always interact informally later also, but go ahead.
Sandhya. Yeah, thank you. I have questions for a couple of the speakers, if I may, very quick ones. First, Gopal, related to your concept of moonlighting in terms of quenching the activity of SIGA, would this not be dependent on the relative concentrations of the different proteins in the cell?
Yes, it would be.
And so
and
SIGA
also
is
the
being-
relative affinities, yes.
Right. So SIGA being so abundant, do you really think that conceptually you can envisage some sort of sequestering?
Yes, I mean, see, one part, it could be the fact that you see several of these which come from the translational mechanism. And the fact that these have to be coupled is one part. And there is some kind of a proof of concept, if one were to look at it that way, that you also have leaderless mRNA. And that brings in a completely different perspective to things. Among the other proteins, yes, we'll still have, there's still work in progress. Yeah.
Smriti, one question, this IDO1 knockout that you made, which I found very interesting, was it a whole body knockout or was it a macrophage specific knockout?
It's a full body knockout.
Right. So do you think you'd have had a greater effect had it been a macrophage specific knockout? And specifically where you depleted it, just prior to infection or a little bit after infection, because that's when you seem to see accumulation of IDO1.
Yeah, that's a good suggestion. And also I would like to add that it's only IDO1 knockout. So if, we can make double knockout, IDO1, IDO2, and TDO knockout also. And that might have better phenotype.
And Ashwini, one question for you. In all the images you showed, there was only one bacterium per macrophage. Is that what you usually see? Or if you did have cells with more than one bacterium, Did you find them localized differently, which might highlight what Andres spoke so eloquently about heterogeneity in the population?
Yeah, so we do see a lot of cells with more than one bacteria. Just for the representation, we have selected here only the cells which have one bacteria, but we do see cells with large number of bacteria, and there is some heterogeneity. That is why it was depicted in the volcano plot also. But there is heterogeneity, yes.
Hello, I see many people raising hand.
But I have my
So
mic.
the one, one over there, Vijitra,
Yes.
keep it brief, please.
Yeah, this is to Dr. Mehra. See, as per the literature data, there are at least two schools of thought about the granuloma. Some researchers, they think it's a host-directed strategy to suffocate the bacteria. But another school of thought that it is a bacterial strategy to stay alive coolly. So in your view, whether both models are equally acceptable or there is a preferential over one model versus the other?
So if I understand your question correctly, what we are looking at is host-directed therapy to kill the bacteria, and MTB can make its own tryptophan. So IDEO pathway is tryptophan based. So MTB can make its own tryptophan and it can survive inside. So what we are looking at is host-directed therapy.
Okay.
Hi.
Yeah, Deepak first and then Sheetal.
Thank you very much. Ashwani, I found your talk very compelling.
Come to the point, because we are running late.
The question is, have you looked at potentially looking at strains outside of H37RV that may have greater virulence? Erdmann, HN878 strains with even greater virulence than that, and would they fall into the pattern that you're describing? And then secondly, have you looked at potentially pulling out lysosomes from different locations in the cell and doing energy or other kind of functional assays?
Yeah, so you are right. We have not done that experiment. We need to do that experiment. We are in process of doing those experiments, but we have not done that. Very brief.
Ashwini, I have a question for you.
Very brief.
Excuse me. So I was wondering whether you've looked at what compartment the bacteria are in when you make them go to the perinuclear space. Are they all lysosomes, or is the positioning affected irrespective of the compartment that they're in?
Yeah, that's a good question. So we know there is a heterogeneity in terms of the cargo. Some are in phagosomes, some are in autophagosomes, some are in autophagolysosomes. So we have not really looked at different compartments, but whenever we treat with interferon gamma, around 40 to 50% of them go to the lysosome. So, but they are in different compartments.
Hi, I'm Garima from Delhi University. My question is for Dr. Ashwini. So we know, and it is quite established, that for phagosome-lysosome fusion, there are a couple of requirements, essential requirements like acidification, gradual decrease in the acidification of the phagosomes, as well as change in the surface markers like RAP5 to RAP7 and all. So when you are inhibiting dynein and bringing the phagosomes which are containing MTB closer to nucleus and thus closer to your lytic lysosomes, what do you think how these other processes are changing so that they can enable the phagosome-lysosome fusion? Because just by merely bringing anything closer to lytic lysosomes may not make them fusogenic to lysosomes, right?
Yeah. So you have rightly pointed out that in the early phagosome, there is RAB5, late phagosome, there are RAB7, and then that help it in the recruitment of RILP, and that leads to movement of the cargo. But Mycobacterium tuberculosis is able to inhibit that process. So that is not operational. But
So exactly, I'm asking that only, that if you're bringing
Perhaps
them
we
closer,
can discuss more later,
Okay, sure.
because there are a lot of other people raising hands. Okay, in the interest of time, I was asked by the organizers to cut short the interaction sessions. Sorry for that. Professor Solia, and after that, we'll take one more question.
Yeah, my question is to Dr. Mehra. HE3J mice are defective in TLR4 signaling. So already you are immunocompromised. On top of that, you are having IDO knockout. And yet the effect is not very striking.
I'm sorry, what did you say at the effect is?
And the therapeutic effect is still not very striking. You know, it's a double jeopardy you are having.
We did see 2 to 4-fold reduction.
But that is something very mild.
Yeah, so but all Kramnik mice don't form caseous granulomas, so we did see some fold reduction, and we might need double knockout mutants also. IDEO1, IDEO2, TDO.
Already you have TLR4 defect. You know, in a
We can
HE3
discuss over lunch, Professor Surolia. Last question over there.
Hi, this is Ved from ICGB. So my question is with Dr. Ashwini. So the host factor you have, whatever you have identified, which play a crucial role in moving MTB towards the nucleus. So have you tried their inhibitors or activators in murine model of TB?
Yeah, so I have shown the data with TFEB siRNA. I have shown also data with GIF4. We have more data with TMEM55B siRNA. So this pathway plays a crucial role for interferon mediated movement of the cells and killing of mycobacteria.
Okay, with that, I think we'll close the discussion. On behalf of Anil and myself, I would like to thank the organizers for giving us this opportunity to chair, and also to the speakers. All of them kept within allotted time, but we started late. That's why we are asked to cut short this. I think lunchtime you can interact with the speakers. I'm sure there are more questions. Thank you all.
The speakers. I thank all the speakers for shedding light on one of the most resilient pathogen of all times. I kindly request the session chairs to felicitate the speakers, Professor Anil Tyagi, to please felicitate Dr. B. Gopal. Professor Anil Tyagi to please felicitate Dr. Smriti Mehra. Professor Anil Tyagi to please felicitate Dr. Ashwini Kumar. I kindly request Professor Nagaraja to please felicitate Dr. Atmakuri. Professor Nagaraja to please felicitate Dr. Tirumalai. I request Dr. Ramandeep Singh to felicitate the session chairs, Professor Anil Tyagi and Professor Balakunja Nagaraja. I again thank all the speakers and session chairs. To continue to enlighten us on the TB pathogen, we now move to session 4, chaired by Professor Umesh Varshne and Professor Avadesh Surolia. Professor Varshne is currently a J.N. Tata Chair Professor and the Dean of the Faculty of Science at the Indian Institute of Science, Bengaluru. Professor Varshne is an authority in protein synthesis and DNA repair in eubacteria. Professor Surolia is a renowned glycobiologist and honorary professor at Indian Institute of Science, Bengaluru. He's a pioneer on lectin biology with pioneering work on anti-malarials and anti-cancer agents. He is a recipient of numerous awards, including the Shanti Swarup Bhatnagar Award. He has published over 425 research articles in almost all top journals and of biology with over 10 patents. I request the session chairs to please come on the dais. We have 5 talks in session 4, each for 20 minutes. Following the talks, there will be a brief Q&A session with all 5 speakers. I request the audience to hold their questions until then. I urge the speakers to please stick to their time. The buzzer will ring at 18 minutes. I request the speakers after their talks to be seated on the dais for the Q&A session. I now request the session chairs to kindly lead session 4. Thank you.
Okay, I think my co-chair has the details, but the thanks to the organizers for giving me this opportunity to come to the conference, which is a really excellent conference. The first speaker in the session is a colleague of mine, Dr. Amit Singh, who is an associate professor at Indian Institute of Science. He is a recipient of Shanti Swarup Bhatnagar Prize, as well as a Bioscience Award, and he's also a fellow of National Academy of Science. He does a lot of things with host pathogen interaction and oxidation reduction related aspects of bacterial and host pathology, both in TB as well as HIV. I request Amit to make his presentation.
Thank you, Professor Varshney, for kind introduction. And at the outset, I'd like to thank all the friends who have organized this conference, Raman, Nishit, Krish, Amit. So I also, because this is such a nice gathering of all the mentors and friends, I will just take this opportunity to thank everyone who have contributed to the research progress and research program, what we are doing. Especially, I'll take this opportunity to thank my PhD mentor, Professor Anil Tyagi, who's here, and also postdoc mentor, Edry Stein, who is also here. So both of them, and I think words are not going to be enough to express the kind of contribution they have made in our research program, and really grateful to both of them. With that, the talk, I'm going to talk about sulfur metabolism in Mycobacterium tuberculosis. And I'm going to talk about a relatively understudied pathway, transsulfuration pathway in MTB. Now, studying metabolism in MTB is very important, primarily for one reason, that this is perhaps the only intracellular pathogen which is able to synthesize or able to carry de novo synthesis of essential amino acid and cofactor as compared to other intracellular pathogens. So if you really want to shorten TB therapy time, which is a theme of this conference, we really need to look at the metabolic plasticity of the pathogen and really want to target them. So with this, sulfur metabolism is, I'm going to talk mostly about biosynthesis of cysteine and methionine, and it's quite complex and it's regulated at multiple levels. As you would see here, most of the cysteine and methionine metabolism is generally studied in the context of reductive sulfur assimilation pathway in Mycobacterium tuberculosis, where inorganic sulfate is taken up by sulfate transporter, and by successive reductive steps, it is able to get converted into cysteine. And cysteine is a precursor for important biomolecules from like microthiol, which is a major antioxidant, iron sulfur clusters, sulfur lipids, and hydrogen sulfide gas. In methionine by active methyl cycle, where it will generate methionine and S-adenosyl methionine. Both of them are very important biomolecules responsible for translation potential of Mycobacterium and also methylation potential. And both these pathways actually contribute to maintaining redox homeostasis in my MTB, antibiotic tolerance and persistence. However, while these pathways are very well worked out, transsulfuration pathway, which interconverts methionine to cysteine, remain poorly understood in tuberculosis. And in this, there are two branches: reverse transsulfuration pathway, which would convert methionine to cysteine, and forward transsulfuration pathway, which would convert cysteine to methionine. So today, in today's talk, I'm going to mostly focus on reverse transsulfuration pathway, and I'm going to touch upon also forward transsulfuration pathway towards the end of my talk. So if you look at reverse transsulfuration pathway, it was discovered way back in 1940s in rats, where dietary methionine, when it is taken up by rat, by active methyl cycle, it is going to get converted into homocysteine. And homocysteine is a very important branch point metabolite because it can go into reverse transsulfuration pathway to produce cysteine, which is a precursor for antioxidant glutathione, or under condition of methionine starvation, it can get remethylated to generate methionine. If you look at reverse transsulfuration pathway, there are two key enzymes, cystathionine beta synthase, which is the first rate-limiting enzyme of this pathway, and cystathionine gamma lyase, right? So cystathionine beta synthase sort of acts upon homocysteine in the presence of serine and carry out beta replacement reaction to generate cystathionine, which is then acted upon by cystathionine gamma lyase, which carry out gamma elimination to generate cysteine. Depending upon the substrate, these are quite promiscuous enzymes. They can also generate hydrogen sulfide gas, which is an important gasotransmitter in many different organisms. In case of tuberculosis, we have some idea that Mycobacterium tuberculosis is able to carry out reverse transsulfuration reaction by Wheeler's elegant study published in 2004, where it has been shown, he has shown mostly with MATB, which is a cystathionine gamma lyase homologue in M. tuberculosis. However, it is now known that cystathionine beta synthase activity is encoded by RV1077 in Mycobacterium tuberculosis, which converts homocysteine to cystathionine, and then cystathionine can be converted to cysteine with the help of MATB, which is encoded by RV1079, cystathionine gamma lyase. One of the things which I want to point out here is that CGL or MATB is a bifunctional enzyme in MTB shown by Wheeler et al., where he has shown that MATB also has a cystathionine gamma synthase activity, which can actually generate cystathionine via cysteine. So both of these activities are at least present in Mycobacterium tuberculosis. And this is perhaps the only bacteria in which a single enzyme has both CGL and CGH activity. So this seems to be very, very important, although at that time, it was not really clear what is the importance of CGS activity in the context of Mycobacterium tuberculosis. So we started working on cystathionine beta synthase, which is the first rate-limiting enzyme of reverse transsulfuration pathway, and has really not worked upon in MTB. Human CVS is very well worked out. It has three components: N-terminal heme binding domain. It has a central catalytical core where lysine, active site lysine is present that binds PLP cofactor, which is vitamin B6. And you have C-terminus CVS domain, which is arranged in tandem, which binds to S-adenosyl methionine, which is a Bateman module. And because of this module, cells are able to sense energetic state because they can directly sense SAM levels. If you look at CVS of Mycobacterium tuberculosis, it has actually lost heme binding domain. However, it is able to retain Bateman module, which binds SAM. And this is also very unique in the case of TB, because if you look at whatever CVSs have so far been published from bacteria, none of them contains SAM binding domain. So it has to be some evolutionary advantage which MTB CBS has over other bacterial organisms. So we purified this protein. As purified, CBS is a tetramer. It is about more than 200 kilodalton. If you look at purified protein, is yellow in color. And if you mutate active site lysine, which binds PLP, color is lost. Absorbance, you can see characteristic absorbance of PLP at about 430 nanometer, which is lost upon mutation lysine to alanine. Also, we confirm that similar to what we have predicted, it binds SAM, and not only it binds SAM, SAM binding is able to activate enzyme as shown by increased production of hydrogen sulfide gas. So SAM seems to be the allosteric activator of CVS and Mycobacterium tuberculosis. CVS is known to have, they exhibit broad substrate specificity, and this is something not new. We already knew from other CVSs, and MTB sort of is a bona fide CBS because it is able to utilize serine, cysteine, or two molecules of cysteine to produce either cystathionine or lanthionine. We confirm these finding by acid anhydride assays, where we can look at cystosine production from serine or O-acetylserine, and also hydrogen sulfide production from either cysteine alone or cysteine plus homocysteine. So with all these assays coupled with mass spec, we have been able to confirm that MTB CBS is a bona fide CBS in this bacteria. So, so far, if you look at CBS, there is a lot of biochemistry which has already been done. But when it comes to structure, CBS tetramer is quite unstable. It has not been so far reported from human and not from bacteria. So it's probably, it's a challenge that full-length CBS, solving structure of full-length CBS is quite a challenge. So we were able to solve this problem by solving full-length tetrameric structure of MTB CBS in collaboration with Somnath Datta at MBU. And here you can look at the trimer structure where all 4 monomers come together to form 2 dimers. And a monomeric, if you look at N-terminus catalytic core region, comes together to form 2 dimers. Dimeric interface is supported by hydrophobic interaction. However, for tetramerization, it requires Bateman domain, C-terminus domain, and tetramerization is actually stabilized by hydrophobic interaction between the Bateman domain, which is at the C-terminus. Bateman domain can communicate with central catalytical region by a flexible loop of about 32 amino acids. To show that Bateman domain actually leads to tetramerization, we have deleted the Bateman domain. And you would see that the deleted form of Bateman domain is actually dimer. And not only it is dimer, the enzyme which is deleted for Bateman domain is basically it has a very high activity, very high hydrogen sulfide producing activity as compared to wild type CBS. So, and if you look at in response to SAM, this deleted version of protein, which does not have SAM binding domain, does not respond to SAM. So it's a very clear cut example of how a protein can sense and activate its activity. Sorry about this. So the reason we think is that it looks like Bateman domain is somehow intrasterically inhibiting the central core region, and therefore it is not allowing its full activation. However, SAM binding seems to reorganize Bateman domain or flexible loop so that central core region gets activated. Now, we need to figure this out, that this is how the mechanism works. So we solve the cryo-EM structure of CBS, which is bound to Bateman, which is bound to SAM. And the binding of SAM is going to take place at the C-terminus region. And the picture which I'm showing is the catalytical core region, which is at the N-terminus. And you will see that the SAM binding in the CBS region at the C-terminus is going to influence the catalytical core. You will see reorganization of loops, amino acid, and this is the active site lysine. So you would see that reorganization is taking place here. If you look at this structure in the absence of SAM, the substrate channel seems to be in close conformation. Active site lysine is hidden. And when SAM is bound to this protein at the C-terminus Bateman domain, at the central core region, which is towards N-terminus, you would have substrate opening, as you can see here. And also the active site lysine is more accessible, which sort of tells you that there'll be more intake of substrate. And this could be one of the mechanisms by this, how this protein and how SAM can activate the activity of CBS allosterically. If you look at humans, in humans, SAM is also a stabilizing factor for CBS. Therefore, we wanted to ask the question whether SAM binding to CBS can also stabilize the protein in MTB. And what we have done here is that we have used azathioprine, which is a specific inhibitor of SAM biosynthesis because it inhibits SAM synthetase. It's a purine analog. We performed LC-MS, and you will see that in a time-dependent manner, MTB exposed to azathioprine showed reduction in the SAM levels. So this azathioprine as an inhibitor is working. And if you compare what is the level of CBS here, you would figure it out that azathioprine exposure actually reduces or depletes CBS by 50%. And this is also something which is very, very similar to what happens in humans. About 50% of reduction when you cut off SAM, and by exogenously adding SAM, you are able to restore CBS levels. So very nice way of sensing SAM and stabilizing and destabilizing CBS. How does it happen? That's the question which we were wondering. how MTB is able to degrade CBS depending upon SAM levels. And we came across a very elegant study done by Heron Darwin, where she has shown that MTB undergoes or control, one of the quality control mechanisms of MTB protein is pupillation followed by proteasomal degradation. And if you look at this paper, in the proteome of MTB, which is going to be pupillated, CBS is one of the substrate. So it appears that CBS is going to be pupillated and then pushed towards proteasomal machinery for degradation. Whether that happens in response to SAM or not, we have no idea. So what we did, we mutated the active site lysine, which is shown in this paper to get pupillated in active site lysine of CBS. And this lysine is positioned in the SAM binding Bateman module, right, which is 428, not the active site lysine, which was lysine 44. So this is another lysine which exactly lies in the SAM binding Bateman module, is also a site of pupillation. So if you mutate that site and now express this mutated variant of CBS in MTB mutant that has a deleted CBS, endogenous copy deleted, you now see that azathioprine is unable to deplete a CBS, right? So the SAM mediated depletion of CBS is actually dependent on pupillation machinery in MTB. So we were able to propose a model, occlusion by occupation model, wherein under nutrient rich conditions, under SAM sufficient conditions, SAM is able to bind to Bateman module of CBS and therefore hysterically inhibits any pupillation of the lysine residues. When there is nutrient deficiency or SAM deficiency, lysine is sort of exposed, and now this post-translational modification pupillation can take place that pushes the protein for degradation. And this could be a very elegant mechanism by which transsulfuration pathway can be regulated in response to methionine or SAM levels. So the next question comes, what's the use? How is this affecting sulfur metabolism, given that MTB has a lot of metabolic plasticity and CBS mutant really does not have any phenotype in any stress conditions. So we wanted to find out more how this is all connected to bacterial survival. So we have done mass spec analysis of MTB metabolites, which very targeted metabolomics of reverse transsulfuration pathway, and also active methyl cycle intermediates in response to azathioprine, which depleted SAMs, like you can see here. And we isolated metabolites at 6 hours, which is the early time point, and 12 hours, which is little later. And we look at the metabolite of reverse transsulfuration pathway, which are here, and also active methyl cycle intermediates. So what you see here is very interesting, that early 6 hours, if you look at 6 hour time point, if you add azathioprine, you see there is downregulation of aspartate, methionines, SAH, and SAM. All active methyl cycle intermediates are downregulated, or they are lower at level. They should be, because SAM is inhibited and also CBS is down. So one of the things which we were hoping that once we deplete CBS, because SAM is not here, we were hoping that we will see downregulation of cystathionine also, because CBS is the major enzyme that converts homocysteine to cystathionine. So if CBS is depleted because SAM is inhibited, you would see downregulation of cystathionine. However, to our surprise, we see a completely opposite data, where at 6 hours, we see a slight increase in cystathionine, and cystathionine pool sort of accumulates by 12 hours. And this was very, very surprising for us. And by looking at all these enzymatic activity, we could only figure out one reason. It could be possible that the next enzyme, MATB, which is a bifunctional enzyme already shown by Wheeler et al., may work in reverse direction to produce cystathionine from cysteine under these conditions. So it could be possible that under the condition when SAM is depleted and CBS is down, MTB start to operate transsulfuration pathway in reverse direction, other way, which is forward transsulfuration pathway, as a mean to sort of control biosynthesis of, or to maintain the level of active methyl cycle intermediates. How to prove that? So we have used an inhibitor, propargyl glycine, which is a specific inhibitor of MATV, and in combination with azathioprine. So the way we are doing it, we are inhibiting both. We are inhibiting reverse transsulfuration by inhibiting SAM biosynthesis. If you deplete SAM, CBS is not going to be there, reverse transsulfuration is not going to be there. And then if metabolic switchover happens to forward transsulfuration pathway, you can inhibit that by propargyl glycine, MATB, which is the inhibitor of MATB. So we did this, we monitor survival, and survival is greatly affected when both reverse transsulfuration and forward transsulfuration are not working. You see increased ROS when both are inhibited, More importantly, you can rescue the survival defect by adding active methyl cycle intermediates to MTB. So it's a very elegant way by which both forward and reverse transsulfuration pathway are important. And forward transsulfuration pathway is only going to work in the MTB when reverse is inhibited. So it's absolutely great way to ensure survival. So this is under SAM sufficient condition. MTB is actually metabolizing a reverse transsulfuration pathway to maintain the pool of cystathionine and active methyl cycle intermediate. Here, CBS converts homocysteine to cystathionine, and under these conditions, MATB is sort of kinetically constrained to produce cysteine. However, if you block SAM biosynthesis, there'll be no cystathionine also. Under those conditions, metabolic switchover takes place, which allow MATB to now start carrying out forward transsulfuration pathway so that active methyl cycle intermediates can be preserved by bacteria, can be retained by bacteria for its own survival. And as a pathological significance of this pathway, we used HIV as a model because HIV shows homocysteine accumulation. And homocysteine being a substrate of CBS, we assume that under these conditions, MTB survival may be dependent on CBS. And the other reason to do this experiment was that we never found any phenotype of CBS mutant under any given condition. So we use HIV transgenic mice where HIV proteins are expressed, and this mice shows phenotypes very similar to HIV patients in terms of organ damage and other pathological implications like nephropathy. Here we could see that homocysteine levels are more accumulated in the lungs of HIV transgenic mice. And under these conditions, CBS mutant has some survival defect, although not a lot, but this is to make, as a proof of concept, under condition when there is homocysteine excess, MTB may require reverse transsulfuration pathway to sustain its survival. With this, I can summarize that MTB CBS is a PLP containing heme-less tetrameric enzyme, which is allosterically activated and stabilized by SAM. And it's actually destabilized by SAM paucity, and therefore it is required to restore AMC intermediate redox balance and survival upon induction of forward transsulfuration pathway. And it's critical for the survival of MTB under HIV-TB co-infection. With that, I end. And I would like to acknowledge fantastic graduate student. And very rare in your life, you will find such a student like Parijat. And I'm really grateful for her. I was just an observer there and could observe how she grew and matured as a scientist with Ishika, and to a fantastic collaboration with Somnath Datta and Sunil Lakshman. I'm happy to take any question later. Thank you very much.
You can come here. Our next speaker is also another Amit, this time Dr. Amit Kumar Pandey. He's currently Associate Professor in the Infection and Immunology group at THESTI. His area of interest includes understanding mycobacterial pathogenesis with major focus on understanding various mechanisms of antibiotic and disease persistence leading to AMR in tuberculosis. His lab is working on the hypothesis that targeting persisters would help shorten the therapeutic regimen and will reduce the frequency of AMR cases in tuberculosis. He has published many research papers on tuberculosis biology and has over 24 years of experience in working with several in vitro and in vivo animal models of tuberculosis infection. Amit Kumar.
Thanks for the nice introduction. And as Krish started this, so I thank the other organizers for giving me this opportunity. So without wasting my time, so my talk would be brief, very few data there, but it will be more of a question and observations that we have, and this is all unpublished data. So we would definitely like to have feedback on what, the audience think about the data. So to begin with, as we all know that the drug pipeline is all drying up, and, on one side you have the drug discovery program, which has its own challenges, and you get very extremely low success rate, and that leads to accumulation of a lot of small molecules that you don't go back and see if they are of any use to us. And technically speaking, it's very important. It has helped us, but then the cycle of generating AMR and resistant and new drug is a vicious cycle that needs to be addressed. On the other hand, you have this extended regimen that complicates this disease further. you start seeing, non-compliance issues, which very well correlates with the drug tolerance phenotype that we see. And the latest, terms that have evolved, which, antibiotic and disease persistence, it's a very new field, a lot of hand waving happening, but, we'll try to be very careful in using these terms. So I already talked about this vicious cycle. I think something needs to be done to address this drug tolerance persistence in order to, augment the drug delivery pipeline, and of course, shortening of the treatment regimen. That is the theme of this meeting. So going further, a brief introduction again, different categories of bacteria that we see in a population. So resistance, everybody knows there's no effect of the drug. And then on the other side of the spectrum, you have the sensitivity, drug sensitive bacteria. Then we have the drug tolerant population, which eventually is cleared off, but it takes time. Then the latest, the new thing in the town is this persistence, which we clearly see by biphasic curve. And let me tell you, this is not specific to TB. This has been seen in other pathogens as well. So the gold standard of measuring the level of frequency of generation of persisters is this biphasic curve that you will be seeing in my presentations. So this is not just in vitro, it has been demonstrated in patients as well. So if you start plating the sputum samples, after say a few days of treatment, the CFU plateaus out. So this is also observed in clinical patients as well. And then the thing gets more complicated when you talk about disease persistence, which is true for TB as well. So TB has both antibiotic and disease persistence. And when I say disease persistence, this is clearly the growth curves that you normally see in animal models, wherein MTB grows really at a logarithmic rate for the first 3-4 weeks, and then it plateaus out. So the plateau would be the disease persistence that I would be referring to in my talks. So, stress is good for, so there's a question that was asked earlier, what is good for pathogen and what is good for the host? I believe, the whole pathogen gives the best under stress, and that is not just true for man, and this is true for all living beings. So the hypothesis that we work on are these stresses, they brings the best out of TB, and TB, I think, is one of the pathogens which is smart because it adapts. So adaptability is key to its survival and propagation. So again, coming to very basics, that we believe adaptability is key, and then targeting adaptability would definitely be an approach which can be, clubbed with the regimen to maybe shorten the duration. So again, for the students, there is always a trade-off. I just want to put forth what we know, and it is up to you to decide which pathogens are more smarter. On the one extreme, you have the plague, anthrax, disease where the pathogen is very, acute illness you have, and the mortality rates are very high. Another extreme is the tuberculosis, which is a chronic infection. You see, mortality low. TB sounds contradictory, but relatively the rate of mortality is definitely low. So, there is a two-way process here. One is like fight, where you do or die, but TB takes a very relatively different approach where it adapts and it believes in live and let live policy. So the jury is out. Which pathogen you think is more smarter is you to decide. So this is a cartoon that says, on one hand, you try to kill each other, but you are evolutionarily evolved for centuries, that you really are not killing each other. You understand each other well, but then you need to potentiating. So basically, disarming the bug would be a key if you are really focused on shortening the regimen. So it should be a two-pronged approach wherein you target the essential genes, but if you can add a molecule that prevents TB to adapt, would be interesting to see if this hypothesis can be worked out. So as I described, we are interested in mutants that go both ways. So ideally, when you infect an animal, you see this typical curve, but we would be interested in mutants and genes that are, they go the other way around. We have mutants that overgrow mutants that don't persist. So I would be describing both these categories as persistent deficient. And I have some data to show that this is indeed true. And these both group of mutants are sensitive to normal the first line entity that we have. So this is a way of identifying adaptive genes that help TB to adapt. And then this is again a cartoon which says that if you prevent adaptability, we might play with the biphasic curve, and the whole goal is to make sure that the line is straighter, and that way we'll reduce the regimen. Again, so this is from a review wherein you all know by now what are the mechanisms and pathway TB uses and to adapt, and then the host component, of course, cannot be ignored. So basically, when you have stress, you activate a lot of pathways, and TB tries to adjust to these. And I think somewhere in the doing so, MTB adapts and becomes more tolerant to drug and induces disease persistence. So again, these are all slides where you can see this kind of stress that MTB faces inside the host. We have a poster wherein we have worked on pH, redox balance. There is again one poster. So the talk that I would be giving is one on carbon switch. As you all know, as Raghu pointed out, my lab started with cholesterol and trying to understand how cholesterol induces persistence. And then, of course, one short story on storage of carbon source. So glycogen levels and how they induce disease and antibiotic persistence. So again, in the interest of time, this is just a repetition of what I said. So coming to my first story. So the background is we have identified a VAP, a ribonuclease toxin, which we have published, and we have demonstrated that in presence of cholesterol, it gets activated, and you can see the kill curve. You can very well appreciate that, absence of this ribonuclease toxin. It decreases the level of persisters in the population. And this was all in vitro data. We replicated the same in guinea pigs. And as you can see, in guinea pigs, these mutants were hypervirulent. So you remember my cartoon, it overgrows for some reason. And, it is hypervirulent in respect to CFU and both pathology. We demonstrated that the strain was hypervirulent to the wild type strain. So this was all in guinea pigs. So we wanted to study the host side of this. What are the genes that are differentially regulated by the host? And does host play a role in this disease persistence? So all this was possible only in mice models. So we replicated the same data in mice, and as expected, we saw similar phenotypes. The red ones, the mutants, were hypervirulent. They were growing really fast with a lot of pathology. So we wanted, cutting the long story short, we wanted to know if these overgrowing, fast-growing cells are more sensitive to the drug. So we did a kill curve assay in animal models, and as expected, we saw and we demonstrated that this growth enhancement or increased growth in bacteria led to more drug susceptibility. So these suggest that MTB purposefully slows down, and that during that process becomes drug tolerant. So now coming to I'll switch gears. So coming to the host, this is what is very new data, unpublished data. So one interesting thing that we observed was so this was a high MOU experiments that we performed, and this was very shocking. So within 18 weeks, all the mutant infection mice died. And believe me, this is black 6, and we all know black 6 are quite resistant. All the wild type infected mice were happy, healthy, but the mutant mice, they were all dead by 18 weeks post-infection. So the interesting thing that we observed was something happened between 3 to 4 weeks where the mice started, showing symptoms, and then we were very excited about this window. At this window, what exactly is happening in the host, which eventually is, leading to its own death with, hyper inflammation and a very high bacterial load. So we designed an experiment wherein we wanted to hit this window, and we did some immunoprofiling studies with help of Dr. Amit Awasthi and the I'm just showing very major findings. We found a very, where is the legend? So, sorry, I think the legend is missing. So you have to believe me. So the green ones is the mutant. And we saw a stark decrease in the monocyte levels 4-week post-infection. And there was an increase in MDSC's population in the lungs. A neutrophil, we saw a lot of neutrophil infestation or in the lungs. And it was true for spleen as well, what we have observed for the lungs. So further, we wanted to see the polarization. If this initial innate immunity, they dictate the polarization of macrophages, which eventually dictates the adaptive immunity. So we wanted to see if this, the polarization of macrophages, how they are different between these two groups. So as you can see, in the mutant, there was a shift towards M2 polarization, which is usually not seen in the wild type. And we did real-time qPCR as well to show this. So the take-home message was this mutant was not able, was skewing the innate immunity was differentiating the macrophage to an alternate differentiation where M2, which was favoring its own growth. So this was very interesting because one gene mutation was, just changing the whole immune profile of the host. So the next step, we wanted to know what are the other pathways that are affected to understand the global transcriptional changes happening in the host. So we did a RNA-seq analysis, and we the list of genes that were up were mostly to do with inflammation, neutrophil related, neutrophil migration. So the take-home message from here was, surprisingly, the initial neutrophil attack has to be resolved at certain time point for appropriate T cell immunity to develop. So for some reason in these mutants, this resolution of neutrophil mediated inflammation was not happening. So the delay resulted in hyperinflammation, a lot of accumulation of neutrophil at the site, which probably was the reason why the mice were dying. So the question here is, are we dictating the immunity, or the TB is the one that dictates? So usually the resolution of neutrophil and adaptive immunity kicking in is described as a host response. But clearly the data shows the one gene mutant is completely, changing the whole host immune profile and prognosis or disease progression here. So while we were at this, because of the COVID, there were a lot of literature coming up. So when we were analyzing the RNA-seq data, there was a lot of similarity with what this group has published and observed in COVID-19 patients. So surprisingly, what they showed was all this phenotype was TLR4 dependent. So we had this mice at our facility, which has a TLR4 point mutation. So the TLR4 deficient mice we used, and we wanted to see if we can reduce some of these mutant effects in these mice, or we can, if we can rescue. So this is the data that we have gotten. So in black, as I said, in C56 and black 6, you can see the growth of bacteria, which was toned down in TLR4 negative background, which was more prominent in spleen for some reason. We don't know why, but whatever I described earlier, it seems was dependent on the presence of TLR4. So to conclude this part, we observed a hyperphilodent phenotype as observed in guinea pig. We replicated in mice. And again, this fails to interrupt granulocyte, resolution of neutrophil mediated pathology, and alters the Th1, Th2 axis, which probably leads to the phenotype, and all these phenotypes got partially, or I would say in spleen, was totally dependent on TLR4. So switching gears now, again, it's a nutritional talk. So there was a hypothesis in my lab that, long-term disease persistence, there has to be a place where bacteria is getting the nutrient. So as we all know, glycogen is a biomolecule that is universally known to store carbon. So we looked at the literature, does glycogen level affect pathogenesis or drug tolerance or persistence in any way? We could not find much of the literature, so we decided to see if this can be explored on. So we generated a mutant which failed to accumulate glycogen in bacteria, and we did some analysis. As you can see, there was decreased in glycogen levels in the mutant, and phenotypically this mutant was very sensitive to starvation, and of course the reactivation was also hampered. So absence of storage of glycogen affected both these phenotypes, and so it was sensitive to pretty much all the stresses that we tried. But where I want to go with this is, again, there was a difference in the lipid profile. The membrane permeability was disturbed again, but the most important thing that I want to show here is, well, it has a defect, though the persistent growth defect of the other type. So one was hypervirulent, one was defective in persistence. But the interesting thing was it had a carbon source specific effect on drug tolerance. So when you do a kill curve, we found that this mutant was defective. It generated less persisters, and as you can see, it was dependent on carbon source. In 7H9, you don't see anything, but when you switch to minimal media, you just see that this mutant was more sensitive to the drugs that we tried, and we observed the same thing in macrophages. This mutant was more sensitive to the drug, and we replicated the same in mice. As you can see, absence of the glycogen does affect drug tolerance in mice. So this is again repetition of what I said. So I request, there are other works that I talked about glycogen and Shefali's work. So we work on redox, pH, and some regulate transcription factors. We see very interesting phenotypes with respect to drug tolerance and persistence. So none of this could have been possible. So Shefali did most of the host work that I talked about. Taruna did the glycogen part, and the whole story was started by Sakshi Talwar. She is now assistant professor in DU. She started the cholesterol work in the lab, and you will see other people in the poster. So I thank all my collaborators, and then funding agency and my director, and of course, my TB colleagues at THSTI. Thank you.
Okay,
You can also come here.
please come here. So I'm pleased to introduce Professor Gordon Das. He is professor of molecular medicine and has several honorary positions around the world. Dr. Das's research interests include the molecular biology of tuberculosis, immunology, host pathogen interactions, and cellular signaling. His current research focuses on investigating the molecular mechanism of MTB infection and host response in order to develop new therapeutic approaches for TB treatment. He has published more than 80 research papers in prestigious journals. Dr. Das has received number of awards and is a fellow of National Academy of Sciences in India. Professor Gordon.
Thanks, Professor Shirolia, for such a generous introduction. And I thank organizers for giving me the opportunity for talking about some of the data what we generated some time back. In fact, the last couple of years, I'm not that active because of several reasons. Anyway, today I'll talk about some of the things what we have done so far. So as you know, we are talking about NTB program and we are aiming for it. But as you know, the TB is a very old disease and probably the oldest known human disease. And every time we try to catch, but it slips away. So that's what I have taken this cartoon from this Hollywood cinema and by Steven Spielberg and say, catch me if you can. That's because TB, it exists in various forms. Sometimes in latent form, sometimes in active form, and that also can be converted to each other. And sometimes it in the out of the cell, sometimes inside the cell, and sometimes it infects macrophages that we all know. And recently it's coming out, it's not only macrophage, some other cells also can be infected by TB organism. So as I was talking about, this is the oldest known human pathogen. So probably this slide is redundant, but still I would like to comment a couple of things. This is, as you know, this is TB. You can really find it out as early as 500,000 BC. And till today, probably TB is one of the biggest killer, and especially countries like in India. And this is also another redundant slide, but I wanted to put it to have put two comments. The number one comment, yesterday somebody was talking about TB and nutrition and this and that. That's true. But at the same time, if you look at the TB, who are the victims? The Amitabh Bachchan and Kamala Nehru, they're having that malnutrition? I don't know. And also, this is to have the attention of scientific-political crowd. So I wanted to talk about what's India is having the TB burden. In India right now, the TB burden is every 3 minutes, probably there are 2 people are dying out of TB. So now the question is, why we are not really successful? Why we really cannot get rid of TB? If you look at the TB, of course, this is infectious disease. The two way to control, one is the therapy and one of the, of course, the vaccine. The therapy we have, there's something called DOTS. This is with the multiple antibiotics, and this we call it short course, but it's a 6-month treatment. And that's for 6 months for the regular TB. However, for drug resistance is, I don't know, 1 year, 2 and 1.5 year and 2 year, and so on and so forth. And that also incorporates risks for the generation of drug resistance variants of organism. The vaccine, we have a vaccine we call BCG. This is almost 100 years old, invented in 1921. So basically, after that, we tried many of the strains, many of the clinical trials, but so far not a single vaccine which is better than the BCG. Where we are doing mistake? So just to give you a little bit of the vaccine pipeline. These are the pipeline looks like in 2022. This is in phase 3 clinical trial. As you see, there are 3 of them, which is the BAP 1002 and MIP and GAM TB VAC. VAP BMP 1002. This is long been after some time back. There's already clinical trial been done in South Africa. Bill is sitting here and Adria is also sitting here. They know what the result is. I really don't want to spell it out. And so as MIP, clinical trial been done in UP sometime back, almost like 15 years back. And so basically we are expecting some good vaccine strain very soon, but I do not know whether they'll be having better than BCG, but till date, we do not have a better vaccine than BCG. So in the meantime, when I started my lab, 2 of the most brilliant graduate students, right now, Dr. Ved Prakash Dwivedi, who is also in the audience, right? Now he is the principal scientist or the group leader in ICGEB New Delhi, and the Sumit Chatterjee, who is associate professor right now in Rajasthan Central University. They wanted to look at it in a little different way. They wanted to measure why and the BCG is not really working and how much immune response we really need to eradicate or eliminate the TB bugs. So they did the experiment in the following way, and what they did is they infected the animals with mice and then treated with the animal with some kind of antibiotics, and then looked at the what kind of immune response we get, etc., and then whether we get this these animals are protected. So to have your attention, this much of immune response we can get it, and the BCG can get this much. And what is the difference in immune responses? As most of you know, the immune responses or the vaccine efficacy is mostly depends on the T cells and memory T cells. What kind of T cells we really require, the major difference with the BCG and the maximum immune response is coming in the differences Th17 response or the IL-17 responses, which is been done also by many other laboratories, including Andrea Cooper's lab. And then they established that for the maximum efficacy of the vaccine, we need the two things. One is Th1 response, and another was Th17 response. Whereas BCG can only mount Th1 response, but not Th17 response. That has been published some time back. Now the question is, what else we need? Of course, for vaccine efficacy, we need not only T cells, but these T cells has to be pushed to the memory compartment. What kind of memory response we need? We need the central memory response. And of course, thereafter we learned not only central memory response, we need some T cells, memory T cells, we call it stem cell-like memory T cells. As you see, this is central memory T cells and the T effector memory T cells, so they exist in the dynamic balance. So how we can push the cells, the effector memory cells more towards the central memory T cells. From many laboratories that includes our laboratory, what we found, there is a difference, the target difference in the effector memory cells and central memory T cells. That is the KB1.3, which is the druggable, was a ion channel. So basically already drug exists. So what we are talking about, the clofazimine. Clofazimine is one of the drug which is already being used for the treating drug-resistant TB and also for the treatment of leprosy. And actually anti-inflammatory, this is already known. It's already been 40 years in the market. So therefore, for obvious reason, what we did is we did the experiment in animal with the treating with clofazimine. And clofazimine, as you see, this is whether our idea is if we can push some of the effector memory T cells to the central memory T cells, and if we can push them a little bit towards the T cell, stem cell-like memory T cells. So here is the result, as you see, probably if we can, the supplement with clofazimine with the conventional vaccine BCG, our efficacy actually we get more than 100 times, as shown is in the CFU in both lung, spleen, and liver over a period of time. To look at the immunology, so it's just a little busy. You don't have to really remember all this, but here's the quantitations. Where is if you substitute the clofazimine, where we get central memory T cells, both in CD4 compartment as well as CD8 compartment. That has been highlighted in several journals, in journal front page discussions, including, in fact, Journal of Infectious Disease. They had put in their frontline discussions and many other journals. Now the question is, places like India, countries like India, already we are vaccinated, we are immunized with various already BCG at the birth. So as we know, the BCG really doesn't protect. So therefore, can we improve our immune response, or can we be, adults can be protected? So for that, using same argument with the clofazimine. So we did the mouse experiment once again with the animal previously, then vaccinated with the BCG, then come back with either revaccinated with BCG or BCG plus clofazimine. Once again, as you see from here, the data, the data looks like this. We make difference at least 1 to 2 logs. So basically it says, indeed, even if we are previously vaccinated with the BCG, still we can really improve. And here is the quantitation data. It's also shown here. Now, what kind of immune response we get? Do we really get the central memory T cell response? Yes, and indeed another thing, what we discovered, not only central memory T cell response, but also we mount actually copious amount of the T stem cell like memory T cells. From here, what we can conclude, probably instead of going to the conventional method for generating a vaccine strain, which can, for developing and the immunization and the harvesting result, it takes 20 to 30 years. Why not to use the BCG plus whatever the repurposing drug, which can actually improve the vaccine efficacy. Now, one can say, yes, clofazimine, it has several side effects. Yes, indeed, it has also bioavailability. If you look at it, actually accumulates in various organs for at least 28 days or more. So therefore, can we have even the better? How do we do that? If we look at some of the food supplements, here we are talking about lutein, which is comes from the celery, parsley, broccoli, carrots, and so on and so forth. So that also another KB 1.3 inhibitor. So we have done the same experiment with this, the lutein. So recently we published this paper in the last in PLOS Pathogen. It actually does the same thing, which is just once again to emphasize this is a food supplement, and with food supplement we can achieve such kind of immune responses. And also we measured the immune responses in terms of the T cell activities, the effector memory versus the T central memory. Indeed, by supplementing with such kind of compound, such kind of food supplement, we can mount T cell memory responses. So I'm a little bit switching gear towards now the therapy as pipeline. As you see, this we have a long list of pipeline. Yesterday we heard of bedaquiline, which is breaking your roof. Now it is actually indeed this is something good happened in this last decade. And today we have drug which is called DOTS, and we have multiple antibiotics that are incorporated just for the generation of drug-resistant variants. And actually, oftentimes because of in compliance, that is the one of the major thing. So now, still, what we talked about, the bedaquiline, still it is an antibiotic. And now, if I can quote Anthony Fauci, who is the director of NIH, so in NID, so we need to better understand the delicate balance between the host and pathogen in the context of the entire biological system, and this requires a radical and transformational approach. So what we thought to look at why the treatment of TB takes 6 months, and for the regular TB, if you look at the kinetics, within the 30 days or the 1 month of treatment, almost more than majority of the bugs, probably 99% of the bugs, they disappear. The Professor Sharman Singh is sitting here. He can tell us even more about it. So basically, what it says, the majority of the bugs are responding to the conventional antibiotics. However, the remaining bugs, they are not really responding to the conventional antibiotics. So actually that means that probably here is the problem which is, it takes more than 4 months. So this is, it's a long story with collaboration with Professor Sharman Singh sometime back. What we found, this, the MTB hides in a niche of mesenchymal stem cells. They actually, mesenchymal stem cells at the surface of the granuloma, where outside the T cell, inside the MTB is surviving in happily. And I'm really pleased to see Dr. Mehra's slide. She was showing that's the ring of IDO. Just to let you know, this IDO, the mesenchymal stem cell, is the one which produces copious amount of IDO, so which actually inhibits the T cell responses. So basically, from there, what we summarized at upon MTB infection, MTB makes its own spear, then mesenchymal stem cells are attracted, and then the T cells are being they kept the T cells at the bay because of the separation of the mesenchymal stem cells. And that is the dynamic balance. And dynamic balance is produced by the IDO or nitric oxide. And then, so that is what the MTB survives within the granuloma very happily. And then one can ask me why then you mount immune response in the periphery. Basically, what we believe, some of the active antigen transport or some of the granulomas are being dismissed, that antigen is being taken up by the draining dendritic cells, and then T cells are primed in the draining lymph node. So now, if I talk, we are talking about this kinetics. I'll take only a couple of minutes. Here is the data of what it says. Basically, if you treat animals with the INH, it actually removes all the replicating bugs, where the dormant bugs are still, hanging around. So I'm not going through the data in detail. And in the meantime, there are several laboratories, including our laboratory. What we found, not only mesenchymal stem cells are coming towards the granuloma, but also some of the mesenchymal stem cells are being infected, and they are going back to the bone marrow. So then we did several in vitro experiment. What is going to happen? Why the mesenchymal stem cells are being infected? What we found upon infection with the mesenchymal stem cells, actually the MTB is acquiring all dormant genes, or the upregulating dormant genes, whereas the downregulating or replication related genes. And also the upon infection with the MTB, mesenchymal stem cells are going for the quiescence. So that already we have done. So that gives us the opportunity to find out what are the targets are being used for the adaptation of the MTB in the mesenchymal stem cells. We did all RNA-seq, and what we found is there are 3 things they're using. Number 1, cytokine network, which is IL-13 receptor plays an important role. Then the autophagy, and they're the 2 major, and the lipid synthesis. So we did all these experiments, and then just to quickly just show that if we can simultaneously use autophagy, autophagy with conventional antibiotics, probably we can do better sterile clearance, and also that majorly prevents reactivation of the disease. So therefore, this is my last slide. So basically, using that concept, can you really treat MDR, XDR, TB? So for that we used clofazimine, of course, and then rapamycin for inducing autophagy. And yes, indeed, we can treat MDR, XDR, TB. So this is the last slide. What it says for various type of TBs. What are the things strategy we should take it in our thing. So these are, you don't have to give attention. These are covered by various medias throughout the globe. And in fact, American Museum of Natural History made some of the, our work, the documentary, and these are all ABC News, Fox News, or whatnot, Brigham, all these. So basically, the last but not least, I have to acknowledge the people in my laboratory. These are the people, they work very hard day and night for achieving all these success. So this is But I don't want to give you a gloomy picture, but sometime back his TB is known as the romantic disease. And now the last slide is the world is as happy as you can make it.
Next speaker is one of the organizers of the conference, Dr. Nishit Agarwal. He is a professor at DHSTI and has been principal investigator of a number of extramural and collaborative research projects. His long-term goal is to understand the underlying mechanism of TB pathogenesis at molecular level. Currently, Dr. Agarwal's group has been involved in the identification and characterization of novel and essential metabolic pathways in MTB using in-house CRISPR mutant library, screening of small molecule inhibitors that target essential pathways of MTB, and understanding the host responses to MTB infection. And he has authored more than 30 articles in reputed journals. Dr. Agarwal.
So thank you, Dr. Sirolia, for introduction. So on behalf of the organizing committee, I would like to thank everyone for their overwhelming response. So this is so nice to see all of colleagues and all associates we have assembled here. So today we have heard a lot about the basic biology, and I would like to add one more talk in that series, but the title is something on a different line. So this is concept of the post-antibiotic effect, which has recently been introduced. I mean, it's not so recent, although, but it has been in the context of tuberculosis. The seminal papers has started coming, and how we have come across the role of a novel transcription regulator on the post-antibiotic effect. So what is the post-antibiotic effect? It's the duration to resume growth after removal of antibiotics. So if you grow bacteria in the presence of antibiotic and remove the antibiotic treatment and plate on the agar plate and monitor growth, something like you see a delayed growth recovery on the plate without antibiotic, but after the brief antibiotic treatment, what is called the post-antibiotic effect. There are papers which describe that this post-antibiotic effect can be regulated by the transcriptional inhibition, translational inhibition, or the efflux of the drug. In this seminal paper, by modeling, it was anticipated that if we can fasten the degradation of ribosome, or slow down the efflux of the drug, or slow down the synthesis of the protein, we will see slower recovery and more post-antibiotic effect. So that indicates that somewhere the proteostatic machinery, the homeostasis of the protein, basically plays a role in the post-antibiotic effect. And if you look at the spectrum of the protein in the cell, it was observed that around 70% of the proteins maintain the level of the 10 to the 3 to 10 to the 4 molecules per cell. So what is protein homeostasis? It is also called the proteostasis. It's a network, it's an interplay between the protein biosynthesis, protein degradation, and eventually leading to the folded form of the protein, which is functional form. It is vital in all the living organisms, and it is regulated by a complex integrated pathways. So if you look at the proteostasis network, it aims to protect the functional proteome. It starts right from the mRNA to the translation of the protein. And the proteins, either they are folded by themselves or they require certain chaperones into the folded state. And those proteins which are misfolded, they are either aggregated and go to disaggregases, or they're degraded by the proteolysis machinery. So I don't think that I need to introduce the importance of proteostasis, because a lot of diseases, the neurodegenerative diseases have been found to be associated with the proteostatic machinery defunction. And almost every disease involves disruption of cellular proteostasis. So the maintenance of proteostasis is linked to the cellular longevity and producing productive aging. So the primary area of our research group is to target the proteostatic machinery. Some of those I have highlighted here, and there are, today's talk, I'm going to introduce this gene, which is R1830, which a few years ago, it was uncharacterized, and recently we and others have characterized its function. So there are series of posters on other work, which I would request all of you to go and provide your feedback and interact with the students. So how did we start? So there's a graduate student in the lab now. She's in the final year of her thesis work. So she started digging in and finding out the genes which were essential and having the transcription regulation function. And we found that 2 of such proteins were and remain uncharacterized. So at that time, we annotated that as a CETR, conserved essential transcription regulator. When we look at the modeling of this, we found that it has a highly disordered N-terminal region, and the AlphaFold predicts its monomeric nature. But the crystal structure is not available, so we do not know the real conformation of this protein. Remarkably, when we perform the phylogenetic analysis, we find that there's a distinct clustering of this protein between the slow-growing and the fast-growing bacteria. So that was very intriguing, that the proteins and the protein homologs from the slow-growing bacteria are clustered together and supposed to be evolved together and distinctly from the fast-growing bacteria. So to start with, we first found out that this gene is expressed individually, not as an operon, because it's closely surrounded by other genes in the vicinity. And using the antibody, we found that the expression is quite conserved over the growth, indicating that possibly this gene plays some important role in the pathophysiology and the physiology of the bacterium. However, as it happens, in the 3rd or the 4th year, we came across this paper from Yang Abahu from the Wuhan University, who for the first time showed that there's an interplay between this gene protein, what he called the MCDR, and YB2, and they claimed that it controls cell division and DNA repair in mycobacteria. Soon after, there's another paper was published in Science, just in December, by Sarah Fortune's group, where they have identified that TB treatment failure is associated with the certain point mutations in this protein, and this leads to the antibiotic resilience. So what they call this as a RESAR. If I can elaborate on it, so they have analyzed the genome sequence of almost 15,000 genomes, clinical isolates, and found that there were many non-synonymous mutations were found in many such genes. And this was one of the genes where the ratio of the mutation in the drugs-resistant bacteria was almost 1.5-fold higher than which was observed in the drug-susceptible bacteria. So what they have done was that they created this point mutation mutant variant R95C, which falls in the helix-turn helix domain. And they reported that because of this mutation, the recovery becomes faster after the antibiotic treatment compared to the wild type strain for a variety of drugs, which some of them are also in the clinical use. To understand the mechanism, they focused on the gene YB2 and FBIA because of the previous study. However, the reason they chose also was that there were some unfixed mutations were observed, which were sometimes there, sometimes not there. And they did the chip analysis and found that this protein was majorly binding in the region, in the intergenic region of the YB2 and FBIA locus. However, this is a quotation from their paper that the magnitude of the resilience phenotype in the YB2 FBIA mutant appeared to be smaller than that of RESAR mutants, suggesting that additional as yet undefined mechanisms might be involved. So at that time, the mechanism was not known. And simultaneously, we were working from here and struggling to get some phenotype. So the questions which remained to be answered from these 2 studies was that why it is essential, if it is essential at all, and if so, then why it is essential, and how does it control the drug resilience if YB2 is not involved in it. So we started working with these 2 questions in mind. And just for the sake of students who are sitting back in this podium, so we have developed this CRISPR interference tool, and this tool is a very simplified tool now. used largely by most of the labs, not only in tuberculosis, but for any other organisms also, for providing the disruption and suppression of genes. So a few years ago, our lab has designed this interference tool, what we call the CRISPR-I tool, where we mutated the 2 amino acids and made the Cas9 into a non-functional Dcas9. And as shown in this cartoon, as we increase the dose of Dcas9, we can see a dramatic decrease in the gene expression. It was very useful for us to distinguish the genes which are essential versus the genes which are non-essential. Not only that, we have also created a multiplex CRISPR-I where we can simultaneously repress genes as many. I mean, in this example, we have shown all the 12 sigma factor genes, but theoretically, there's no limit for how many genes you can simultaneously suppress. And I'm happy to see that this is being used by all over the world. And using this tool, we have created almost 600, more than 600 knockdown strains in the lab, also covering almost all the genes which are predicted essential by the transposon mutagenesis. So coming back to 1830, first we wanted to see its effect on the silencing, effect on the growth. So we designed the guide sequence against the promoter, and we found that there's a strong dose-dependent suppression of the gene, both at the RNA and the protein level. Looking at the growth, we found that interestingly, this leads to bacteriostatic growth. So this was the first observation which we observed, that it is not killing bacteria, but it's leading to the bacteriostatic growth effect, either in the dose-dependent or the time-dependent kinetics manner. So to understand the MTBP its effect on the physiology of bacterium, we did the RNA-seq, and we intentionally kept its suppression twofold so that the bacteria don't go into static phase of growth, early time point. And among all the genes, the genes which caught our attention was those which were involved in the information pathway, because almost majority of them were downregulated. And when we look at them closely, the entire ribosomal genes were downregulated along with certain tRNAs which were downregulated in the mutant strain compared to the control. And the tRNAs were basically upregulated. So there was a reverse correlation suggesting that something happening at the protein synthesis pathway. And we've also performed the metabolic analysis with the help of Yashwant in THSTI, and found that several of the amino acids were getting accumulated, again reiterating that probably the protein synthesis is getting affected, and because of that, the amino acids are unable to be metabolized in the cell. So an excellent collaborator, Prem, is sitting here from RCB. So he helped us in doing this ribosomal profiling, and what we found surprisingly that in the mutant, all the ribosomal components were downregulated, which were highly complemented by putting back the wild type copy. And using the puromycin assay, we showed that the rate of translation is also getting affected in the mutant, which was again coming back to the near to the wild type level by complementation with the wild type copy of the gene. So now we wanted to understand how this is regulating protein synthesis, because this is a transcription regulator, and but why so much effect on the protein synthesis genes? So then we looked closely at the operon and took use of the Youngabo's paper, where he has identified the sequences at which this protein was shown to bound. And we first what we did was that we cloned this promoter region under the reporter gene and looked at the expression of this promoter in the background of the 1830 knockdown, and took another unrelated promoter as a control, and found that there was a significant drop in the activity of the promoter of RPLN operon when we are down-regulating RV1830, suggesting that indeed this might be regulating the expression of the large subunit of the ribosome, and that is why we are seeing some effect on that. So when we map the consensus sequence which they have identified in that paper, we have found that there was fairly good consensus present upstream to the -35 region. So what we did was that we amplified the full length and the truncated version without the prospective consensus sequence and subject them to EMSA. And as you can see here, there's a dose-dependent binding of this transcription factor with the wild type full length, but not with the truncated version, even at the highest concentration which we have used. So that proved that it is indeed binding with the RPLN promoter. Although with the kinetics, I will not say that's a very strong binding, but as we know in nature, we don't always need the strong binding. Sometimes the weak binding also works. So we were quite convinced that probably because of the direct binding, this protein is regulating the ribosomal operon, leading to protein synthesis defect. So as I showed earlier, there is a clear-cut dichotomy between the fast-growing and slow-growing bacteria, homologs of this protein, those bacteria. And when we compare the sequence, surprisingly, what we see that this dichotomy lies only at the N-terminus. So there's a strong difference in the sequence between the fast-growing ortholog and the slow-growing ortholog at the N-terminal region. So we wanted to check whether there's any effect of this on the binding affinity. And as I can see, show you here, that the protein purified from Isogmatis, which has the sort of deleted N-terminal region, shows a weaker binding compared to the one which is coming from the wild-type MTB. So again, we went back to the MTB component and did certain truncation, and again proved that the removal of N-terminal region completely abrogates the binding with the RPLN promoter, but not the removal of C-terminal region, suggesting that the N-terminal plays a critical role and which is also very disordered. So coming back to the resilience phenotype, because while we were doing this, this paper came on in Science about the drug resilience. So we wanted to test whether it has any correlation with the resilience phenotype also or not. But for that, we have to understand how this can be regulated at the protein synthesis. So I came across this very elegant paper, very nicely written paper, which titles as a number game, ribosome densities, bacterial growth, and antibiotic mediated stasis and death. So what authors did here was that they sequentially deleted ribosomal operon. So it has 7 ribosomal operon. So D1 to D6 means more to less ribosomal operon. So this one has only 1 ribosomal operon. And then they treated the bacteria with all the bacteriostatic drug, tetracycline, azithromycin, and chloramphenicol. And the take-home message from this paper is that the bacterial susceptibility to bacteriostatic drugs increases as we move forward with the decreasing number of ribosomes. And the duration to recover from the antibiotic effect, which is shown in this graph, is also increased. So in short, what the message which was, which we obtained from this paper is that the ribosomes somehow are involved in regulating the post-antibiotic effect. So we tested the resilience phenotype with the mutant strain, and as we can see here, the kill curve analysis, there's no significant change, but we found a dramatic change in the post-antibiotic effect recovery. So what the phenotype they observed, we found the reverse phenotype of that. Means when we mutate 1830, we see a much slower recovery compared to the wild type against the frontline TB drugs. Again, we wanted to test whether it is regulated by RPLN. So we did the similar analysis with the RPLN mutant and got the more or less similar result. However, when we did that against the YB2, there was no change. So this clearly indicated that possibly the regulation of resilience phenotype by 1830 is mediated via its effect on the ribosomal genes, not through the YB2, although I'm not contesting that it is not regulating YB2, because like Youngabo's paper, we have also observed there's a strong binding with YB2 promoter. But at least as far as the resilience phenotype is concerned, we did not find any correlation of RE1830 with YB2. So because we found the bacteriostatic effect, and like we know that many bacteriostatic drugs show the bactericidal effect in the during infection, so we wanted to test whether it has actually any effect on killing bacteria in the host during infection when we were down-regulating this gene. So we planned a very simple experiment where we mutated, we infected with the mutant, and then a subgroup was given only sucrose versus the one which was given sucrose plus doxycycline to suppress the gene. And as we can see here, there was a significant drop in the survival of bacteria as against its bacteriostatic effect in vitro, both in the lungs and the spleen. And also we see a very dramatic change in the profile of the pathology of the lungs infected with a mutant compared to the wild type. So in summary, what I've shown here is that RE1830, which is annotated as RESAR, I'm taking it as a RESAR and not CMDR because the phenotypes what we have studied was more of them were related to the resilience phenotype. It controls the post-antibiotic effect in MTB. Conditional silencing of RE1830 causes static growth in vitro. How the transient silencing of 1830 results in massive transcriptional reprogramming with the perturbation of around 20% of the entire MTB genes. So most likely these genes are not directly affected. A large chunk of these genes are because of the effect of this gene regulator on the ribosomal genes and operons. And we have shown that this silencing of this affects the availability of translating ribosomes and protein synthesis in the cell. And the N-terminal plays a major role. Because this we have found essential for virulence in MTB and TB pathogenesis, we believe that even though the clinical mutations have been found enhancing the recovery, but if we can suppress the expression of this gene with the help of some inhibitors, it can be explored as potential anti-TB drug target. So with this, I would like to acknowledge so the work which was done by this extremely hard-working graduate student, Pramila, and this guy, Yunus, is very talented, is just joined. So both of them were very aggressively involved in this project. And the support which I got from Prem for the ribosome profiling, Yashwant for metabolomics, Dirk for providing the ATC indicible system, and Bill Jacobs was there to provide whatever strains and the plasmids I have asked him for very generously. And lastly, I would like to thank DBT and the CSIR for providing funds. So thank you very much once again for your attention. Thank you.
Brings us to the last speaker, Professor Deepak Saini. He is a professor at the Department of Developmental Biology and Genetics, as well as associate faculty at the Center of Biosystem Science and Engineering and Center for Infectious Disease Research at the Indian Institute of Science, Bangalore. His primary interests are on understanding fundamental processes in cellular signaling, with special emphasis on signaling pathways in age-associated disorders and inflammation. Another area of interest of his includes deciphering fundamental designs in intracellular signaling in MTB using two-component systems as a model. And he interacts very closely with engineers as well as chemists to develop newer areas. Thank you.
Thank you so much for the introduction, and thank you organizers for asking me to come and give a talk here. And first of all, more thanks to the organizer for allowing me to talk a translational talk in the basic research program schedule. So why I'm going to talk a little bit about bacterial pathogen detection. I think this infection I caught when I was doing my PhD, and I have to thank Dr. Tyagi for that, because she had one arm in diagnostics, and I think we got exposed to very interesting challenges, merits, demerits of why we should be doing diagnostics. But that doesn't still explain why I actually started to look into diagnostics. And this part, I have to acknowledge the people who were involved in this entire study. And this is, the genesis of this project is actually because of the multidisciplinary nature of Indian Institute of Science, where you tend to bump into chemists, spectroscopists, some computational and data scientists, and also some microbiologists like us, and who actually ask a question, can we put everything together in one box and generate a Bisibella Bhat and see what comes out of it? So this is a Bisibella Bhat which comes out of this collaboration, which I've had with a lot of interesting people. And what's interesting part is most of the work is actually done by chemists and computational data scientists. A lot of biology has been done by clinicians in Bangalore Medical College. And my part was just to make sure the Bisibella Bhat does not become too spicy for everybody, to keep you toning it down to the right amount of information which goes to everybody. So what is this talk going to be all about? I'm going to talk to you about something which has found a lot of applications in biology. More recently is Raman spectroscopy. And I'll tell you about how we have been using Raman spectroscopy platform to come up with a very interesting diagnostic platform, which we think can change the way we process samples and collect data to identify pathogens. And I'm going to present you some data which does not limit it to only to tuberculosis, but also how we have used it to look at escape pathogens and how we can diagnose or detect escape pathogen using direct clinical material. So I'll skip this slide because I've already talked about, but I'm also going to talk to you a little bit about how we used a computational approach of deep learning, which is slightly more detailed form of machine learning, to come up with a platform where you can detect pathogens at a single bacterial level using a Raman microspectroscopy platform. So this is the list of bacterium, which does not need an introduction to the audience here, but I put MTB separately here because this is where we started the project. It was a happenstance that there was a talk given by one of the spectroscopic, Professor Umapati, who was talking about detecting explosives without really opening the luggage. Now, not that I was interested in explosive, I don't have any interest in explosives, but I thought the technique looks pretty interesting and pretty powerful to look at biological signatures. And for the discussion with him, he actually got interested. Let's see if we can actually adopt Raman spectroscopy, and if we can hook it onto a microscope to make a Raman microspectroscope, and if we can detect Raman signatures from a single bacterial level. So later, these are the pathogens which we ended up studying using the collaboration with Bangalore Medical College in the later part of the study. And this slide does not really need introduction to anybody that why we really need to identify bacteria from clinical material. There are a lot of reasons. One main reason is the limitations in the conventional approaches. So when we get bacterial material from a lot of biological fluids, there are many ways clinicians embark on for diagnosing the bacteria. There are very traditional, very cheap, and very effective ways to culture. The only problem is it's very time intensive. It takes a lot of time, especially for a bug like TB. It happens to be a very rate limiting step. Techniques like Vitek and BACTEC have reduced the time, but still is a technical challenge because you want to have result as early as possible. Sequencing approaches, nucleic acid-based amplification methods, or MALDI-TOFs have actually found a lot of application and diagnosis, but all these techniques require trained manpower, expensive equipment, and also we need to break the bacteria open to extract material for which you want to process, which is a very interesting challenge, which has been solved by multiple groups in multiple formats, but it still stays as one of the major bottlenecks for diagnosis. Now, what we thought that can be circumvent all these processes. So these are the list of pros and cons, which I think you can read from here, but main issue is the time and expense involved, whether we can do identification of something called viable but non-culturable bacteria, VBNCs. And I'll show you a little bit of evidence in the end that we think the Raman can actually also identify VBNCs directly from clinical material. We still have not reached a stage where we can actually put it as a device, but that is something which is we are on process of developing for future. And there's the cons of various techniques. I mean, basically their ease of processing, specificity, sensitivity. The yardstick of a diagnostic test lies on how sensitive and specific the platform is. So what we were aiming for to have a rapid, culture-free, label-free, non-invasive method. It was too much to ask to begin with, but this is a slide which we made afterwards. We have done everything. Then we realized this technique turns out to be meeting all the criteria which we think we should have. So one of the major challenges in trying to look for a bacteria from a clinical material is that amount of bacterial sample which you can get. This sample technically turns out to be very, very limited in volume. And the way we amplify the bacterial volume is by culturing them so we can identify them. Now, once you culture the bacteria, we would like to identify it by breaking open the bacteria, isolating DNA, RNA, and proteins. What we aim to do was to look at the bacteria without culturing, but we want to put it on a substrate which gives us the spectral signatures. I'll tell you about what spectral signatures Raman gives us, but the main challenge which we faced in our studies was how do we collect Raman signatures? So Raman signatures generally is collected from chemicals. You know, you have explosive material or some polymers which work beautifully, but in biological material, the choice of the substrate on which you put this biological material becomes very critical. We started putting our samples in glass, which was very easy. Turned out to be it was the crappiest material you can use with because Raman is inherently a very weak phenomenon. A lot of scattering was coming from glass. So we abandoned it. We went to quartz, more expensive, very difficult to make quartz cover slips. Again, junked it. We went to aluminum, magnesium. We kept trying and we kept failing because of a variety of reasons. That we were not able to get a material which was so smooth that we can put it under a microscope and we get a very smooth scattering coming from the sample, but not from the material. So that became a challenge, and that challenge was solved by another group of engineers who were there. And one engineer or happenstance, he was talking and he said, why don't you use a silicon wafer, which is used to make chip? So it's very smooth and very, very clean. So actually what we found that silicon wafer actually does the perfect job. So this is a silicon wafer where we actually put a biological material and then we shine incident light, generally very high power incident light, because Raman phenomena is inherently a very, very weak phenomena. What happens when a sample interacts with a biological material? So there are multiple things which can happen when light interacts with material. It can come back as a Rayleigh scattering, which is the most dominant signature here. It can also come as Stokes or anti-Stokes Raman scattering. Stokes scattering is when the wavelength of light shifts to a higher wavelength, and anti-Stokes when the wavelength shifts to a shorter wavelength, which is very, very rare. But this tends to be still a stronger compared to this phenomenon. So we focus on Stokes Raman scattering and other phenomena, which is the fluorescence. So using appropriate optics, we can get rid of Rayleigh scattering, the fluorescence signal, and we can focus on Stokes Raman scattering. So the idea is very simple. When a high intensity laser beam interacts with a biological material, we're going to get Stokes Raman scattering. We take the scattering, pass it through a spectrometer, and we look at the wavelength shift from the excitation or incident light to see what kind of signatures are we getting. And these wavelength shifts happen because of interaction of light with various chemical entities present in the material. So what could be the advantage? It's non-invasive. We are not ripping open the cell. It's non-destructive. It's rapid. It's label-free. Requires small sample volume. We can do it in a single bacterium. And beautiful part is has minimal interference from water. So we take biological material. We don't worry about water. We put it on the Raman substrate and start collecting spectra. That's the easy bit. And this is the setup of a Raman spectrometer. You can see the way we separate the excitation light is using a bunch of notch filter, diffraction gratings, and also some sort of spectrometers, which allow us to collect this sort of Raman signature. Now, this is basically Raman shift from the incident light, and each of this peak corresponds to some sort of chemical signature from the source where the scattering is collected from. And this signature tends to be very specific to the material it is coming from. So over the years, what people have done, they have actually collected Raman signatures from a lot of biological material, and we have assigned each one of these peaks to specific biological entity from which this scattering comes from. So I will skip this part because I've already talked about that, how in the biological material, you can have a lot of different chemical entities, and each chemical entity tend to give a very specific Raman signature. And this is what we did. We actually had a Raman bacterial sample, and there was a processing step which we included, and this is something with the clinicians' resistance to do something new was very critical. They simply said, I have a technician which is trained in ABC protocol. Don't change anything. So that became a challenge because when we had to put the sample to a Raman spectral acquisition, we had to make sure the user is not exposed or the operator is not exposed to a pathogenic organism. So we had to come up with some interface where the sample is processed, but at the same time, the pathogenicity is lost, but we do not have a lot of spectral characteristics changes such that we can identify the bacterium. And once we get the spectra, we do something called as pre-processing, where we get rid of the noise coming from spectral or noise coming from cosmic rays, random noise, and we get a Raman spectrum. Then we flatten the Raman spectrum by averaging it, and we do a computation analysis to find out where is that spectrum coming from, what is the source of that signature. So this is the substrate development which I've told you about. So what we ended up after making the substrate, we actually first did something which is identify the bacteria using non-pathogenic bacteria to see if we can actually effectively detect bacteria with good specificity. Then we try to look at if we can inactivate the bacteria so that it becomes very innocuous, non-pathogenic. And then we again use the platform to see, can we separate live and dead using such a platform after processing the sample. And what we did initially was to use certain very well-established methods used in the microbiology lab to fix bacterium. We use paraformaldehyde, sodium azide to kill the bacterium, which is very short duration exposure, and glutaraldehyde. And we collected Raman spectra to see which of these signatures actually allow us to, which are the closest to the live unfixed bacteria. This was done with all soil microbacteria, which is M. smegmatis. Though we saw differences in almost all fixation steps, we found the paraformaldehyde tend to be the closest to the unfixed sample. And paraformaldehyde is something which is widely used to fix samples. And for TB also, it's quite effective. We stuck with this design of things. So what we do is we take the sample. This is for how we build our database. In the database building exercise, we took clinical material, grew the bacteria on a plate, fixed the bacterium, and collected the Raman spectra. And collecting Raman spectra was done after fixation with paraformaldehyde and just some water washes to get rid of the host material. And then we mounted them on a substrate, which is a silicon wafer, collected the Raman spectra, and this is the various sources of the biological material which we use to make our signature database. Now, traditionally, people make database by lab-grown cultures, which is not the right representative to make database, which we also tried first, and we realized we are not able to match a lot of signature to clinical material. So then we started making database using bacteria, which is actually coming from clinical sources. So we use Staph aureus from pus and blood, E. coli plus urine, and all these different sources of bacterium, which is actually from clinical material, collecting from clinical material, were used to make a Raman database. And once we did the database, we actually started subjecting our signatures to all kind of Raman processing, which I've already talked about. And then what we do, something called as spectral normalization. And after normalization, we get a clean spectra. And then we've used the clean spectra to identify what does the spectra telling us. The easiest is univariate analysis. What is univariate analysis? We look at peak 1 and compare with the peak 2 between two samples and say peak 1 is bigger in E. coli and peak 2 is bigger in Pseudomonas. So peak 1 sample which gives peak 1 is E. coli, sample which gives peak 2 is Pseudomonas. I wish the life was so simple when you're identifying. It is not. So peak were all over the place. What we have to do is something called as multivariate analysis. This is all called chromometric analysis. So what we did, we tried to look at multiple peak ratios. So we do peak to peak ratios to identify if we can do something called multivariate analysis using unsupervised and supervised analysis if you can discriminate. And what happened initially was the following. I will skip this part here, but initially when we're trying to do this ratiometric analysis, which is univariate analysis, we were able to identify certain bacterial species quite effectively. And when we do with 4 bacterium here, we got very nice clustering of 4 different bacterial species. Basically, based on signatures, we were able to classify each bacteria as individual species type. But this was what was done using a lab-grown bacteria. The thing was very easy. The problem started coming when we started dealing with the bacteria coming from clinical samples, and we got a lot of spectra collected, and we subjected them to Raman spectra analysis. When we looked at 2 bacteria, we were able to discriminate beautifully. When we had to do, this is called principal component analysis, we could discriminate between Pseudomonas and Staph aureus. And then we started doing 3 bacterium and things started getting dirty. We said, okay, this is getting messier. And when we started doing for larger number of bacteria, what we ended up getting was this, which is exactly what we anticipated, garbage in, garbage out. So a lot of information, we could not make head and tails exactly what is identified here. And that's where we hit a bottleneck. And we said, okay, no, we don't have a way forward because all the tricks which we had about sleeves, about univariate, multivariate analysis, we ran out of them. And that's when our computational guys stepped in. He said, you know what, all your data is a digital data. For each wave number, you have an intensity information. Let us run them into machine learning and deep learning algorithms. And that's when exactly we actually started subjecting, this is what pre-machine learning algorithm, and then what we moved into deep learning. So what is deep learning and what is machine learning? I'll just quickly spend roughly 2 minutes on this. In machine learning, we decide the rules of discrimination. Let's say there's a stop sign and there is not a sign which says do not cross. Basically, an operator can put in parameters which machine will learn to discriminate between A and B. So operator puts features, extract features from various input data and tells machine what it should be doing. And this is what machine learning does. Basically, it tries to learn from the parameters which has been set in by operator. What is in deep learning? What deep learning does is we give them a lot of data set to a machine and said, A and B are different. I do not know how. You need to find out how A and B are different. You take up whatever feature you want, but you need to tell me accurately every time I give you an unknown sample set, you need to tell me it is A or B. So in deep learning, the intervention for feature extraction by an operator is not there. The machine learns it by itself. It does feature extraction as well as classification and allows us to give a predictive algorithm which can say, is sample A or B or not? Now, what is the problem is we do not really know what is under the hood in these deep learning algorithm. We do not know what feature it extracted. But this is something which is an area of investigation where people like biologists want to go back and see what is the feature using which it did the discrimination analysis. But we applied this deep learning platform, which is called the ResNet, and we use this to extract features from the training data set, which came from the clinical material. And using this training data set, we applied our Raman spectra, and then we start to validate our data set. So what we did something called as blinded data analysis. And what we found that our deep learning method was quite reliable in terms of sensitivity and specificity score. And we were able to predict most of the pathogens coming directly from clinical strains quite effectively. Now things get very interesting here because at this stage, we were planning to do some blinded study with clinicians. And what happened, there was a miscommunication between the person who was collecting the sample and my student. And what happened, they started giving us the biomaterial without even trying to do typing at their end. So they were saying, you take the material and we do it our end. And my students also start running it by completely blinded. That means both the operator and the sample collector person were blinded. This is a double blinded study. And a lot of samples are not even cultured to validate. And then using this deep learning platform, we realized that not only we are able to identify them, we are able to identify them with very high specificity and sensitivity. So what we have using a blinded data set, which was used to training, we have actually got almost 98% accuracy at a single bacterial resolution using a Raman microspectroscopy platform to identify clinical bacterium. Now, can we use this to assess viability? This is the last few slides. We tried to use this in MT, Mycobacterium smegmatis. We killed it with a bunch of antibiotics, various degrees of death was seen. And then we tried to classify them in a live, dead, or fixed category using a fixation platform. But what was interesting is here that when we were able to fix the bacteria and we used to classify them in dead and live classification using Raman. So the way we did it, we treated the bacteria with a bunch of antibiotics, ensured the bacteria is dead, put them on a Raman spectrometer, put the live one, allow the back system to train itself, and then we gave an unknown bacteria, say, tell us not only what it is, tell us whether it's live or dead. And it was able to classify them very accurately and at the same time tell us whether it's living or dead. Now we think this platform will help us identify VBNCs or viable but non-cultivable bacteria in clinical samples. So to summarize, we have a robust methodology for bacterial identification of clinical samples using Raman spectroscopy platform, using deep learning methods, which allows classification of bacteria using various classes. The conventional methods of Raman data, which is like PCA, LDA, we are not able to discriminate clinical material, which is anticipated. But what we found using deep learning, we are able to classify them very accurately. Now, as a biologist, we would like to identify or interpret the basis of these deep learning methods of what lies under the hood, which allows us to discriminate based on biological signatures. But the aim is to develop a Raman spectroscopy platform as a proof of or a point of care devices. So I'll stop here and thank you for your attention.
Thank you to all the speakers and the audience. We will now take questions. Please be brief. I can say already that all of the speakers have done really wonderful job. So you can probably keep the appreciation, save the appreciation that can be done during the lunch time. So you can just go straight to the questions and short questions, and from my colleagues here, short answers, please. Yes, please go ahead.
Shall I go ahead? Yeah. So my question is for Nishit. Hi. My question is for Nishit. Nishit, really nice talk. I wanted to know if the clinical mutation, you've checked whether it has a gain of function. Whether the clinical mutation that you found in 1830, whether that is a gain of function mutation, because the phenotype is opposite.
Yeah, so that is what we are pretty much interested to look forward. So we are creating those mutations and try to find out whether it has a gain of function or not. So that's a good suggestion. Yes.
Thank you. My question is for Deepak, sir. Hello, sir. Excellent talk, and you've developed a very interesting model. My question is, will your model be able to differentiate between, like, there are so many species in the MTBC complex, right? So will it be able to differentiate between all of them, because they're like very similar, they're 99.9% similar, right? So if there's a case of mixed infection, will it generate different Raman signature, and will we be able to differentiate which is the causative agent in that case?
So the short answer, as Umesh told, yes. It all depends on how good is your training data set. If you can train the system well, you can distinguish between very fine features also.
Okay, thank you, sir.
This is for Amit Kumar Pandey. Thank you. Lovely talk. Why do you think
the bacteria MTB has retained VAP-C12 instead of getting rid of it?
Well, to keep it very short, I believe MTB really wants to stay there. So, and cholesterol, as we all know, is all around. So my take would be that, this is what is driven by MTB, and this is a way to modulate host and keep the host alive so that, it can propagate and persist. So that would be my take.
Yeah, so to Deepak, very nice. Have you tested this in complex samples like sputum or tissue sections?
Yeah, so if you see it's a very interesting question, by the way. So we did test them in sputum and pus and blood. So what we found, that is something which we were actually kind of worried about, because clinicians tend to base their identification based on a single signature. We were getting some complex signatures in our Raman. But when we went back and talked to the clinician, they actually said some samples actually had multiple infections. So yes, it will work as long as you can detect bacteria at a single cell resolution, and you can collect multiple spectra from multiple samples. You should be able to work with sputum and all other biological fluids also.
Sorry.
But after processing.
Just a follow-up. So like you scan this entire area?
Yes, we scan an area. So the way it works is we scan a low resolution scan with a biological signature, which is well conserved, like scanning for DNA. And once its DNA areas in the visualization field is mapped, then we do a deep scanning of that area to see what kind of signatures come out from them. So there's a low resolution scan and then deep resolution scan.
Thanks.
Deepak, yeah, nice talk.
Thank you, ma'am.
I wanted to know that have you been able to correlate the different peaks in your fingerprint with any particular chemical identity, like cell wall proteins or glycoproteins or lipids or anything like that? Because that may add robustness to your interpretations.
So most of the signatures which we record actually are already mapped based on previous studies to certain biological entity like proteins, lipids, and all that. What we see is a change in ratios of the peaks. So it's very difficult to say what exactly is contributing to that peak, because it could be coming from multiple biomaterial. For mycobacteria, we do see a very nice signature coming from mycolates, and also coming from xanthines or molecules which are involved in oxidative stress, the colored molecules, which gives like carotenes and all that. They give very interesting signature. But whether they are responsible in the deep learning algorithm, I do not know that right now.
Yeah, I think a fantastic session, really nice. So this is for Amit, basically. I had one for Amit and one for Nishit. There's a very brief VAP-C. The VAP-C1. so it's a transcriptional repressor. So you see hypervirulence in vitro, and you see hypersusceptibility in vivo. Is it okay? Is it right what I'm saying?
So if I got the question right, it is metabolically more active. It grows fast. So that correlates with the drug susceptibility phenotype that we see in the host. So metabolically less active is more refractory to drug. So that is what MTB wants. But if you remove this VAP-C12 toxin, antitoxin locus, it starts growing. So it's a break that is off. So MTB is more metabolically active, grows fast. And in this presentation, I showed that holds true in vivo as well.
Okay, yeah. Just for Nishit, Nishit, the 1830, how do you really explain the post-antibiotic effect? What is the mechanistic aspects of it? It's very, very complex, because some beta-lactams have very small post-antibiotic effect, right? Aminoglycosides have large post-antibiotic effect. It sometimes is basically dependent on the antibiotic class.
Yeah, so you're very well right, Anil, because there are multiple mechanisms, as I showed in the first few slides. The effect is at the transcriptional inhibition, at the translational inhibition, at the efflux level also.
And depending upon which particular pathway is kicked in, you see the effect, the magnitude of the effect. So what I have found from the literature and from our study, the effect is very high if you target the protein synthesis. Because, and that a very clear example comes from the chloramphenicol versus streptomycin. One is the bactericidal, one is the bacteriostatic. So basically, if you see the post-antibiotic effect, it's nothing but the preparatory time bacteria take to recover from it after you have removed the antibiotic. So all those experiments which we do for that, basically we wash the cells off multiple times to remove the antibiotic and then let them grow on the plate without antibiotics. So it's basically the time which they take, and all depends upon which particular mechanism is hit by particular antibiotic. I think that is what I can.
I have two excellent sessions. I have two quick questions. One for Dr. Deepak, which is if you treat the clinical specimen before Raman effect, so for signature sequences, have you ever tried to, because very often the clinical specimen are multibacterial. So if you have treated within a specific antibacterial, bactericidal agent and then seen the Raman signatures, that is for you. And for Dr. Amit Pandey, I have one small question. When you showed the effect of carbon source, so you were showing that if there's a depletion of glycogen, so then the antibiotic effect is more, if I remember correctly. Is that right? So my question to you is that, was it a time period study or was it done immediately after depletion of glycogen? Thank you.
Okay, so let me ask you. So you're asking the question that did we treat it with some antibiotics?
Yes, to
No, we
compare
did
the
not.
two. Yeah.
We did not. We just took the material and we treat them with regular Petrov's method, fix with paraformaldehyde, wash with water, and go out for Raman spectra collection. There's no treatment which we do in samples.
No, like if you do it, you can see a difference between which one has
It's an
been grown.
interesting point. We did do some in the lab signature analysis. We realized our signature is actually coming from the antibiotics more than the biological material. So it made things really dirty. So we have not gone with the territory yet, but this is going to be a next challenge for us to work on too.
Sure, thank you.
So to keep it very brief, glycogen indeed it is required for drug increase in the frequency of drug persisters. So having glycogen helps TB to be refractory to antibiotic. Mechanistically, if you ask me, I don't know the answer, but the observation is that glycogen is indeed promoting drug tolerance in TB.
Thank you.
I have just a question for Dr. Das. How do you see clofazimine, which is antimicrobial drug, and high retention time to affect the survival of BCG if you want to use it to augment BCG efficacy?
Clofazimine actually is, as you know, it induces autophagy. Autophagy is one of the mechanisms to eliminate the bugs. So basically clofazimine, on the other hand, the clofazimine is the inhibitor for KB1.3. KB1.3 is majorly expressed in effector memory cells. So basically, if you inhibit the effector memory cells because they are in a dynamic balance, you produce more of central memory cells. So that is what your idea is, to increase the number of central memory cells, which is a long-lived, and that is, if it is antigen specific, that will give you the vaccine in future.
So I have a very short question here. So this is Kajal from Amity University. This is for Dr. Deepak. So I'm just curious. Since you checked so many materials that it doesn't have any background diffraction. So I'm just curious that the material which you use, the silicon, did it have any background diffraction or just no diffraction?
So there is always some background material from any material which you put to Raman scattering. It all depends on whether you can subtract that signature from your biological material. We realize with silicon wave, silica, we're able to subtract the background much clearly compared to anything else, which was, we used glass, quartz, that was getting more difficult to separate. So that's why this turned out to be a good substrate.
Thank you.
So my question is for Dr. Nishit. As you are using dCas9 for knockouting the cell, but so can we use CPF12 as it is in Cas12a for preparing knockouts? Like in this, we have to only synthesize about 25 nucleotide CRISPR RNA, but for the Cas9, we have to synthesize around 100 nucleotides sdRNA.
No, I mean, we have tried multiple things. CPF12 is one of them. The difference is that the the PAM sequences are 80 rich for the CPF one. And for Mycobacterium, as you know, that it is 67% GC rich. So finding out the 80 rich sequences might be less frequent than GC rich. So that's the one reason. And secondly, we have not tried the knockout, it's the knockdown. We have tried knocking out by disruption, and the success rate is very, very poor. So we are trying to understand why is it that. And you may see, I don't know whether it's there in the poster or not. So we are also trying the Cas alpha, which is the very shorter version of it. So hopefully we'll get some result.
Okay, thank you, sir.
I have a quick question with Dr. Amit, working on cholesterol. So I just want to know whether there are any reports which says that people on lipid lowering drugs are less prone to develop tuberculosis?
Actually, Dr. Dash showed the slide. So my take would be you have to have some cholesterol, and that is good enough for TB. So you can go up to a level to decrease your cholesterol levels, but the intracellular TB hardly requires any. And this will be the last statement here. Cholesterol as a carbon source is less important to me than cholesterol as a signaling molecule. So
I think because we are now just, between lunch, a lot of the questioning can be done during lunchtime. So we'll stop question answers now and back to the audience.
I request everyone to be
I request,
seated.
everyone to be seated. There are a few announcements. I know you guys are all hungry. Food is not ready yet. So if you go now, you will start having to cook a little. So best is to stay here.
I thank all the speakers for enlightening us on one of the most willful pathogen of all times. I kindly request the session chairs to felicitate these speakers. Professor Varshney to please felicitate Dr. Amit Singh. Professor Varshney to please felicitate Dr. Amit Kumar Pandey. Thank you, Professor Varshney. I kindly request Professor Surulia to please felicitate Dr. Gobardhan Das. Professor Surulia to please felicitate Dr. Nishit Agarwal. Professor Surulia to please felicitate Dr. Deepak Saini. I request Dr. Babatosh Das to please felicitate the session chairs, Professor Umesh Varshney and Professor Avadesh Surulia. I again thank all the speakers and the session chairs. I kindly request your attention for following important announcements. A, I request everyone to be part of the signature campaign by signing the poster that is placed right outside the door to your left. B, post that all delegates of the symposium are requested to proceed for the group photograph at the amphitheater by exiting the auditorium complex from the doors in the gallery, which is right behind you, which is connecting between the dome first and the dome second area. Participants are encouraged to visit the stalls put up outside the auditorium. D, in regard to the posters, I request all the participants and the poster presenters to be at their posters for discussion. E, the posters are placed in the first dome area and the gallery behind you, connecting the second dome area where the lunch is arranged. We will now break for the group photograph for the delegates and the poster session and lunch simultaneously. I request all of you to gather back in the auditorium by 2:45 p.m. for the start of the next session. Thank you very much.
Please proceed for the group photograph right now. Thank you.
Ms. Zora Hashmi. I am a PhD scholar at Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis, the disease. Person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months. Remaining MTB feels good during intensive period and stops medication. The surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance and hence until scientists find strategies to shorten this regimen. Completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance and hence until scientists find strategies to shorten this regimen. Completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease if the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease if the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous. Intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism
We'll start. Good afternoon to all. I, Kamala Priya and
Lavnish Thakur
extend a warm welcome to all of you for the succeeding sessions of day 2 of the symposium. Kindly mute your cellular devices, and I request the same for those joining the symposium online. We will start the program momentarily. Early diagnosis of TB is extremely important, not only to keep the burden, morbidity, and mortality low, but also significantly hamper the positive outcome due to appropriate anti-TB treatment. While diagnosis of latent TB continues to be a huge bottleneck in endemic countries to TB, simple access to existing diagnostic tools is often a hurdle towards rapid, timely, and appropriate care and treatment. Thus, it makes it very compelling to have the 5th session on TB diagnostics. This session is aptly chaired by Professor Jaya Tyahi and Professor Sandhya Vishweshwaraya. Professor Jaya Tyahi is a former NACI senior scientist and chairperson of the Department of Biotechnology, AIIMS, New Delhi. She has made significant contributions to the field of mycobacteriology, including discovery and characterization of the crucial DEV-R-DEV-S system of M. tuberculosis essential for dormancy. She developed proof of concept for DEV-R as a dormancy drug target. Her contributions to the field of mycobacteriology and TB research have had significant impact in improving TB diagnosis, anti-TB therapy, and prevention of TB transmission. Our next chair, Professor Sandhya Vishweshwaraya, is an honorary professor at the Indian Institute of Science in Bangalore, India. She has made significant contributions to the fields of cyclic nucleotide signaling and host pathogen interactions. She is a fellow of the Indian National Science Academy, Indian Academy of Sciences, and the World Academy of Sciences. We are indeed delighted to have them with us. Moving on to the next session, we have 4 talks in session 5, each for 20 minutes. Following the talks, there will be a brief question and answer session with all the 4 speakers. I request the audience to hold their questions until then. I urge the speakers to please stick to their time limits. The buzzer will ring at 18 minutes. I request the speakers, after their talks, to be seated on the dais for the following question and answer session. I now request the session chairs to kindly lead session 5.
A very good afternoon. It's always a tricky session after lunch because a lot of people are hanging over at lunchtime and also at the posters. So I have to only thank those who are here and do hope that the others join us soon for what is going to be the session 5, very relevant session of TB diagnostics. As already mentioned, we have 4 speakers and we will not spend too much time in introduction, but go right into introducing Dr. Sonali Sarkar, who is professor in the Department of Preventive and Social Medicine in JIPMER, Puducherry. And she has contributed immensely in improving rural health services in the community. She has more than 120 publications in well-established and peer-reviewed journals. And most recently, she has been very much influential as being a key member of the Report India team, which she's heading in the 2nd version. And she's playing a very important role in developing the protocols, setting up the labs and clinical capabilities to carry out this very large cohort study in collaboration with Boston Medical Center, Massachusetts, and Rutgers, New Jersey, both in USA. She has been a chair for the Clinical Epidemiological Working Group for Report India Consortium. And as I mentioned, she's Indian chair for the 2nd phase of Report India. So without further ado, I warmly welcome Dr. Sonali Sarkar to please inform us about the latest developments.
Thank you very much, ma'am, for that kind introduction. A very good afternoon to all. And as Dr. Jaya just mentioned, it is challenging to get people's attention in the postprandial session. So, and it's more so for me because I think I'm a relatively rare species in this group because there might be very few epidemiologists with a medical background among a large group of basic scientists. So I would be talking today about the TB diagnosis, the present status and challenges. Not much of data, would be focusing a little bit on what the present challenges we face, what are the present updates, and what we are doing, as Dr. Jaya just mentioned, part of Report Network in India, since last 1 decade, what we have done and what we are doing. So before starting, I would just thank Dr. Raghunanth. In his concluding remarks in his talk, he just mentioned the summary of what I would like to convey. It is about collaboration. That is the purpose I'm here among so many basic scientists, and he made my job easier. So to go ahead to bring you down, I mean, from molecules to cells to organs, to a complete human being, and that too at the national and at the global level. It's not new. All of us know that when it comes to the number of TB cases diagnosed, the estimated numbers always India has the largest bubble, almost 28% of the global TB burden post-COVID. Earlier we used to be contributing to the quarter, not only of all forms of TB, but also MDR, drug-resistant TB. India contributes to the largest numbers. And sadly, the number of the gap in the notification of TB cases and those estimated is also highest for India. COVID played foul for all programs, but TB was one of the worst affected. And the number of reduction, the proportion of reduction in case notification globally, same again, India shares the largest. The bars in the red show the countries which contributed to more than 90% reduction in the case notification in 2020 and 2021. Still, India continues to share the largest burden of the reduction in the case notification. We know that when cases are not notified, that means they are not diagnosed, they are not put on treatment, they continue to spread the infection. So the burden of TB, according to the India TB report 2022, there has been very minor decline in the incidence of TB, very minor. But what is worrying is the mortality due to TB has actually increased in India. So also globally, post-COVID, what we see is the mortality is increasing. The mortality increase means there are undiagnosed TB out there in the community who are dying because of lack of treatment, and they would have spread the disease to so many others, which would now have an impact on increasing prevalence and incidence for few more years till the program catches up with all of them. So the prevalence of TB in India, as per the ICMR report, the nationwide survey, which was concluded recently, shows the prevalence is about 316 per lakh, quite high. And all forms of TB, all ages, it is 312. There is wide variation across different states and regions in the country. And the report says that we have around 1/3 of the population which is infected. But more worrying is this scenario. The report says that out of the symptomatic population, nearly 64% were not seeking treatment at all. Why? Most of them ignored the symptoms. Many of them don't recognize the symptoms as because of TB. Many of them go for self-treatment, and many others do not afford to seek care. Now, this is a report about a study that was conducted to show the cost incurred by TB patients. This is the the cost that is incurred by TB patients due to the diagnosis at government hospitals. As you can see, this is in Indian rupees. Nearly 3,000 rupees is being spent at diagnosis, maximum going up to 10,000. Even in private, again, it's about 5,000 going up to. So the cost, it's not just private, but government also. Patients are spending so much on diagnosis, which is supposed to be free of cost to all patients in India. In a scenario where the population TB patients, their household income, almost 25%, a quarter of the TB patients earn less than $100 per month, which is maybe about 8,000. So with that income, they are having to spend on diagnosis about 3,000 rupees, 3,000 to 5,000 rupees. Now, I wanted to bring the cost into account because many technologies are now being developed, but as a public health professional, our concern is always to take the technology to the people at an affordable cost. So this is again a very well-known figure. At what rate we are now globally declining TB incidence, what we should do to reach the NTB goals. And this is not possible only by optimizing the current and new tools, but we have to develop newer tools. The WHO recognizes that the rapid and easy point of care diagnostic test is one of the key strategies to control TB globally and so also in India. So what are the difficulties in diagnosis of TB? We know this TB control relies heavily on the early and accurate diagnosis so that the chain of transmission stops there. But it is important to now diagnose the drug-resistant TB as well, because India isn't sitting on a silent bomb of XDR TB. So there are various forms of drug-resistant TB, mono, poly, MDR, RR, multi-drug, and now extensively drug-resistant TB, and some reports have also been made of totally drug-resistant TB. The identification of infection is also important if we have to control TB, because these are the people, 1/3 of the population. We know that lifetime risk of TB disease is about 5 to 10% among those infected. But what is needed is a rapid test and cost effective, which can help diagnosis of TB at an affordable cost, not only to the patients, but also to the country. The government is paying whatever the provision is made to the people free of cost. So diagnosis is still problematic because most of the diagnosis as of now is still sputum based, and we know sputum based tests are not very good because even though we have assays which are more sensitive or specific, but the expectoration of sputum itself is a process which is infectious, and most patients are not able to expectorate good quality sputum. So it's always challenging, more so in patients in TB, forms of TB which are paucibacillary, like the extrapulmonary TB. And extrapulmonary TB, again, the challenge is how do you get the tissues, or often it has to be invasive procedures to get the specimen like CSF for TB meningitis, and all forms of extrapulmonary TB, we require invasive procedures for diagnosis. Tuberculosis in children, people with HIV, and pregnant women. So we'll see each of them. Paucibacillary disease is the reason in children, but more because they are not able to expectorate, they cannot cough out, and that is why only 40% of the pediatric TB is actually confirmed microbiologically, and that hampers their treatment. The challenges in extrapulmonary TB, they comprise about 1/5 of the all TB cases, and as I said, it requires invasive procedures. We do have assays now which are better sensitive, like gene expert assays, but many researchers are now trying to identify biomarkers which would help diagnosis of extrapulmonary TB. Some of the markers have been identified, but they are still to be validated. These are small studies and few reports, and cannot be extended to the population yet. Challenge of diagnosing TB in pregnant women is also there, because pregnancy with TB increases the risk of perinatal mortality, and almost 6 times higher perinatal mortality for the fetus with mothers having TB. And this women who have TB and pregnancy, they have very unusual non-specific symptoms, and therefore many times they go undiagnosed. And in fact, 16% of maternal mortality in India is because of TB. We've often ignored, because maternal mortality we think are all reproductive, but not so. 16% is nearly 1/5 is due to TB. Challenges in diagnosing TB with HIV, we all know this is again paucibacillary, but co-infection is present in almost 8%. Thankfully, it is lower in Indian setting. It again differs from region to region. Some regions do have higher. I think Mumbai region has a higher prevalence of HIV among TB patients, but many others have lower. Then the frequency against smear negative TB and EPTB is more common. And whatever biomarker studies are done have not enough sample of PL-HIV, and therefore the power is low, and much more studies are required to validate those for effective use in the programs. Challenges remain in diagnosing LTBI. First of all, LTBI, now we don't use this term, we use TB infection, but TB infection is a state where people are asymptomatic, so they would generally not come to seek treatment. It has to be, the system has to go, health system has to go to the people and screen them and diagnose. But diagnosis is difficult. We know the problems with the tuberculosis skin test is tuberculin skin test is because it is cross-reacts with BCG. IGRA we have, but it is still very costly and not easy to do. Requires technical expertise, which is not available everywhere. So with lot of challenges, we are having developments also. In fact, before coming to this conference, I always felt that TB is neglected. Research in TB is neglected in India. But after listening to excellent talks in this conference, we realized that a lot of work is going on. Plenty of basic science research into various aspects of tuberculosis. But there is a need to link the clinicians, epidemiologists, people in public health to this research. And I think this symposium is a great initiative. So the WHO endorsed products we have as of now is the LPA, Gene Expert, LAMP, and TrueNat that is produced in India by MolBio. The other emerging technologies that WHO endorses are the centralized DST, point of care NATs, sequencing, and Expert Omni. Many of these tests you would know better. I will not try to explain them to you because they come from this research that you people do. So next generation expert assays developing on what we have, the CBNAT technology is on. Because this is one of the good technologies that came. But there are, again, the advantages are there, the sensitivity and specificity there you can see, but challenges this requires, again, technical expertise and the infrastructure facilities to run these tests. I mean, one of these challenges, the requirements is a power supply has to be there. And many parts of the remote parts of the country may not be having that facility to run expert. So battery-based assays have been developed, like TrueNat can work on battery. But again, there are difficulties in using the machine because many district health centers which receive the TrueNat as part of the ICMR supply are not able to use because though it is a cartridge based, but before loading the samples into the system, there are a lot of processes that need to be done where labs do not have those expertise. There are newer advancements also in expert assays. Various other types of experts are now coming up, which would be able to do the tests more point of care, closer to the patient, and also try to cover a wider range of drug resistance. I mean, resistance to drugs like INH, fluoroquinonal, ethionamide, second-line injectable drugs, so that we can diagnose not only RIF resistance, but also other polyresistant and also XDR-TB. But again, these are still in the development stage, and large-scale clinical trials are needed to validate these diagnostic tests for further use. So the WHO guidelines, again, I'm not going to go into the details about whatever the technology which WHO recommends for updating the diagnostic tests. LAMP is again another good test which is useful in peripheral health settings, but I'm not sure how much is it actually being used, because again, technology expertise to do this is not available in the resource constrained settings in our country, and the places where we have more of TB patients are the places where there's more of poverty, poorer health systems, and the facilities are not equipped to handle or use these advanced technologies. Lyme probe assay is also in use in different IRLs across the country, but still even though the advantages are there of ability to diagnose RIF as well as INH resistance, but use is not wide enough to detect all MDR TB cases in the country. So I'll skip this. This is a promising test. It is already marketed as by Eller and recommended for use in HIV screening of TB in HIV patients. Though the Eller product has lower sensitivity, it is as a triage test, it is good to rule in patients who would require further investigations, and therefore WHO has recommended, and it is already now started to be used in India. Other sequencing based tests are also now coming up, targeted sequencing, but we are still far away from using those in the community, in the health system. So this, as we know, still, though the costs are coming down, this cost is still high, and we do not have the lab facilities which can reach all TB patients. So novel technologies are now being developed, but many of, like we're hearing about so many technologies people are working on and have the potential to come up to to develop, come to a level where they can be tried and tested and come to clinical trials. That is really exciting, encouraging for us as public health professionals, but they need to be validated. I mean, we have to work more on, all of you know, this would need much more work where they can come to the lab for use in patient care. They also need, most of them would need algorithm generation, and they also run the high risk of bias. So use of some of the newer technologies which would not require sample collection are the AI-based cough sound, I mean, trying to diagnose tuberculosis based on the cough sounds. So this is found to be, I mean, a useful tool, can be done through portable digital recording or using mobile phones, digital stethoscopes, found to be having good sensitivity and specificity, but again, needs to be validated because there are a lot of other lung diseases which would be causing bias in the diagnosis. Then the other newer technologies that can be useful is the electronic nose, which actually tries to identify a signature of volatile compounds from the exhaled breath of TB patients. Again, studies report high accuracy, but they have to be validated. There's very early studies that show there are promises, this is promising, but needs to be more studied. This is more in use now. We have products like Cure AI and other many people are working, even in JIPA, we have our PhD students working on a platform which would be freely available to people because Cure AI is still costly. Use of AI in computer-assisted diagnosis of TB from chest X-rays. This is promising, has public health application because can be applied when we take large number of X-rays in screening in the community. So we do not have enough people to read those X-rays. So AI can be of help. So these are also good developments that are happening. So need of sputum-free TB test, we just discussed. We need to diagnose subclinical TB, and because these are actually people who are transmitting TB disease but do not recognize. Many of them are not aware that they have infection and they are transmitting the disease to others. One of the tests is the MTB aerosol capture methods through the mask, and then those are the aerosols actually can be cultured too and can help in diagnosis of and this can be diagnosed early even in subclinical cases. And tongue swab again is another development. This can be used for the samples can be used for LAMP, can be DNA can be recovered from the samples. A lot more tests that you propose, you are all working on can be done from the DNA that is obtained from the mycobacteria, if at all they are culturable from the tongue swabs. The use of stool, urine and blood samples would be of use in diagnosis of TB in children where they are not able to expectorate. So for blood, there are some antigens that have been identified. Immune cell profiling is proposed, host transcriptomics, and cell-free MTB DNA detection. All these are again in the development stages. And studies are able to find better association with mRNA signatures and TB risk. I just briefly tell about the Report India Consortium. We started in 2013 in collaboration with between NIH and DBT across 7 sites in the country. At JIPMA, we had the 2 cohorts developed of TB patients and their household contacts, a number of samples collected at different time points. Here, I would just stress upon the fact that we are waiting basic researchers like yourselves to come and utilize those. Please do collaborate with us. These are well curated samples, well characterized. We have all the clinical data available. So this is worthy of a good research, can be used. And we have some of the common protocol samples also stored at the NERT biorepository, as well as the individual parent protocol samples at each of the sites for both TB patients and their household contacts. One of the studies that we did of the samples that we collected, transcriptomics, we tried to see this different various signatures, multiple transcriptomic signatures have been identified across the world. And we find that most of the signatures are actually able to differentiate between the TB patients and their household contacts in our population as well. So whatever is transcriptomic signatures in other parts of the country, world do work in our setting also. And based on that, we have actually developed TB signature profiler. This is the bioinformatician, Dr. Ivan Johnson from Rutgers. He is leading this, and we have been able to identify a 29 gene signature called Predict 29, which is going to be validated in our phase 2 of Report India studies. We have the diagnostic cohort trying to test newer diagnostic tools. I'll just finish in 1 more minute. Not going into the aim of the report phase 1 diagnostic cohort. These are some of the studies that we have proposed. M6 aptamer, Dr. Tarun is going to speak about, nanostring, plasma cytokines, urine lamp, but all these are samples now stored, and we have much more samples which can be used. And these are the number of participants we propose to enroll. We have already covered 25% of this. And we are also proposing to use a 3-gene Sweeney's 3-gene signature as a host response cartridge, which is being manufactured by Cepheid, and we will be using that very soon. These are the our events that we do in our cohort of diagnostic cohort in presumptive TB patients. And these are some of the projects in addition to the report phase 1 and phase 2 that we do in JIPMER. Not going into these details, being an epidemiologist, most of them will be more of epidemiology than basic science. Some of the research questions that Gerhard Walz's group identify, which need to be prioritized for TB diagnostic tests. And I leave with this. Thanks to everybody who have helped us in doing all that we have done till now. Thank you.
Thank you very much, Dr. Sarkar, for giving us an excellent overview of the traditional as well as the new upcoming technologies that are available. I invite Dr. Tarun Sharma to give the next talk. He's going to be speaking on harnessing aptamer technology for the detection of TB. Dr. Tarun Sharma is an associate professor at the Gujarat Biotechnology University, where he is heading the Department of Biotechnology, Medical Biotechnology at Gandhinagar, Gujarat. And Dr. Sharma has also earlier worked as Innovation Awardee at the THSTI, so he is no stranger to this institution. What he's particularly interested in is in how to work in a multidisciplinary way to address biologically pertinent questions, and his field of interest and strength is aptamers. So, Dr. Sharma, you please may speak on your experiences in developing aptamers for the detection of TB.
Thank you very much, ma'am, for kind introduction. And it's like, I'm feeling like Ghar Wapsi because I spent almost a decade here. I joined as an innovation awardee, and then I served this institute as a senior scientist, and then I moved to Gandhinagar. And I would like to thank THSTI team also for inviting me for this talk. So I'll since it's a mixed audience, there are many students also. So I'll about aptamer technology as well. So you can see here, this is image of Lord Shiva, which is depicting the Tandava. Tandava is a celestial dance. So if you see this image from the design perspective or shape perspective, each image is depicting a different shape. And you know that DNA, the basic function of DNA is to pass genetic information from one generation to the next. But when it folds into a particular shape, it can perform a different function. So you can see here, these are the non-stereotypical function of DNA, and we call it functional nucleic acids. It can form stem loop-like structure, it can form hairpin, it can form pseudoknot, it can form G-cardoplex structure also. So what exactly aptamers are? So aptamers are synthetic single-stranded DNA and RNA molecule that we generate through a process of in vitro evolution, which is like Darwinian evolution. And the good thing is it can be developed against any target, doesn't matter whether it is immunogenic or non-immunogenic. You can generate aptamer against a small molecule. You can generate aptamer against large cell, protein, antigen, or any other target. So these are some of the successful example of aptamer-based test. One is for the aflatoxin. Then you have one test for the foodborne pathogen detection. Then this is aptamer-based drug, which is Macugen, used for age-related, wet age-related macular degeneration. So it can be used as a diagnostic tool as well as a therapeutic agents, and it can be used for bioimaging agent as well. So aptamer are considered as a chemical rival of antibody. So whatever antibody can do, aptamer can also do the same thing, and it has certain advantage. The first thing is, being a nucleic acid, it is of a small size in comparison to antibody. And because it is nucleic acid, you can store it at room temperature, while for protein or antibody, you need cold chain. And you can easily synthesize it like a primer once developed. So there is no batch to batch variation while you generate antibody in the animal, and you cannot control the physiological response of animal. So there is a batch to batch variation, and you can easily modify it with variety of moiety. You can take biotin, you can take fluorophore, you can take nanoparticle as well. So the cost wise also it is relatively cheap. So I mean, you already know that TB is one of the largest infectious disease killer, but for a student, I'll be briefly tell that broadly it is of two types. One is pulmonary TB, which affect lungs, and other is extrapulmonary TB, which can affect any part of our body except lung. It can affect your meninges or brain and central nervous system. We call it TB meningitis. It can affect your pleural cavity. We call it pleural TB. It can affect your abdominal. We call it abdominal TB, and it can form your eyes, bone, and other part of the body as well. So to put thing in perspective, I'll tell you a small story. So there was a guy close to my hometown. His name was Vinay. He was 8-year-old. He was perfectly fine, but after 6 months, he was presented with different symptoms, like he was having fever, his mental status was changed, he was having nausea and vomiting, and he was also having the headache. So his father took him to the doctor, and clinician prescribed multiple tests, which include your blood-based test, the biochemical test, microscopy-based test, culture, and the gene expert as well. All these tests turned initially negative, but the disease got aggravated, and finally he was diagnosed with a TB meningitis. And eventually that guy died. So he died not because we do not have the treatment, but the problem was the diagnostic. We were not having a good diagnostic test. So we thought that can we develop some sensitive diagnostic test? And so I'm not a TB guy. So I was mentored by Professor Jaya Tyagi, and madam is sitting here. So this is our team. Abhijit was a PhD student, and our clinical collaborator was Dr. Neera Sharma. So we first think that aptamer could be a good tool, and how we can develop aptamer. I'll give you a metaphor. Like you have a bunch of keys, which has many keys. From this bunch of key, you need to fish out 1 or 2 keys that can open the lock. So in our case, the bunch of key was a library, aptamer library that we have designed, and that this lock was the antigen, which is HSPX. So this HSPX is a heat shock protein of 16 kD, and there are several reports on the detection of HSPX antibody and HSPX RNA in the active TB patient. And a study from Professor Tyagi's lab has demonstrated that this particular antigen can be a potential biomarker, because they have shown the presence of this antigen in the CSF. So what we did, we first developed aptamer against this antigen using this process called CellX. And after iterative round of aptamer selection, we got few aptamer candidates. So you can see here, those which are in the red are giving very good binding in a assay called ELISA. Then we have evaluated their specificity. You can see here that some of the aptamers like S63 and H66, they are specific or selective rather in comparison to the antibody. Antibody was showing cross-reactivity to the other antigen also. So we picked this aptamer called H63, which is giving very good specificity. We predicted its structure. So it is forming 2 stem-like loop-like structures. So our aim was to determine which particular stem loop-like structure is actually responsible for the binding. So we truncated the aptamer. You can see here, this bigger stem loop-like structure is actually contributed for the binding, not the small one. Then we introduced several site-directed mutations, and variety of mutant variant were designed, and one variant, which we call M6, is giving binding better than the parent aptamer. So we took this particular aptamer and proceeded, and we did proceed, and then we developed an assay called ELISA. So it is aptamer link immobilized sorbent assay. So in principle, it is similar to antibody based assay. What we are doing here is we are taking patient sample, putting it on a 96 well plate. Then we have a aptamer, which is biotin label. And so biotin labeled aptamer will bind to the antigen. Then we are adding streptavidin HRP. So if aptamer is there, it will be probed by the streptavidin HRP. Then using the substrate, you can easily detect it. So first we have evaluated it on CSF because we want to detect TB meningitis, and we compare in all these cases, we have compared the performance of our test with the culture, gene expert, and qPCR based assay and microscopy as well. But in the interest of time, I'm not showing all the data. So in TB meningitis, we got close to 100% sensitivity and approximately 91% sensitivity. Similarly, in case of pulmonary TB also, our sensitivity and specificity was very good. In this case, sample was sputum and the sample size was more than 300. So precisely, I think it was 314 samples. And then in case of abdominal TB also, our sensitivity and specificity was good. And same is the case for pleural TB. But this test was taking close to 5 to 6 hours. So what we did is, we thought, can we develop a relatively rapid assay? So what we did is, we have labeled our aptamers with two things. On one end, thiol, on other end, a redox probe. So on a gold electrode, so this is screen printed electrode, and this is the prototype device. We have immobilized the aptamer on the electrode, and this redox level is here. So you see here that when there is no antigen, this redox level allow rapid electron transfer. But as soon as aptamer bind to its target antigen, there is a structural change, target induced structural change in the aptamer, which is placing this redox label apart from the electrode, which is causing reduction in the current. So you can see here, when there is no HSPX, the peak is very sharp and big. However, in presence of HSPX, this peak is decreased, and this decreases in the antigen concentration dependent manner. So through this assay also, we were able to detect both the extrapulmonary TB and pulmonary TB. But in case of pleural TB, the specificity was not so great, which need further optimization. And so this is how the entire TB diagnostic test evolved. So first test was developed using the PCR, which were taking around 2.5 hours, and the cost was close to 250 rupees, and sensitivity was less than 90%. Then there was an antibody-based assay, but that was showing batch-to-batch variations. Then we developed aptamer-based assay, and the cost is also reduced. Dr. Sonali was talking about the cost of this test is quite cheap, less than 100 rupees per test, with a good sensitivity. And then this electrochemical sensing based assay. So this test also around 30-minute test, just 2 microliter sample you need, and the cost is 300 per test as of now, but you scale up, if you scale up, the cost would reduce. So this is our test against the WHO target product profile. So for any new test for TB meningitis, the sensitivity should be greater than 80% and specificity should be close to 98%. And our test is meeting this criteria. And same is true for a triage test. The sensitivity for a new test should be greater than 90%, specificity should be close to 70%. In our case, both are greater than 90%. And in case of pleural TB also, the sensitivity is greater than 96% and the specificity is 95%. So our test, whatever data we have, although it need to be further validated on a large sample size with a different geographical variation, and we are now planning to have multicentric trial, and one site is JIPMER. And but as of now, if you see that it has potential to be translated into a product. So this is another applicability. So this work we did in collaboration with Dr. Kamal Kataria, who is in the surgery department and AIIMS. So I was having discussion with him, and he has suggested an interesting problem that because he treats both breast TB patient as well as the breast cancer patient. So there some presentations are or symptoms are overlapping, and the treatment regimen is different, because in case of breast cancer, you have to prescribe certain steroid to the patient. However, in case of breast TB, if you will give a steroid, the disease will further aggravate. So he is interested to have some test that can distinguish whether the patient have TB meningitis, or not TB meningitis, sorry, breast TB or the breast cancer. So we took certain sections. This is very preliminary data on very few samples, but it is certainly giving some hope that in case of negative sample, negative control, you see there is no staining. Here we are using MPD64 specific aptamer, while in case of tissue section, which is AFP positive, it is giving staining. And in case of those sample which are negative by the microscopy, but clinically suggestive of TB, in there also our aptamer is able to detect the presence of antigen. So these are some of the publication from this work. And in addition to this, just for your interest, I want to highlight two more papers, where we have shown the utility of aptamer both as a diagnostic tool as well as a therapeutic agent. So in these two papers, in one case, we develop aptamer against the GLUC-B or malate synthase, which has two roles. One is it behave like an enzyme, and when it is present on the surface, it behave like adenosine. So in other case, Hub-B also. So Hub-B also behave like adenosine. So the aptamer that we developed against malate synthase and Hub-B, they can actually bind to the surface associated protein and can block the bacterial entry inside the cell. So in conclusion, that nucleic acid aptamer can be generated against various antigens of bacterial pathogen, not limited to TB, and they can be used as a diagnostic tool as well as an inhibitor, and it can be a useful tool in clinicians' toolkit to diagnose TB accurately. So that's it. I would like to thank my mentors, Professor Jaya Tyagi and TH Style leadership, who has actually supported me throughout my tenure at THSTI. So Professor GB Nair, Dr. Kang, Dr. Garg, and Dr. Sanjini Bhatnagar. And these are all collaborators, Dr. Mainidu and Dr. Tane and Dr. Bhalla from NITRD, Dr. Neera Sharma from Ram Manohar Lohia Hospital, Dr. Sagarika from PGI, and Dr. Rajni Yadav and Dr. Kamal Kataria from AIMS. And these are the students who actually did the bench work. Abhijit and Abhijit did this ELISA-based assay. Ritu did electrochemical sensing work, and Surbi did this pulmonary TB work, and Pooja did the extra pulmonary TB work, pleural TB and ascitic TB. And Archit also contributed to this work, and Anjali did this breast TB and breast cancer work. And I would like to thank funding agencies also. Thank you.
Thank you, Dr. Tarun. Please join us here. We have a talk by Dr. Gulshan Wadhwa. You will notice that his name is not in the scheduled list, but he is going to speak to us in this diagnostic session. Dr. Wadhwa is a scientist F from the Department of Biotechnology, Government of India. And he has 25 years of experience in government. Prior to that, 5 years in industry. So he is actually a program officer and member secretary of many committees of DBT, and was also involved in establishing the Biotechnology Information Systems Network, BTIS Net, which is existing in around 170 bioinformatics centers at present. So I invite Dr. Gulshan Wadhwa, please, to give his talk.
Down arrow to move slides. So thank you, Professor Soumya Swaminathan. We have been hearing about you for quite some time, and maybe had few interactions before. But yesterday when I heard you, it was really worth the knowledge. Anyway, why and how I'm connected to TB is that in our BTS network, we supported a program on
national TB database, and that program ran for almost 8 to 10 years. Involved major institutions around the country which are dealing with TB, including JALMA, where Dr. Katoch were there, NTI, and 6 other institutions. So that somehow it touched my heart, the way it is working, the way strategy they are taking. I think we should take that kind of strategy for almost all diseases which are coming again and again, and which are hard to treat and things like that. So today I'll be discussing mainly the policy portion of this. TB, as you all know, is a disease of ancient times, known by many names: white plague, King's Evil, Tabes, and so on. So until the discovery of antibiotics, the treatment was limited only to the warmth, rest, and good food. Then we had a few facilities which were developed. Robert Koch discovered MTB microbe, and in India also we was set up the first sanatorium in 1906 in near Ajmer in Tulania. And then, of course, the discovery of the BCG vaccine, most of us know it, and that is the only vaccine till today which is being utilized. And we are in the process of developing many more vaccines, but of course, none has come to that level. So discovered by Albert Calmette and Jean-Marie Calmette-Guerin. So it is called Bacillus Calmette-Guerin, BCG, as we all know, in 1921. Antibiotics were a major breakthrough for the TB treatment, and as it has been being told since yesterday, these 4 antibiotics are being used throughout for the treatment in the first phase, and another 3 out of these are being used in the second phase. This is the trend in the past, in the last 150 years, of how TB has, the incidence of TB has come down with the new practices and new tools and technologies coming in, new treatment methods coming in. So then in 1993, WHO declared TB as a global emergency and launched the directly observed treatment, so-called short course strategy. And all governments became active with that, bringing this into the policy level and giving a commitment that yes, compulsory medication, free medication, medication to all, and things like that will be given to all the patients. And that scheme we can see all around the country, and parts of the world is being implemented very strictly. Then in 2006, WHO launched the Stop TB strategy. With a vision of TB-free world, with a goal to dramatically reduce the global burden of TB by 2015 in line with the Millennium Development Goals, where the target of the Millennium Development Goal 6, target 8 was to halt and begin the reverse of incidence of TB by 2015, and to reduce the prevalence deaths due to TB by almost 50% compared with the baseline of 1990. So by 2050, they estimate that TB to eliminate TB as a public health problem. So that is the background with which I will start, and I'll be discussing some of these things. The components of the Stop TB strategy were to pursue high quality DOTS expansion and enhancement program, address the TB and HIV together. So because HIV patients and TB where they are together, it leading to more number of deaths, and multi-drug resistance TB and other challenges similar to that. Contribute to the health system strengthening, engage all care providers around the country, and empower people with TB and communities to be engaged, enable and promote the research. I think in the last 2 aims in this, objectives in this, we are all major part of this. And of course, doctors are part of the healthcare providing system also. So the Millennium Development Goal for tuberculosis up to 2015 is over, and we have achieved 41% decline in the prevalence in 2014 since 1990, and a 47% drop in the mortality rate. In the process, we have saved around 43 million lives between 2000 and 2014. I'll be talking mainly about the pre-COVID era because the data, stable data is only available for the pre-COVID era, whereas all developments data is still being analyzed at different levels, and it may be coming, some data is coming 2021, some data is coming to 2022, but the data is still not stabilized to that level, as I heard from the Ministry of Health and Family Welfare. So, but the people are talking and developments are taking place at a much faster pace than it was in the past. So we will be able to achieve the goals set up as a part of the Millennium Development Goals by WHO and the individual countries. TB situation in India with respect to COVID-19, in the last 2-3 years of the COVID, patients have been found to be 0.37 to 4.47% in different studies, which are having prevalence of TB. Overall decline in TB notification by 26% during January to June 2020. It has been notified at different levels. So the major challenges what we are facing is lack of awareness and resources, poor infrastructure. Of course, these are improving, increasing drug resistance cases, poor notifications and negligence at the part of patients because of the long treatment periods and other things related to that, non-availability of the resources and funds. So if we eradicate poverty and undernourishment on the other side, we educate the people, the masses, and eliminate the stigma attached to the TB, maybe we can hope for a disease-free future very soon. The latest TB treatment guidelines in India involve the treatment initial phase 2 consisting of 2 months of these 4 drugs: isoniazid, rifampicin, pyrazinamide, and ethambutol. And the continuation of the phase should consist of another 3 drugs given for at least 4 months, which can be extended depending on the condition of the patient. I have listed some of the standards for TB care in India, which has been given by the different departments, different involved stakeholders. And this includes testing and screening for pulmonary TB, diagnostic technologies, test for extra pulmonary TBs, diagnosis of HIV co-infection, and so on. You can see there, it involves even the society, it involves even the NGOs, it involves even the government and the public healthcare systems, private healthcare system, and all the resources in the country to put the system in place and take care of the TB, because TB is such a disease which takes a long time for its treatment, and patients can find it very difficult to cope up with the whole system. So TB incidence per 1 lakh population have been told by my earlier speakers also somewhere. The end-TB strategy set up by WHO defines milestone for 2020 to 2025, and the targets for 2030 and 2035 deaths. The targets set for 2030 are 90% reduction in the number of TB deaths and an 80% reduction in the TB incidence rate. New cases of incidence rates of the new cases per lakh population per year compared with the levels of 2015. The milestone for 2020 are 30% and 35% reduction in the number of TB deaths and 20% reduction in the TB incidence rate. Although very high targets, but yes, with the kind of system governments and WHO have recommended and accepted and adopted, we are able to achieve to a greater extent. Strategy also includes a 2020 milestone that no TB patient and their household faced catastrophic cost as a result of TB disease. So government is providing free facilities, free medicine and related things to all the patients, even the MDR patients. So TB incidence per lakh population for all the 6 continents here given, where you can see Africa and the Asia, Southeast Asia is coming a maximum of around 286, 282 to somewhere between 282 to 300. You can see it here. And India is coming in the middle of the whole country-wise list. So we are at a better platform and we can achieve much better in this case as per the planned objectives. So global commitment to end TV, moving from halting TV earlier to now ending TV by 2030 or 2035. Strategic development goal target 3.3 by 2030 to end the TV endemic. And some of the aims objectives are mentioned here, methods of their achievements. So barriers to achieving the targets here apparently are a weak health system. We have to strengthen the health system. We have realized this in the COVID period, and underlying determinants such as poverty, undernutrition, migration, risk factors such as smoking, silicosis, and so on. A lack of effective tools, which we are strengthening with time. Continuous unmet funding needs, of course, are always there.
Can you wind up in a minute or two, Dr. Wadhwa, please?
Sure. So the NTV strategy is just repeated in terms of numbers here. And the pillars and principles are here. Integrated patient-centered TB care and prevention. The second pillar is the bold policies and supportive system, intensified research and innovation. In the next few slides, I'll be discussing only these. So I will just be showing slides as such and will not be speaking much. In this, the government stewardship and accountability with monitoring and evaluation is there. Strong coalition and civil society organization and communities will be, are being involved. Protection and promotion of human rights, ethics and equity, and also adoption of strategy and targets at the country level with global collaboration, including WHO. So this is how it decreased, proposed to decrease over a period of time. These are the components of the integrated patient-centered TB care and prevention system: early diagnosis, treatment, preventive treatment of all patients, and collaboration TB and HIV. So these are basically discussed slide by slide because I've been told to wind up, so I'll be moving faster. This is how TB and HIV trends over the past 15 years or so. The preventive treatment of persons at high risk. We go to the second pillar, the bold policies and supportive system, which involve political commitment and adequate resources for TB, engagement of communities, civil societies, then social protection, poverty alleviation, and actions on the other determinants, universal health coverage policy and regulatory framework, especially the case notification. So all these things are being discussed here and the figures shown to develop the ambitious national strategic plan, mobilize adequate resources for the mitigation of TB, engagement of communities. These are some case studies you must have heard about. The policies and all have been strengthened in the past few years, and TB notified cases have been decreasing over the years.
Dr. Wadhwa, could we
1 or 2
please
minutes more, please.
close?
So this is how the
Because
global
you're running into others' times.
in India, social determinants of TB, and over the world, how much population is facing catastrophic health expenditures up to 2017, and same can be projected further. Social protection and poverty elimination. The third pillar, the key is a discovery, development, and rapid
Dr.
uptake.
Wadhwa, you really
It's through.
must have to stop now.
These are the diagnostic.
We have 2 more talks.
1 minute, please. TB diagnostic being developed. The last test recommended by the WHO, clinical developments of the TB drugs, global development of pipeline for TB vaccines, and the upcoming vaccines, MTB-Vac and UT, and vaccine, research optimizations, and this gap of around 3.6 to 4 million around the world. Achievements are 54 million lives have been saved between 2000 to 2017, and 33% drop in TB deaths between the same period.
You stopped your slides, Dr.
Last
Wadhwa.
slide. MTR TB crisis and funding gap, which have to be taken care of. Thank you very much.
Also, Dr. Wadhwa. Dr. Wadhwa, I'm here.
Yeah,
The
please.
distinguished scientist next to me is Professor Jaya Tyagi, not Soumya Swaminathan.
Okay, I'm sorry.
All right. So we can move on to the next speaker, I think, immediately. Dr. Avinash Bajaj. Why? Dr. Avinash Bajaj is a professor at the Regional Center for Biotechnology in Faridabad. And his group is interested in engineering advanced healthcare materials for biomedical applications, which include antimicrobial therapy, gene therapy, cancer immunotherapy, etc. He's made very interesting findings in bile acid-derived amphiphils, and which can target specific microorganisms for early detection. His group has been actively engaged in engineering lipid-based nanomicelles and hydrogel implants that can maintain sustained release of combination of chemotherapeutics with the potential, again, for antimicrobial, anti-inflammatory, and anti-cancer therapeutic applications. He's going to talk to us today on membrane-specific probes that can detect mycobacteria in human gastrointestinal tissues. Dr. Bajaj.
Okay, good afternoon, everyone. And first of all, I would like to thank Raman, Krish, Amit, and Ganesh for their kind invitation. So today I will be talking, so before going to my talk, so this is not the title of my talk. So what we do in our lab is, I am basically trained as a chemist, and we engineer different kind of advanced healthcare materials for different biomedical applications, including antimicrobial therapy, gene therapy, anti-inflammatory therapy. Although one of the key area of interest is cancer immunochemotherapy. And other area what we are working on in the sphingolipid biology and therapy, where we are trying to find the new targets in the sphingolipid metabolic pathway and to find new diagnostic and prognostic markers, different diseases like IBD, liver, and cancer. So as the key, we start as from the chemistry point of view, we engineer different biomaterials. So today I will be talking about one aspect, which I learned from my TB friends. I'm not a mycobacteriologist specialist, so it is all started, this work started with the initial collaboration with Raman, and then it started with Dr. Vinay, and then we started collaboration with clinicians, including Dr. Vineet from New Delhi. So I think I will be talking about the molecular probes for the detection and differentiation of gastrointestinal TB, and I need not to introduce you people about the TB. The key area that we know now from the earlier talks that 20% of the TB cases are extrapulmonary TB, right? And one of the TB is gastrointestinal TB, which affects any of our organs of the gastrointestinal tract. So I was basically introduced to this area where Vinay called me in one of the meetings where all the TB people were there from Delhi, and where I came to know that, okay, there is a gastrointestinal TB, and the challenge of detection in gastrointestinal TB is, as we know, there are few bacteria available, there are no specific antibodies, PCR is not a good test, as well as the key challenge, what I found, what is that there is a need to find the ways where we can differentiate between the gastrointestinal TB and the Crohn's disease. So keeping these challenges in my mind after I came from that meeting, then we thought, okay, these are the questions what we found that can we detect and differentiate the mycobacteria? First question is that, can we do that? And can we differentiate between gastrointestinal TB and the Crohn's disease in the patient samples? And can now be used the mycobacterial membranes as a target? I have been trained in IISc on the lipids. So I love lipids. So we thought that, okay, can these mycobacterial membranes be used as a target? So as we all know that if you see the mammalian cell membrane, which has phospholipids and cholesterol, right? Whereas you go to the gram-negative bacteria, everybody knows the LPS is a very good target. Go to gram-positive bacteria, you have the peptidoglycan, and again the phospholipids, which are very common, although there is some difference among the mammalian cells and the bacterial cells. And now you go to the mycobacteria, right? If you see the mycobacterial membrane, everybody knows that this is waxy, highly hydrophobic in nature, right? And it's very rich in mycolic acids. So very difficult to treat these bacteria because of this membrane. So we want to see that, can we make a probe which targets the mycobacteria? And know that peptides, which are the part of the innate immune system. Now, if you see any of these peptides, either they are in alpha helix or in the beta sheet, and if you see their conformation, they are like this. On the one side, you will see the charged amino acids, and on the other side, you will see the hydrophobic amino acids. And these antimicrobial peptides, basically, they interact with the bacterial membrane and disrupt that membrane. And that's how they play a part of the innate immune system. So now I have been working on this scaffold on the bile acids Now, if for the last 15 years, we have made around 400, 500 molecules to test for the different applications. But if you see this molecule very carefully, this is like the amphiphilic character. On this side, you see there are 3 hydroxyl groups which are polar in nature, and on the side you have the hydrophobic methyls, which can like a mimic of the antimicrobial peptides. So what we have been doing is that we have been derivatizing these 3 hydroxyl groups with different amino acid, different other groups, and as well as this carboxylic acid chains. We are in depth doing the very interesting physiochemical characterization of these molecules, their interaction with the different bacterial membranes, and as well as both as at the computational way as well as the biochemical ways. And we found that, okay, we can make a specific molecules which can target the gram-negative bacteria by using LPSA target. We have a molecule which targets the mycobacteria, which I will show you. And now we have a molecule which targets the Candida, and it targets is the lipid metabolism of the Candida. So that can I tell us. And the recent study which we published in Journal of Medicine Chemistry, where we saw that one of these molecules target the 2-component system also, and it inhibits the bacteria to develop the vancomycin resistance. So keeping this idea in mind, among all the molecules which were screened initially with Raman, then we come to these 4 molecules, right? So if somebody asks how you come, it is from the screen of around 300 molecules. We came to these 4 molecules. We attached a fluorophore here, very simple fluorophore. This can be further modified. And we use the 4 different groups. This is the ammonia, this is trimethylamine, this is a, you can say pyridine, and this is a dimethyl aminopyridine. So this is a, you can say electrostatic interactions, whereas these molecules can show the hydrophobic as well as the electrostatic interactions. So first very simple experiment, this was all the probes was properly characterized, so we will not go into the detail. So first experiment which we will do is a simple with the Ambovis, and we titrated all the 4 different probes, and we see a different concentration. Now you see this was done by flow cytometry. Now you see at 1 micromolar concentration, although it's a very high concentration, I will come to the nanomolar concentration also. So 1 micromolar concentration, you see more than 90% of the bacteria was stained. And this was only stained with the M4. Later it will be P4 probe, it is the same molecule. So 1 micromolar, you can see more than 90% of the bacteria was stained. Right. Now we want to see whether this is really targeting the bacterial mycobacterial membranes or not. So what we did with these simple SPR studies, we isolated the mycobacterial lipid, put on the SPR chip, put all the 4 molecules, titrated that, and you can see that here from this graph that P4 molecules, right, is specifically binding with the mycobacteria membranes, which lipids what we have isolated. So then to see that whether this molecule now P4 we are focusing is only binding with the mycobacteria, not with other bacteria, we screen by flow cytometry with all the other bacteria of gram-negative, gram-positive, see that only the mycobacteria which is specifically binding with this probe. And then we go to the M tuberculosis, which was done by Dr. Vinay's student Mehak, and she also observed that nearly more than 90% with a good mean fluorescence intensity, both at 1 micromolar and 2 micromolar, you can see that the probe is able to stain this mycobacteria. So then we came to, because full flow cytometry is not the ideal, because we have to work on the human tissue biopsy samples. So we went into the fluorescence confocal microscopy. We took the different bacteria. You can see the 3 different bacteria, which are 2 of them are M. cherry, 1 is not. And we see that this is a green fluorescence probe, which we have a P4 probe, and you can see that all the mycobacteria have been stained with the fluorescence probe. And this was done at 20 nanomolar concentrations, and whereas none of the gram-negative and gram-positive bacteria was able, these probes were not able to stain. So then to see, we have to go into the mechanism studies and to see how we engineer also the biotin-based probes. So we remove the fluorescence and we use the biotin so that we can explore the step-3 biotin chemistry easily. So we did the electron microscopy and the super resolution microscopy where Sheetal helped us, and we can see that here the molecule is binding at, is residing mostly at the membrane. So now to see that, every who works on the mycobacteria will always ask me that what are the lipids where it's binding. So this was the question which a reviewer has also asked. So what we did, we ordered all the lipids, we got all the lipids, where firstly we tested against the mycobacterial lipids. So what we use is the biotin-based probe. We used the technique called biolayer interferometry, where we took the probe with the biotin labeled biotin conjugate and screen against the lipids. Firstly, you see that total mycobacterial lipid is the P4 biotin, which is binding, whereas none of other bacteria, other lipids you see the probes are not binding. Only these are the two lipids which we could find is that trihylose monomycolate and PM6-phosphatryl inositol monoxide. So these are the two target lipids where this probe is binding, which is specific to the mycobacteria. So now, this was the in vitro experiments. Now we have to see that whether, because when you have in the biopsies, they're very heterogeneous in nature, polymicrobials, and as Ashwini has been working on the biofilms, what happened to biofilms, whether this molecule can go intracellular in the mammalian cell, whether it's toxic, can it stay in the intercellular bacteria? So these were the few questions that we ask, and to probe these things, firstly we did is that we mix one mycobacteria in with 10,000 other bacteria to see the sensitivity, and we can see that it can detect even the one mycobacteria in 10,000 other bacteria. We went ahead and did the biofilms. You can see it can stay in the biofilms. So extracellular matrix is not a problem for this. And even in the polymicrobial biofilms, where we use two different bacteria, mycobacteria and other bacteria, you can see that here that this probe very specifically only stains the mycobacteria even in the polymicrobial biofilms. Then intracellular bacteria also, it does not stain any of the other bacteria like E. coli. So then this was the mice infections. So we have used the mice lung infector models, which was done by Mehak. So we see that we use a positive control antibody, which can stain the mycobacteria. And you can see that wherever there is a good co-localization with the antibody and with the probe, so that you can see the mycobacteria even in the infected lung tissues. Whereas on the drug treatment, you see that number of the mycobacteria in the lungs have reduced. So it can also be used as a quantitative method to see the effect of the different drugs when we use the preclinical studies. Then we collaborated with Dr. Vineet Ahuja, he's a gastroenterologist, and Dr. Prasenjit Das, he's a pathologist at All India Institute of Medical Science in New Delhi. So we took, I'm just showing very representative figures from different cecum tuberculosis, then gallbladder and lymph node tuberculosis. And we use the antibody as a positive control, and you can see that there is a clear localization of the antibody as well as the probe in the mycobacteria in all the tissues. So after this good validation, we collected more samples, both for the gastrointestinal TB patient as well as the Crohn's disease patient. And this was around 36 samples we screened, although there are 4 samples where the probe didn't work. So I'm showing some of the data here. You can see that among different kind of the gastrointestinal TB patient sample, we see that wherever the antibody is binding, your probe is also binding, and it is able to differentiate between the gastrointestinal TB and the Crohn's disease. So we are now planning to do a big cohort along with Dr. Vineet. So to see the different other bio applications, can we work around the chemistry? So we use this same the biotin-based probe now, right? And we can see that you can use the streptavidin quantum dots. You can see streptavidin fluorophore conjugates, and you can detect the bacteria along with this. So you need not to have only the fluorophore, which has its own disadvantage as well. And then we use the sandwich assay, where we took the biotin probe on the streptavidin glass plates, put the bacteria, then put again our probe, and you can see that we can entrap these bacteria in the sandwich assay and detect that. So the future plan, what we are thinking of doing is that can we make a MRI-based probe for the detection of the TB in the mice samples, and then for later for a clinical application. Although we are finding some challenges how we will use this in the mouse models, because we don't have any imaging systems in the BSL-3. So these are the challenges which are stopping us to do this thing. So this study was just last year published in Advanced Healthcare Materials. So what we have shown is that there is a folic acid-based molecular probe which can specifically and selectively bind to the mycobacteria. It can detect the intracellular mycobacteria. It can detect the mycobacteria in polymicrobial cultures as well as the biofilms, and it can differentiate between the gastrointestinal TB as well as the Crohn's disease. So I'll take 2-3 minutes. I have the time, I think. So just come back to the other discussion which one of the students yesterday asked about the drug delivery system, the oral delivery systems, right? Oral delivery systems, everybody seeing is that a more effective way of the system. But you see, if most of the patients could not compile with the whole 6-month plan. So what is the need is that can we make a sustained drug delivery systems which can deliver the combination of the drugs, like 4 drugs, 6 drugs for a long time. So you need not to take the medicines every day, which is a challenge. bench, right? So I will introduce the topic of hydrogels, which are the self-assembly of small molecules or the polymers. So these are a supramolecular self-assembly which can entrap a lot of drugs. And most of the reports, if you see that, you will see on the polymer systems. And there is a challenge basically with the low molecular weight hydrogel systems, although they're easy to synthesize, but they cannot entrap the combination of the different drugs because they will disintegrate. So our group has been working from last many years to make the different small molecule hydrogel systems which can entrap the combination of drugs and can release the drug in a sustained manner. So what we have done, as we know, the bile acids in our body will self-assemble and help in the fat metabolism, right? So they can self-assemble to make the nanomicelles. So what we have been working to explore these bile acids to make the nanomicelles or the hydrogel implants. So one of the molecule, I will not go into the detail of the screening. I am just showing the one lead molecule, which we call it LCGG. If you see this molecule, this is the bile acids, blue backbone. We have attached only a dipeptide here and the benzyl group here. And this molecule self-assemble in water, and at 750 to 70 mg, it will make a hydrogel implant. And you can see if you put a fluorescence fluorophore dye in this, and this upper one, if you see, this is called NIR gel, you will see here the slow release for till 20 days, only where the gel is injected. Slowly this drug will getting diffused throughout the body. And it will also release the drug till 50 days in rats. So 20 days in mice and 50 days rats. And if you make this kind of system, we explore this system for the cancer therapy. Then what we thought is that, what about if we put all the 4 drugs, which is used as the first line chemotherapy drug for TB, and we inject this gel, because this gel stays for 3 weeks. So if we put this gel for 3 weeks and compare this with the oral delivery system where you have to give the mice every day oral drugs, what will happen? Whether a TB gel is effective than the oral system or not. So what we did, we took all these drugs. Now, if you see these 4 drugs, these 3 drugs are highly soluble in water, right? But they have the low permeability. Whereas the rifampicin, which is a more hydrophobic drug, but it has a low solubility as well as a low permeability because of this chemical characters. So, but in the hydrogel system, if you see that all the 4 drugs this hydrogel can entrap. After that, we synthesize many hydrogel, but we could not get any other gel which can entrap these 4 drugs. So this is the specificity of this gel. We characterize this gel in detail. I'm not showing that data here, but I will go to the animal data, which is very interesting. Now we see that this is the untreated mice. Then we took only gel. Only gel is not effective in clearing the bacterial burden. Now, if you see, this is the oral as well as the TB gel, right? And that was X by 2 dose as well as oral and TB gel by X dose. Now, if you see that TB gel at X by 2 dose is as effective as the oral dose of the X, right? So TB gel X by 2 is equally effective as the oral of the X. So you can you can reduce the dose to half and it makes that equally effective. And if you compare the TB gel at the X dose, it is more effective than the oral at the X dose. So this clearly shows that these kind of systems, although this is not a perfect system, but these kind of the systems can be explored for the sustained release of the drugs. And now the people are working even the remote control based the drug release in your body. Whenever you want to release the drug, you can control remote from outside your body. So it is a wireless based system where you can control the drugs, and which is very effective for this kind of treatments. So now what we have done, we have made another gel where you can see here, when we injected this gel with NIR probe, you see 365 days. This gel can stay in the mice for 365 days and it can slowly release the drug. You will not see it here, but if you do the mass spec or HPLC, you will be able to detect this dye in the plasma. So with that, I would like to thank my student Siddhi, who was a postdoctoral fellow in my lab, who did all the work related to the mycobacteria. Deepak has synthesized our molecules. He is now doing PhD in UK. Mehak helped in all the experiments related to mycobacterial tuberculosis. And this was not possible without Vinay's help. Whenever I went to him with any crazy idea, he always said, yes, let's do this. And Vineet helped in all the clinical and understanding this whole problem to us. Prasenjita helped in the pathology. Sheetal helped us in the confocal super resolution microscopy, and Archana helped in the electron microscopy. And Chayam, we did all the characterization of the probe. He's a physical chemist. So this grant was supported by SCRB and DBT and RCB for the intramural funding. Thank you so much.
Thank you very much, Dr. Bajaj. Thank you. If you'd like to come and sit here. I think we'll move on to the next speaker since we're running a little late. The next speaker is Dr. Ranjan Nanda. He is a group leader at THSTI and the Center for Genetic Engineering and Biotechnology here in New Delhi. The primary focus of his group is to better understand host pathogen interactions in TB, with a major focus on clinical patient-based studies for hypothesis building, and also to employ in vitro and in vivo models for mechanistic validation. He's been innovative in trying to identify diagnosis methodologies for tuberculosis, as well as other clinical research, and has received a lot of competitive funding support from both national and international agencies. Overall, the group's aim is to develop translatable solutions by detailed understanding of TB disease pathology. And we look forward to his talk on pharmacogenomics in TB, a neglected aspect.
Thank you, Professor, for the kind introduction and the THSTI team for inviting us. And it's a privilege to be here in this August gathering to present some of our activities. So this pharmacogenomics project is pretty new, for us, and last 6-7 years we have worked on it. So I'll not talk too much about the TB as a global threat. We all know maybe from yesterday afternoon, we are keep on listening like how many people we are losing across the globe and in our own country. I'll just focus a little bit on the different risk factors. We know that HIV infection creates high risk. Diabetes causes diabetic patients are 3 times have higher risk of developing TB. Iron overload, on which our team works, but today I'll not talk much about it. And then we all know whether it is malnutrition or immunosenescence, that also increases TB susceptibility. The most critical one we may not have discussed till now is the host genetics. We are very careful about the pathogen genetic diversity, whether it is susceptible to the existing TB drugs or it has resistance. But what about the host genetics? It also might be playing some critical role. And there is nice reviews where people say that, oh, the existing TB drugs clears the pathogen in 97% of the cases or 95% of the cases. Only 5% cases, because of the host genetics, it may not be working or it may not be giving the desired results. I think I don't agree with it. And I'll explain like why we don't agree with it. So, so what happens like a TB patients, if somebody has the symptoms of 2 weeks of cough, fever, weight loss, night sweat, hemoptysis, they come to the clinic and the doctor recommends for a diagnosis. These are all the most common diagnostic methods like sputum microscopy. Although it has a 60% sensitivity, still we use it because that's most efficient, less financially challenging, and the turnaround time is 48 hours. So we get the test, and still we are using it. We talk a lot about nucleic acid amplification test results like GeneXpert, and GeneXpert gives results in less than 2 hours, easy. But Dr. Sarkar has already explained that it needs air conditioning room and training, and then continuous electricity and all. And these are all challenges, but it has given quick results with 2 hours of turnaround times. Culture taste, yeah, of course, it's the best, but best comes with a price and lots of other limitations. So many of the times we don't get access to the culture taste. And based on the taste results, we determine whether the subject is having TB or not. And then if the person has TB, then the clinicians recommend a lengthy treatment of like 2 months of intensive therapy, which is like 4 drugs, rifampicin, isoniazid, pyrazinamide, and ethambutol, and then 4 months of continuation phase of treatment with rifampicin and isoniazid. And if somebody is, if the TB patient is infected with a drug-resistant TB strain, then we of course have the second-line drugs. So what happens? Like once the clinician has recommended for the treatment, the person takes the TB drugs, and who has decided this drug doses and durations? It's like, okay, so somebody has decided, and it may have some effective results. So after taking the TB drugs, it is distributed. It's get observed in the intestine, and then gets, before it gets distributed, it may also modify in the liver. And there are certain transformation happens in the gut at the absorption site or gets modified in the liver. And then whatever is unutilized or modified, it goes, it is excreted from the system. Today morning, just after waking up, I saw LinkedIn, there was a nice creative animation made by Yoda, I think Amit's student. It's a a brilliant piece of creativity. And in the what I found is the message is let the patient to complete the treatment. But is it like the patient doesn't know that he has to complete, he or she has to complete the treatment? Of course, yes. But there is some issue with the TB drugs. Majority of the drugs which the clinicians recommend, we do expect as a patient that it should do the wonder and it should clear up the disease condition, but which is not the case. The most commonly used hypertensive drugs don't work in 30% of the patient population. Heart failure drugs doesn't work in 15 to 25% of the population. Similarly, antidepressants, cholesterol, and asthma. So if we expect that the drug is there to clear up the disease, yes, that's the primary job. But it may show certain population-specific differences. So this is a normal population distribution. So the drugs, many of the patients may respond, and they can go ahead with the traditional therapy. But there are certain certain patients, they may respond in the toxicity may increase, and those patients stop taking the treatment because they need a better life first, then completing the treatment. And there is another group of population which will not respond to the treatment irrespective of how much drug they take. So isoniazid, if a TB patient is infected with a pathogen, like with a drug susceptible strain, then it has to take 6 months of isoniazid treatment. So once we take the isoniazid, it gets converted in the intestine as well as in the liver. And there are different host enzymes are responsible for this conversion. And this convert the byproducts. Byproducts can be hepatotoxic, or it can also generate peripheral neuropathy. And that causes the patient not to continue with the treatment. And NAT2, CYP, GST, these are all the important enzymes which is responsible for its conversion. So we had a fantastic postdoc, Mrinal, long time back, maybe 8 or 9 years before. So he did a literature search, and what we could find is acetylation plays a critical role in isoniazid modification. And in 1980s or 90s, there was a group of researchers, they have showed that South Indians are slow acetylators. And 30 to 35% of the North Indians are slow acetylators. So what happened to the Northeast Indian population? There's not much studies has been done. So what we had an ongoing project. So what we decided is that let's take 20 TB patients and do a very simple study of the pharmacokinetic study, and to understand what is happening to the concentration of isoniazid drug and its byproducts in the excretory bifluid, that is in urine. So the symptomatic subjects, they provided the sputum sample, and in the sputum, if the bacteria is present or the genetic material of MTB is present, we group them as active TB patients, and we excluded the non-TB subjects, and these are all adult TB patient populations. And we are collaborating with, we did this work with collaborating with Agartala Medical College. So we had the access of these TB patients, and these are all actually, when we recruited, they are drug naive TB patients. They have never taken any TB drugs. So just after taking the first dose, we collected their conjugative urine samples. And in general, pharmacokinetic study is not done using urine sample. it's all done in serum sample, but as it is an academic exercise, and we find that it is easy to collect urine samples longitudinally. So at 2 hours, 6 hours, 12 hours, 24, 36, and 48 hours of taking the first dose of TB treatment, we collected their urine sample. We processed them and we quantified their TB drugs and their byproducts. Here I give the example of isoniazid. All these yellow highlighted molecules we could identify using a single GCMS run. As expected, the isoniazid and acetyl isoniazid and many of the byproducts, they reach a maxima at 6 hours. Interestingly, India is the only country, a couple of years before, used to recommend the TB patient 3-dose TB treatment. It's the only country. I don't know what is the logic and what we have achieved out of it. Just see based on the profile, it seems like by 12 hours, the drug is not there in the system. If the drug is not there, of course, it will not have any effect. And just by taking the ratio of acetyl isoniazid to isoniazid, we can say that majority of this Agartala populations are actually slow acetylators. If a patient is a slow acetylator, then that means the drug will stay for longer time in the circulation. And if it stays for longer time, it will have a hepatotoxic effect. And if hepatotoxicity causes, then of course it will create lots of other complications. And this was nothing new. Only thing is that the patient population was new. 1960s, Professor McQueen showed that majority of the Japanese populations are slow acetylators. This is the slide I borrowed from Binod. He has shown that how different genes are responsible for modifying different drugs that we take. So the question came like, okay, you are telling based on the metabolite analysis, they are slow acetylators, but they may not be the case. Have you checked their genetic differences and all? So then we recruited TB patients, similarly, from Agartala Government Medical College and Nagaland Hospital, using the simple inclusion criteria of they should be above 18 years old and from both the genders. And we excluded pregnant women and the persons taking immune suppressive drugs. And if they are HIV positive, we excluded them. And the total population, and this work was done by a brilliant graduate student, Faruqi from Bangladesh. So a total of 101 subjects we used. The Nagaland patient population is low, 17, but majority were from majority are from Tripura. So we took the whole blood, we extracted the genomic DNA, and then took the specific PCR primers to expand specific gene fragments, and then we did the Sanger sequencing to identify the allelic differences. So for NAT2 is responsible for the acetylation. So these are all the PCR primer sets and the PCR settings. And based on this reference SNPs, we can show that what is the percentage population are rapid acetylators and which are intermediate, and which population can be grouped as slow acetylators. So it seems like the pharmacokinetics data what we have generated, which showed that majority of Tripura population were slow acetylators. In the NAT2 genotypic distribution also, we found that actually the Agartala population, the Tripura population are slow acetylators. Whereas Nagaland, which is adjacent to Tripura, although the population size is too low, but it seems like majority of them are rapid acetylators. So if they are rapid acetylators, that means what? The drug will have a maxima within 2 hours, and then it will go out of the system. Whereas in the slow acetylation status, the drug will stay for longer time. So if you're giving 6 milligram per kg body weight of isoniazid to a patient who is a slow first acetylator, same thing can be achieved in a slow acetylator by just decreasing the drug dose to half. And that's what has been proven in different African and other populations. And that will help to decrease the accumulation of toxic products and all. So then we also profiled the CIP1A2 enzyme, which is also is used for the conversion of certain TB drugs. And then it seems like, so these are all the primer conditions as well as the primer details and the PCR parameters. And it seems like the variation of CYP1 is very similar. They don't vary too much between Tripura and Nagaland population. And then the last gene we targeted was GSTP1, glutathione transferase, which takes care of the lots of drug metabolism. So GSTP1 has a 290 base pair gene product, and these are all the primers what we have used and the PCR conditions. And based on this SNP variation, we can group them as slow or intermediate or rapid metabolizer. So based on GSTP genotypic differences, it seems like Tripura and Nagaland, they have similar genetic profile. So the majority of the changes what we saw, if the GST conjugation is not happening properly, then there will be accumulation of the acetyldiazine, ketene, acetylunium ions, which increases the hepatotoxicity. So to conclude this part of the story, it seems like the Tripura population are slow acetylators. And if they are slow acetylators, with decreasing the dose to half, we can achieve the same effect, antimicrobial effect, and without compromising the bacterial clearance. And whereas in Nagaland, they are rapid acetylators, and we need to decrease, we need to take care of their dose so that the patient will not develop hepatotoxicity and peripheral neurotoxicity. These are all baseline data, which needs study with by increasing the population size and all. And I think as a graduate thesis, this was a good story to generate the baseline data. And then we wanted to, like, from Tripura and Manipur, from Tripura and Manipur, Nagaland, we compared these results, and then we thought, like, let's adopt a completely different approach, take another unique population, that is Manipur, and then see what all is happening. And all this work, this part of the work is done by Dr. Reena from Manipur University, and Nilkant is the PhD scholar. So here we have taken, again, we followed the same procedures of recruiting the patients. We took the symptomatic subjects, and based on the sputum microscopy and gene expert test results, we grouped them as either active TB or non-TB. And we also took lots of healthy subjects from this population. So a total of 178 active TB patients were taken. These are all, again, with the same inclusion and exclusion criteria. They are all adult subjects. And then here we have taken, probed 4 different genes, that is NAT2, GST, PXR, ABCB1. Only thing, the difference is that here we have used TACMAN allelic discrimination probes to using real-time PCR to identify what are the genetic differences. Manipur, Nagaland, and Tripura, these are all very nearby states. But it's very interesting to see that more than 50% of the Manipur study populations were actually intermediate acetylators. So if they are intermediate acetylators, there is no need of giving the same dose which you are giving pan-India. We can achieve the same target by giving less drug dose. So many of the times we feel like we have talked about hepatotoxicity. Is it like there is some experiment we have done to show that really the TB drugs induces hepatotoxicity? So we have taken a patient population of 32 subjects, and we check their liver function test. And out of 10 molecules, 10 markers, it seems like 6 molecules shows deregulation. So after completion of 2 months of therapeutic intervention, because of the TB treatment, this patient shows hepatotoxicity. And this can be taken care by reducing the drug dose. So if you want to end TB, drugs are essential, vaccine is essential, but whatever the drugs we have, we need to precisely use it to treating the patients. And we need to do therapeutic drug monitoring, whether it is based on some small assay or through mass spec. That time will tell, but we need to do just like the way we do in cancer subjects. It's done. So can we improve the traditional symptomatic approach of TB treatment modality? It's only possible by personalized treatment, and that will give us better treatment outcome, and it will significantly reduce hepatotoxicity, and that will increase the adherence. Third is it might also reduce the relapse rate. And all this work is done by the graduate scholars, and I have acknowledged them. And my clinical collaborators, Dr. Khamo from Kohima, Dr. Anjan Das from Agartala, Dr. Rima from Imphal, and Dr. Lokendra from JNIMS. These are all my active clinical participants who has helped us to reach this stage, and nothing is possible without the funding support. So all these project activity is supported either by Department of Biotechnology or ICGP. And I thank them as well as for you to kindly bearing me for last 20 minutes. Thank you.
Thank you very much. Now, are there any burning questions? Because we'll cut into the poster sessions and the tea time. So if anyone has any really important questions for any of the speakers? No? Yes. In the front here.
My question is for Ranjan. Basically, have you checked that the acetylation in latent TB subjects, because for preventive isoniazid preventive therapy, how could it affect?
Latent TB, I don't know why we have to treat latent TB. That's my personal opinion. We can discuss about it. All these subjects, what we have never done their TST test or quantiferin, because both the tests may mean something, but I don't know how clinically important it is at this stage.
Moreover, the expression of these acetylation enzymes is MTB dependent, or there are basal level already high in these populations?
That's a nice question. If you remember the Rachels, whether it is Manipur population or Nagaland or Tripura population, always we have mixed the findings. So it's not biased only towards the TB patients.
Okay. Thanks.
Any other questions? Just two more there and then we'll close. Yes.
Okay. I have a question for Avinash and Rajan. Avinash, the slow release hydrogels, how do you control the dose? Yeah.
Two way is that one is the first of the hydrophobic drugs will go very slowly, right? And hydrophilic drug come very fast. So you can increase or decrease the gel strength. If you increase the gel strength, you can slow down the release, right? And it also depends on the dose. More dose of hydrophilic drug, if you put it, the release will be faster. So, and you can modify the drugs also to make them fast or slow.
So when you have them for, let's say, for a month together, if you want to release,
yes,
do you have to have them in a very concentrated form?
So right now what we are focusing on one gel strength where we optimize that the gel will remain 21 days. So drug release will happen 21 days. So we have not worked towards that, how to make it less or more. So we are focusing only now on the 21 days dose. And other gel, there we can play more because that is for 1 year.
So Ranjan, quick question. You spoke about INH. What about rifampicin, ethambutol, other?
That's a nice question. Maybe in the next TV meeting, we'll discuss about it. We have some results, but we are not.
What I mean is somebody who is fast acetylator for one drug can be slow for another.
Yeah, we'll discuss over tea or something.
Avinash, this question is for you. So where are you depositing the hydrogel in the mouth?
This was a subcutaneous injection.
Okay, and what are you monitoring? You're monitoring the gel itself, or you're monitoring a dye that has been impregnated into the gel?
So we have done both. Firstly, we put the dye and see how it is releasing. And then we took the gel at different times and see that it is degrading, because right now the reviewer asked that what are the metabolites, right? So we see that, okay, this is a bile acid, these are amino acids, And we did also the immune profiling to show that there is no immune reaction. So there is no immune reaction, and whatever the little bit neutrophils increase, that anyway go after 10 days.
So if I understand, for similar release kinetics to be achieved for all drugs.
No.
Oh, so you can.
No, it will be no similar release kinetics. So rifampicin, as I told, will be released very slowly. Like other 3 drugs will be released very fast. Right, because rifampicin goes from, we have done in vitro release where we have seen rifampicin goes very slow. So it's very hydrophobic, so it needs to come very slowly outside. Yeah. So kinetics will be different. Yes.
It would be interesting to do this in TB infected mice and just see the proportion of resistant or tolerant bacteria that you get by using different compositions, because I'm guessing that because the release kinetics is different, you might get different bioavailability in the lung.
Yes, yes, different viability, right.
So,
right. Absolutely.
that
Yes.
needs to be worked out.
If there are no more questions, at least in the hall at present.
I
there's
just had one question. Can I ask?
Okay, you have the prerogative as an organizer.
I've worked very hard, keeping going morning. So the question is follow-up of those two asking about the hydrogel in terms of the release. Have you quantified how much drug gets released over a period of time?
So in mice, we have not done it.
And I'm confused as to whether it's sort of a sandwich where basically it sort of dissolves and the drug gets released, or how does it happen? Because how is the drug release controlled in terms of how much is the quantity that is getting into the body?
So drug will get released with 2 phenomena. One will be the simple diffusion, right? And other will be the degradation of the gel.
Okay.
So these are 2 principles. Although we have not quantified the drugs, the reason is that because when you get drug will quantify, as Ranjan showed, there will be many metabolites, right? So you need to quantify all the metabolites. So that's what I didn't go into that, because then I need to go into the deep mass spec and find. That's why I didn't go with that.
You will need another session then.
Yes, yes.
Thank you.
With the, okay, there's one more last question. Archana, please make it
So
short.
my question is with Dr. Sonali.
Yeah.
So I want to ask you that how have you characterized your household contact cohort, like your LTBI group, whether based on only MON2 or MON2 plus IGRA both?
The household contacts were enrolled based on their exposure to the case, and later on we did both TST and IGRA has been performed in the household contacts. But the definition of contact doesn't go by the TST or IGRA result. It goes by their contact duration exposure to the case.
What was the duration?
We have a definition which goes by, which is universally accepted, that at least 2 hours of close contact in a day or for over more than 3 days a week would be considered as a person who's exposed would be considered as a contact.
Thank you.
All right, I think we'll close the session now. I'd like to thank all the speakers for their interesting lectures, and we can move over for posters and tea.
Excuse me.
I request all of you to stay for.
Yes. I thank all the speakers for shedding light on the latest in TB diagnosis and its importance towards end TB goal. I kindly request our session chairs to felicitate our speakers. Professor Jaya Tyahi to Dr. Sonali Sarkar. Professor Jaya Tyahi to Dr. Tarun Sharma. Professor Jaya Tyahi to Dr. Gulshan Badwa. Thank you, ma'am. I request Professor Sandhya Vishweshwaraya to felicitate Dr. Avinash Bajaj. Professor Sandhya Vishveshwaraya to Dr. Ranjan Nanda. Thank you, ma'am. I request Dr. Nitya to felicitate the session chairs. Professor Jaya Tyahi. And to felicitate Professor Sandhya Vishveshwaraya. Thank you, ma'am. I again thank all the speakers and chairs of session 5. With respect to the posters, we request all the poster presenters to be present at the respective posters for discussion. The posters are placed in the first dome area, that is to the left of you. And we now break for tea and poster sessions. The tea is arranged in the second dome area, that is to the right of you. And I request everyone to assemble back in the auditorium by 5:25 p.m. for an exciting keynote lecture by Professor Talwar. Thank you.
Just a small change in the time at which you need to be back. It'll be 5:10 rather than 5:25. 5:10. Thank you.
Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing, coughing, and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tubercular treatment starts. ATT period consists of 2 phases, intensive phase and continuous phase. The intensive phase is for starting 2 months, which reduces the burden of bacteria, while continuous phase extends for next 4 months to kill remaining MTB. When the person feels good during intensive period and stops medication, the surviving bacteria might persist and can get activated at later stage of life with a property of drug resistance, and hence, until scientists find strategies to shorten this regimen, completion of both the treatment phases is recommended to kill the bug and cooperative management of the disease. The fight is still on. Until then, stay aware, stay healthy. Hope you got valuable knowledge from this presentation. Thank you very much for your time and attention. Hello everyone. My name is Zora Hashmi. I am a PhD scholar at Translational Health Science and Technology Institute, working on accelerating TB treatment strategies. Let us understand tuberculosis. The disease is caused by the pathogenic bacteria, Mycobacterium tuberculosis. It spreads when an actively infected person comes in direct contact of an uninfected person. Direct contact means sneezing and talking. The primary site of infection is the lungs. After entering the body, the bacteria can cause active disease. If the person is immunocompromised or weak, or it can go into latent phase if the person is healthy enough to contain the disease. The body's defense mechanism tries to limit bacteria within a structure called granuloma. As soon as the person gets immunocompromised, the boundaries of granuloma gets weak. MTB starts spreading and damaging tissues, developing into active disease. Symptoms of active TB includes consistent fever with chills, coughing, chest pain, drastic weight loss, and sweating at night. The TB is diagnosed by tuberculin skin test, acid-fast staining, chest X-ray, nucleic acid amplification test, and interferon gamma release assay. Once the person is diagnosed with active TB, the anti-tuberculin
Please note, this is especially for all those participants who are present outside the auditorium to immediately gather inside the auditorium. The keynote address by Dr. Talwar is going to start in another 5 minutes. All those who are at the posters are requested to come back into the auditorium.
Welcome back, everyone. I hope you all had a great interaction with previous speakers over tea. In endemic countries for TB, including India, despite diligently vaccinating more than 98% of neonates with BCG, India continues to have almost a quarter of TB burden and TB-related mortalities, thus creating an urgency on developing new, novel, and superior TB vaccine. Thus, it is befitting to have the most appropriate keynote lecture at the juncture of the symposium. Following the keynote address, we will have session 6 on landscape of TB vaccines. On the behalf of Symposium Organizing Committee, I warmly welcome Professor G.P. Talwar to deliver his keynote address titled Marvelous Properties of Mycobacterium indicus pranae for Treatment of Category 2, Difficult to Treat Tuberculosis.
Slide.
Professor G.P. Talwar is the Director Research at the Talwar Research Foundation, a scientific and industrial research organization. He was the Professor and Head, Department of Biochemistry at the All India Institute of Medical Sciences, New Delhi, from 1965 to 1983. He has been the Jawaharlal Nehru Fellow from 1979 to 1981, the Scholar in Residence at the National Institute of Health, Bethesda, from 1982 to 1986, and the Founder Director, National Institute of Immunology from 1983 to 1994. Besides, he was the Wellcome Professor at the Johns Hopkins Medicine, Baltimore, USA in 1995, and Professor of Eminence at the International Center of Genetic Engineering and Biotechnology between 1994 to 1999. He has received many awards, including the J.C. Bose Award Life Sciences, Jalma Trust Prize in Leprosy, Ramesh Varkas Birla National Award, and Ranbaxy Research Award. He is also the recipient of Lifetime Achievement Award of AIMS for outstanding contributions in medical sciences and humanity, and Bharat Ratna Mother Teresa Gold Medal Award for individual achievement and national development. Professor Talwar was conferred Padma Bhushan by President of India and its equivalent by President of France. He was elected as the President of the International Society of Immunology of Reproduction and was the first President of the Federation of Immunological Societies of Asia, Oceania. He has published over 600 publications in reputed journals. He is the editor and author of 10 books and monographs. Besides basic research, his notable research contributions include the development of Mycobacterium indicus pranae, which is a unique vaccine based on a non-pathogenic organism usable for treatment of leprosy, polyherbal microbicide Basant, and probiotics ProWork Health for reproductive health of women, immunotherapy of advanced stage cancers, and recombinant vaccine for birth control. Sir, I request you to please share your valuable insights with us.
First of all, thank you for asking me to be with you. What I heard in the session before, I was quite impressed with the type of participants you have, with the areas you are covering. I think it's one of the better meetings that I have attended. Now let me go to what I am asked to speak on. I would like to recall an event which took place in 1970 when Dr. Howard Goodman, who was the head of one of these things in WHO, and along with Alvin Cabot and, I think Barry Bloom, they came to me to say that please take up the headship of the WHO Research and Training Center in Immunology for India and Southeast Asia, the so-called FIMSA. They wanted me to take up the headship of it. I was reluctant because, one was already doing a lot, and what is there to take up yet another responsibility? So, I tried to evade their request. And then they told me, look, India has the world's largest number of lepers. Do you think, oh my God, sorry, I'll put it on silent. Sorry. So, I was reluctant, but when they told me that, do we expect Americans to come and solve your problems? I felt very ashamed. That is the time from where I started working on the so-called mission-oriented projects, where one has an aim to do something for a problem which is right there. And what I'm going to tell you is a resultant of that. It's the mission-oriented research, where you start first thinking and working on a problem. And then, you say that if you find a solution, you take it right to the end. Oh my God. Please spare me. I'm in a conference. So you see, as the Howard Goodman had told me that India has the world's largest number of lepers, and it's a awful disease. Whenever we see a leprosy patients, we feel so concerned how his whole lesions in body and etc. So I said, well, if I have to do a mission-oriented research, let me do something for leprosy. And that is how I started working on it. First of all, of course, one has to know why people get leprosy, because everyone doesn't get leprosy. So why does a person become victim of this awful disease? After finding what it is, then you have to take it right through, can we resolve it? And that is what I'm going to share with you. And interestingly, this is a wonderful vaccine that we discovered, a wonderful mycobacteria that we discovered, which is saprophytic. It's not a virulent infectious bacteria, but a saprophytic. This is the bacteria whose gene sequence is now known. And that is why it has been named as Mycobacterium indicus in India. Pran is my first name, and NII is the National Institute of Immunology from where the trials were conducted. So at present, it has gone through the whole cycle and it is approved by both our DCGI as well as by the US FDA. And it is the only vaccine of its type in the world at present. Next slide, please. Next slide. Oh, इधर से यार आप ही किया कर दिया करें. Oh, अच्छा यहाँ. नहीं मैं कर लूँगा. ये यहाँ से करना है ना. अच्छा, thank you. अच्छा. You see, the nature of defect in leprosy is the inability of the person who becomes a victim to mount to react against M. leprae in a immunological way so that he can mount. And this thing, the nature of defect, we have published in 1972. So having found that this is the defect, the next issue was, can we do something to resolve this, to overcome this shortage in the individual? So the strategy adopted was to search for an immunologically cross-reactive bacteria which could elicit immune response not only from the tuberculoid leprosy patients, but also from the lepromatous leprosy patients who otherwise are totally non-responsive. So that was the next strategy. And that ultimately led us to discovery of this mycobacteria from amongst many that we had, at least 5 that we had shortlisted. And I've told you why we have named it as Mycobacterium indicus pranae. It was investigated under the code word MW. Now, the interesting thing is that in this bacteria, the antigens that matter, that evoke the type of desirable response, they are heat stable. So therefore, you could use it as an autoclaved or a heat-killed vaccine. And that simplifies the toxicology studies, which otherwise take much longer to be done. This is the bacteria. It's a beautiful bacteria in my eyes. You see how nice it is. This is you see the Raman spectrum, the atomic force microscopic image of the mycobacteria. It's indeed, you will see it does so many things that one should love this bacteria. Also, its genomic analysis has been done by Professor Syed Hussain, who used to work with me at that time, Akhilesh Tyagi and Anil Tyagi laboratories. And so we know its gene sequence, and it is an ancestor of both M. tuberculosis, M. leprae, and M. avium. So the reason that for tuberculosis I'm speaking on this bacteria is that it is cross-reactive. And this is why it has been named what it is. Now, of course, this particular bacteria has gone through the whole road, before anything can be approved by the drug controller, you have to do all this circuit, the toxicology, phase 1 safety, phase 2 and 3 clinical trials, and then field trials. They were all done. And luckily, in everything, it came out not badly. It came out quite positively. So it's approved by the DCGI of India, as well as by the, by the US FDA. And it is passed on to a company, because if it's to be made available to the public, you have to give it to a company, and we give it at a very, we were grateful that somebody will make it. So we gave to Cadila, and they are selling it under this name. Now you see the benefits of MIP is that if you combine it, chemotherapy you have to do to any patient suffering from leprosy. So all that you can do is you can give the drugs to one and then add the vaccine to the other and see what happens. So if you were to combine MIP along with the chemotherapy, it's much faster bacterial clearance, shortens recovery time, which is usually very long for leprosy, and then it clears granulomas. The reactions which the drugs produce are reduced. And what is amazing is that several people who are normally lepromin negative, which means it's CMI test, they get converted, and the lepromatous leprosy patients always remain lepromin negative. Even after drugs, they are treated, they hardly ever, if they are treated with drugs alone. But if you have included MIP for treatment, many of them became lepromin positive, which means that they have now the immunity to counteract against any future infections. And they are effective in slow responders. You see, this is how it is. Many patients don't get treated with drugs. The curve is very slow moving, but the moment you put in the vaccine, it accelerates the action of the drugs and recovery of the patient. Again, this is a conversion to positivity, lepromin positivity status. Normally, these patients don't become lepromin positive, and they are always subject to recatching the disease, the relapse of the disease. But if you have combined it with this vaccine, then there are many of them, not all, but quite a large number of them become lepromin positivity, which means the future infection will not occur in them. This is how the patients look like when they have been treated with MIP in addition to the drug. See the patient initially and then see what she becomes when she's fully recovered after 1 year. 1 year of such awful lepromatous leprosy patient becoming cleared because along with drugs we added MIP. And see when she becomes all right, you could never imagine that she was once a patient of leprosy. You see, normally with drugs alone, the leprosy patients look always as if they had had leprosy. These are the type of few cases that I'm showing you how they clear up if MIP has been added to the treatment. See the back of the patient and the recovery. Nobody ever thought that he had leprosy. It's a normal back. So this is the other advantages of this wonderful vaccine. It's indeed another one. Look at have you ever seen drugs treating in the way that this result occurred when MIP was added? This fellow was from Nepal and he came and touched my feet. आपका शुक्रिया मैं कैसे अदा करूं? So it is like that. Now I'm going to the subject of today, which is tuberculosis. Interestingly, we have seen that MIP, if you give to the to the guinea pigs, challenge with tuberculosis. See, the A and B is what they become tuberculous. But had you given MIP, see that neither the lung has increased nor the spleen. They are both looking normal. So therefore, it appears that it has acted. This is a trial which is very interesting, and that is that there is a category of tuberculosis patients which are difficult to treat, and category 2 tuberculosis patients. Now, if you were to give along with the drugs, MIP, MW at that time, well then 48 out of 49 patients were fully cured. But with drugs, if you had given only MDT, not all of them get cured. 21 out of 27, and which is therefore the percentage is much improved by adding MIP or MW along with the drugs. And you see, so this is a quite impressive type of results that have been obtained in one of the trials conducted by others, not by me. So it's I'm not influenced in any way. I'm only reporting the results which they sent me after they had conducted these studies. And you see the relapse rate in category 2 tuberculosis cases was much lower if you had given them MW or MIP rather than only the drugs. So in the drugs alone, the relapse rate is much higher. In 2 years, it becomes about half of them go back. On the other hand, had you given MIP or MW, it's hardly any, very few cases relapse. So in both cases, both for treatment as well as for relapse, the MIP played an important positive role. You see, as I told you that this is a wonderful, oh, what happened? क्या हो गया भाई? You see the other things, Professor Sumesh Gupta at AIIMS, you see, uses it for anogenital warts. See, these are the type of warts. Can you imagine MIP curing those warts? So nobody knows what microorganisms were causing that. And this is how they become clear at the Medical Institute, Delhi. See, marvelous effect of MIP on these cases. See the feet becoming absolutely cleared by MIP. So just imagine, MIP has interesting properties, and I'm now concluding that it's a potent invigorator of the immune response, of the immune system. The immunity it evokes, it puts up to whatever is the body suffering from. This is on myelomas, which has been done at the Indian Institute of Science, Bangalore, and they can cure the MIP. And the third one, how the cancer can be prevented or cured even. So to sum up then, it's a marvelous vaccine which is based on a non-pathogenic cultivable mycobacteria. Its gene sequence is determined, so it's not anything. We know what we are handling, and it has been named as Mycobacterium indicus pranae. It expedites bacterial clearance, clearance of granulomas, upgrades CMI responses of BL and LL patients, and it upgrades immune status to react to fresh challenge with mLepre, has high therapeutic action in difficult to treat tuberculosis patients. You see that category 2 were difficult to treat tuberculosis patient. And then it heals ugly warts of anogenital regions and on feet or wherever they are. Imagine warts. And has preventive and therapeutic action on myelomas and many cancers. So my conclusion is that it's a very potent invigorator of the immune response. And what I have presented to you, the credit should also be given to the people who were associated with me in carrying out this work. All of them deserve the credit. Thank you very much, and I'm there to answer anything that you would like. Please feel free to load me with questions. Well, if there are none, then should I go?
Sir, what is your experience with the National Tuberculosis Eradication Program, including this as a part of vaccination strategy in India?
Sorry,
the National Tuberculosis Eradication Program. What is their response to any proposal to include MIP as a part of India's vaccination strategy for all infants?
I have not yet approached them. And maybe
so may I request you to
they don't know that this one acts on tuberculosis also. You see, this study of category 2 tuberculosis is only published in one paper. So it is not largely known phenomena that it is also effective against tuberculosis. Hence, nobody has asked me, but if there is a tuberculosis control program, I would suggest that please try this. It's an already ready, approved vaccine by not only DCGI, but also by US FDA. So it's a very unique vaccine, and it may be helpful in controlling tuberculosis also.
So one more thing, sir, I like to say that you are the inventor of the concept of therapeutic antibodies, including those for cancer. By your work on the HCG vaccine, you demonstrated for the first time that it is possible to generate antibodies against self molecules that are transient in nature when they occur. That has, of course, been translated not by you, but others into blockbuster products like Humira and so on and so forth. But in the light of that experience, would you then recommend that the MIP should be tried in geographies other than India, because there are many factors in play in our country that militate against large-scale acceptance unless it is done in other geographies?
I think it deserves to be used. For the benefit, I cannot foresee the results we will obtain, but chances that it will do very well. I mean, we have had surprises in many situations like these anogenital warts. They were hardly one would have ever thought. But Professor Somesh Gupta at All India Institute of Medical Sciences had difficulty in treating these warts. And he just used it. कि जी पढ़ा है तो देख लेते हैं.
Sir, I have one question. So, first of all, thank you very much. So you have accepted our invitation. So
thank you for inviting me.
It's our pleasure, sir. So I was wondering, has it been compared with the BCG for vaccine during the childhood?
BCG, I can assure you, yes, we have even published. You see, BCG has genetic restriction. It has, this particular one has no genetic restriction. We have conducted studies
I'm asking in terms of the efficacy of this vaccine.
Sorry?
So efficacy of the vaccine, protective efficacy.
It's very, very effective.
Okay.
For tuberculosis, experiments have been done by our own laboratory in mice of various genetic strains, and it works better.
And looking at the results with the leprosy, can we expect this as a therapeutic vaccine for TB?
I think it is therapeutic as well as protective, both immunoprophylactic and immunotherapeutic properties. You see, that's the wonderful thing about it. And what I had been talking and we want to see is, I wish it could be used for eradication of leprosy. Something जालमा के यहाँ है कोई, अगर कोई बता दें कि क्या हो रहा है क्या नहीं हो रहा. You see, because here is something, it's a awful disease, and India still has the largest number of lepers in the world. Why don't we use this vaccine? That is my complaint. It is available. It is approved not only in India, but also by US FDA, which doesn't always happen. All that our DCGI approves is not necessarily approved by US FDA, who are very particular before they grant the approvals. So this particular vaccine has also the US FDA approval. So why don't we test and eradicate leprosy if possible? I am prepared to join hands in anybody who does. And for tuberculosis, please do anything that you would like. I will like to support you and join hands because it has potential. It has potential.
Thank you, sir. It was so great to see you here and hear your talk. So, my question is related to the two questions asked, that in the recent times in COVID, this BCG was being considered.
Oh, by the way, yes, there have been 2 trials on COVID also. Both of them are published. One conducted at AIIMS and other institutions, one conducted in Bombay. Both of them are published. Would you believe it works in COVID? Who expected that to happen? I didn't expect that to happen.
Because
So
you're saying it's
that is why my conclusion is that it's a potent invigorator of the immunity. So anything that the body faces, it can react to.
So that's your answered my question. Yes, this is what
Thank
I was
you.
asking.
Thank you. Chale?
Thank you, sir.
Welcome, ji, welcome.
Can I request Dr. Anil Tyagi to felicitate Dr. Talwar?
Sure. Sure. Yes, please.
I request all the member of organizing committee to join us on the stage.
Dr. साहब आ जाइए.
Thank you, everyone. Moving on to session 6. This session is split into two halves. The speaker of the first half will deliver their talk online. I request Professor Adri Stein and Professor Syed Hasnan to chair this part of session. Dr. Stein hold parallel appointment at the African Health Research Institute, South Africa, and the University of Alabama, USA. At AHRI, he leads the Human Lung Project efforts. He is also a leading authority. He is a leading authority on bioenergetics and immunometabolism of MTB infected host cells, and also investigating the role of nitric oxide, carbon monoxide, oxygen, and hydrogen sulfide in TB pathogenesis. Sir, I request you to chair this session.
Is he on? Is he online? There he is. So it is my pleasure to introduce. It's my pleasure to introduce to you Dr. Carlos Martin, who is a professor of microbiology at the Faculty of Medicine at the University of Zaragoza, Spain. He is a member of the steering committee of TBVI with more than 30 years of experience in microbacterial genetics and molecular epidemiology of TB. He and his team aim to develop novel TB vaccines and vaccination strategies to improve protection against pulmonary TB. Dr. Martin's team designed and constructed MTBVAC, the first and only attenuated live M. tuberculosis vaccine in clinical trials. Dr. Martin has published more than 190 publications and has been continuously funded by the national and European research programs in TB research since 1992. His best hope is to accelerate the phase 3 efficacy studies of MTBVAC in collaboration with BioFabry, the industrial partner of the University of Zaragoza, and with the support of TBVI European programs, EDCP, CTP, and IAVI. Carlos, welcome, all yours.
Thank you very much. I'm going to try to share the screen. Can you see the screen? Yeah.
Correct.
Okay, it's really my pleasure to invite it here. I feel very sorry to don't attend in person the meeting, this important meeting in the day of TB. I think I'm going to try to present the work that we did in the vaccine for the last years and where we are now, that we are now in the phase 3, starting the phase 3 in newborns. It's important to say that most of the work it was founded by different foundations because, as you know very well, working in TB is very important to have the funds for the public money. So in TB, I think we have to be very modest because TB is a very complex disease. It's the one of the kill more people in all the history of the humanity. And we don't understand very well the immunology. We know and we understand that we need interferon gamma, CD4s, but we don't know which is the protection. So as Professor Douglas Young always said, we need good vaccines to have protected people and unprotected and to know what does mean the protection. And we need to understand the immunology to get these vaccines. And for that, it's important to go from preclinical data to be safe the vaccine and then to have efficacy data that will be protect against the disease. When we have a look to the diversity of the TB vaccines in the human trials, in clinical trials. There is 14 vaccines, 2 more in RNA vaccine, but it's not, we don't know which antigen they are using. But if you have a look, you have viral vectors, you have adjuvants that they could have. I'm going to try to use the pointer, sorry. Yeah. We have the viral vectors, we have adjuvants, and they are a limited number of antigens that they use in the different vaccines. You know, you can have the inactivated, as Professor Talwar present the Indicus prani, that they are inactivated. We don't know the antigens, how they change. And we have the old 100 years old BCG vaccine or recombinant BCG with the antigens of the M. bovis strain plus the sign genes that they increase the CD8, or we have Mycobacterium tuberculosis vaccine that is from human origin that we are going to talk in the conference. So we have 100 years old vaccines that they were constructed and we were used for first time 40 years after Robert Koch described the tuberculosis pathogen, the Bacillo de Koch. And after passage of cow strain, what he got the first vaccination, and he vaccinated 50,000 babies, and he saw the decrease, they saw the decrease of the mortality, but not the mortality by TB. This is important for the non-specific effect of the BCG, and it's for that that we are using BCG. But we are conscious that the most important deletion, that is the RD1, the loss of this region is what makes the attenuation of the BCG, the different BCG strains. But it's the antigens that we use for detect the people who is infected, that differentiate from the vaccinated. But by the experiment I'm going to present to you, there are antigens, these SR6 and CFP10 conserved very much in all the strains, that they are essential for the protection too. So it's very busy slide, sorry, but this is the beginning of the construction of the MTB bag that we will be in detail by these 2 independent deletions on FAT26 for the PDIM or the FOP by the regulation of virulence genes. And this in mice, in guinea pigs or monkeys, they were tested by safety and by efficacy in different laboratories of Europe inside of the European programs, and 2 in laboratories of Latin America or Australia. So this preclinical data, they are done by different independent laboratories. And since 2008, we have a small company, biopharmaceutical in Spain, that is called BioFabry, today belong to Sendal Group, that make possible to do the industrial construction of the vaccine, the industrial scale of the clinical, for the production for the clinical trial, but to the industrial scale production. And this is important because in 2012, I'm going to talk later, we have the first data of safety in humans by increasing the dose. And after that, we have the safety data in newborns, and we have now the dose for the newborns and the adults, and this is the start, we are going to talk later, the phase 3 efficacy in Africa. So the concept of the MTB bag is a very simple concept, is the use, the Pasteur principles that they are. He attenuated the pathogen from human origin, not from M. bovis, select the strains that they are more distributed. In this case, we select the lineage 4 that is important in distribution in Europe, in Africa, and in America. But here in India, we could have different strains, but we will see later, we will test that they are efficacy with other strains too. And this is very important, which genes we want to inactivate it. And we want to delete and eliminate the antibiotic markers. It's important because there are not going to be any reversion, because they are deletion. And the deletion, for example, in the 100 years of the use of BCG, never was get any recombination with M tuberculosis, and this is important. ERD1 region never was recombinated using with BCG, is what we expect with this MTB bag. And another point important is to avoid this distribution between laboratories, to use an industrial partner to construct a vaccine that is going to be used in different passages, avoiding all the different chains in the genome. So the clinical isolated, the mother strains of N tuberculosis that they are more frequent in the world is the L4 in red, the L2 in Asian lineage, and in India you have East Africa and Indian lineage that they are here. Okay, we select lineage 4 because it was the most worldwide distributed. And when you compare this with the BCG, you can see that there are some difference, but the main difference is in the ERD1 region. This is a small exported protein, CFP10 exported protein. They are conserved in all the strains of N tuberculosis, and there is no change at all. So the difference between BCG is that the intracellularly BCG is not producing CFP10 or SAT6, and BCG is not exported, is not producing, is not exported, and MTB-BAC is producing both and exporting only CFP10. Why is this important? Because SAT6 exported is virulent, but without the exportation, we have attenuation of the strain. So what we have is the same main of administration of the vaccine. This is a lyophilized that we could vaccinate 20 newborns or 10 adults. And when you compare with the same BCG, the number of epitopes is more than 50% in the difference between MTB-BAC and BCG, and is especially for this region. I will insist in this region. So the final construction of MTB-BAC is a deletion of the two-component system, the FOP, that it controls the 2% of the genome of M tuberculosis. And when you have this deletion, this is implicated in lipids, this is implicated in living intracellularly, in respiration, and the exportation of the SAT6, that this is the important attenuation point. And the WHO asked for a second deletion independent in other region of the chromosome, and we select this FAD26 that is selected for, the people working on the lab, when you subcultivated the strains in the lab, if they lost the DIN, they are virulent. And all the clinical isolated with PEDIN, they are virulent. When we eliminate this FAD26, not only eliminate the expression of the gene, we eliminate the expression of all these genes. So this second mutation is more stable, and the attenuation is conserved all the strain. So we could have here the picture of the M tuberculosis when it distributes compared with MTB-BAC. MTB-BAC, Mycobacterium tuberculosis vaccine, was tested by lipids and microarrays with Musselberg collaboration, proteomics, with different laboratories, with the Stewart Cole or metabolomics is the last thing that we found. So it's a very well studied. This mutation of the FAD26, DIN minus, and the FOP, what is making is you lost all the lipids that interfere with the immunosystem. You can see here that you exposed to the immunosystem. And in addition, the CFP10 is secreted that we think is essential for the protection. And antigen 85, that is under control of TAB, is hyperexpressed too. So MTB-BAC, we have two stable deletions, no mutations, that they are very difficult to be recombinant because never was tested in Mycobacterium tuberculosis. After that, we did different experiments in animal models in collaboration always with different laboratories. I'm going to talk especially in the training immunity results that we have, because BCG is protecting against other infection, and it's important to see if this non-heterologous protection is conserved in tuberculosis. I'm going to show you that it's protecting against different lineage of the strains, and some results on the non-human primates done in the UK. So first, the training immunity is important because BCG is protecting against other disease, and how it's doing that is doing by epigenomic reprogramming, that is the accumulation of histone methylations. They have seen markers in the different cytokines. We saw the same that in BCG in monocyte of the human. In experiments in the mice, what we saw is when you vaccinated the animals, the mice, and then you challenge with streptococcal pneumoniae, not including the vaccine, you can see a reduction of the TFUs and the survival of these infected animals. So MTBVAC is able to protect against other diseases. This was in collaboration with the group of Michael Netea in Holanda. So what in our work, it's only done by single strains, but what we did is to test if this L4 vaccine is going to protect against the other lineage, like lineage 2, that is the Beijing, or lineage 3 that is more common in India. And what we found is that this MTBVAC L4 is the more attenuated of all the construction that we did in the different strains, and is producing the protection against all the three different lineages. And this is important for the future if the vaccine is going to be able to work in the field. So the experiments on the non-human primates, they were published in 2021 by the group of Salazar. The main investigator was Andrew White. And it's important because in the mice, you could use the same inoculation by intradermal route. You can use the same dose of the vaccine, and you can do the challenge by aerosol, like it's not possible to do in the humans. What we saw is the animals not vaccinated doing the pathology after the challenge. You can see a lot of pathology with the BCG was reduced, but with MTBVAC was significantly more reduced. And what is very important is the immunogenic, because we try to see the exact immunogenicity that we are going to see in the clinical trials. We see that the animals that they are vaccinated with MTBVAC, they recognize CFP10, that they are not recognized by BCG, and the unvaccinated neither do. What happened then? That is the control of the disease is better in the MTBVAC, is not so good in BCG, and is not protection at all here. And what you can see here is the MTBVAC recognize this deletion on the BCG vaccine. So I'm going to tell you quickly, sorry, because I think the time is going, but the phase 1 that it was first in human, that it was done in Switzerland, in Europe, because the idea was to test in adults the different dose escalation, 10 to the 3, then safety committee, 10 to the 4, 10 to the 5, like the BCG. So the results of this is published in 2015 by the François Spertini, and the results that say that the safety is similar to the BCG, and the dose response, you can see that they are dose dependent, and there is increase of the immunogenicity in CD4 in this high dose. And after that, the idea is to go to the places where the BCG is used, and then we have the experience of the MBA in 2012 that it was tested in Worcester, here, very close on Cape Town, at the team of the SATV, that they are very expert, and Michel Tamaris is the principal investigator. Why here? Because the incidence of TB is very high, and then we can compare with the BCG. The first was safety too, and then was tested BCG, 10 to the 5, and then 10 to the 3, 10 to the 4, 10 to the 5 of MTBVAC. That you can see the CD4 stimulated with M tuberculosis proteins, that you can see any cytokines will be interferon gamma, interferon alpha, or EL2, and you can see that the 10 to the 5, 10 to the 3 didn't work very well, MTBVAC. 10 to the 4 is identical, the immunogenicity CD4 at day 20, and when you see 10 to the 5 MTBVAC, it was higher than the BCG, and this is the same for 1 year after the vaccination. For this is more immunogenic here. The numbers make that they are significant, this different. What was different to other vaccines in the SATV? That the more different, here you have the quantiferon, that is recognized as CIS and CFP10. And you can see that the babies vaccinated with 10 to the 5 and 10 to the 4, BCG is 0, the reactogenicity. But when you compare the 10 to the 5 and 10 to the 6, there is close of 75 or 80% of the babies, they recognize these proteins. And we believe that is mainly the recognition of CFP10, that it was the correlator protection in animal models. So this was published too in 2019, and the highest dose induced a response higher than BCG. It's important. And it was the CD4 immunoresponse, cellular response, was predominant by the cytokines that they are expressed for the CD4 is highest than the vaccinate, and they support the next step to the clinical trial. So this is unpublished. It was finished, the phase 2A in babies, 2 in South Africa, and 99 babies, they were vaccinated. The 10 to the 3 was eliminated, 10 to the 4, 10 to the 5, 10 to the 6 to see the higher dose. And you can see here that we select for the next study the cohort 2 that is 10 to the 5. So the safety immunogenicity data allow us to do the dose selection for the next trial. And this is where we are now. We are in the phase 3 in newborns, that the idea is will be vaccinated. They start at the end of the last year, vaccinated in 4 different sites in South Africa for efficacy and for immunogenicity and safety in Senegal and Madagascar. This is important because if we get the protection that we believe that we could have, then the immunogenicity we could compare with the other populations like in Madagascar or in Senegal. So this is founded by the EDCTP, the European project, and they are very excited about these 7,000 babies that they are going to be vaccinated, half with BCG, half with MTB-BAC. We are going to identify the case, and we are going to see if the protection of the MTB-BAC finally is better than BCG. So as conclusion, where we are in newborns, we have the phase 1 that it was in adults first, then in babies that they was safe and immunogenic. Here we will select the dose that is unpublished, these results, and then they start the phase 3. So we are going to have data in the next years. It depends on the incidence of TB, of course. In adolescent, this is more slowly that we go because I think it's cleanest to see efficacy first in the babies, but at the same time, we are working in collaboration with the IIV. It's the BioFabric collaborating with IIV for the phase 2 safety first in HIV, and then to go for efficacy. So, and I have to announce that the last year in this TB Day, it was a collaboration between BioFabric, this is the company in Spain, with Bharat and in India, because you will ask, what about India? India, I think, is very implicated in the development of new vaccine and to test new vaccines. And Bharat and BioFabric, they get an agreement. And the idea is to start phase 1 and phase 2 of safety and immunogenicity as soon as possible, and to develop the efficacy phase in the next future. So we are very happy to collaboration with Bharat Biotech, and we hope in the future that we could have some result. And thanks again to all the groups that they help us, IIV and TBVI at the university that we did the construction, and BioFabric, who is doing the clinical and industrial development, and many things to all the organisms that they give money, because in TB, we don't have funded for the big companies, international companies. We have to, the ideal is to have a vaccine, a single dose that it could transport at 4 degrees, and we could use in the place that is more necessary. Thank you very much for your attention, and I will be happy to answer question now or after, as you decide.
Thank you very much, Carlos. I believe we have time for questions.
Thank you.
Good day. Excellent, illuminating talk. A simple technical question. You mentioned about H3K4 methylation. I was just wondering whether, did you do a genomic mapping on that, on the entire epigenome using a chipset, or is it at the same time when the H3K4 methylation is going up, do you see a concomitant K9 repressive marker is going down in the select set of genome or all across the platform?
It was the stimulation of the monocytes with the vaccine, and it was done in the laboratory of Mikhail Netea. That this was the same that it was done for BCG. And so after the stimulation, what was done is the sequencing of this region and see and study the methylation of these regions, of the different interleukins that they were in the promoters, only the promoters. With your questions? Or?
No, I was just curious, was there any heterogeneity across the genome, or was it localized to specific regions?
No, localized of the promoters of the different IL-2 interleukins, the cellular interleukins. No, it was sequencing of this region. Because it's very important now to have a vaccine that could have the same non-specific effect as BCG, because if you want to replace, it's one of the first conditions that we will know. We don't know lepra because Dr. Talwar was speaking about the different interference with the protection of different disease, and this is very, very important. Thank you for the question.
Hey, Carlos, Deepak here.
Hello.
It's a great day for live attenuated TB vaccines when one is going into phase 3. So congratulations. Do you have any indication of the type of CD8 responses that MTB-VAC elicits, or is there a plan to check them in future?
No, we test the CD8, but we don't see much response in the CD8. The CD4, there's very clear compared with the BCG, but not the CD8. But we studied it already. We didn't find.
Yes. Sheet from THSTI. So excellent talk. And congratulations. So I was wondering, how did you come to these two genes for deletion? I mean, was it like...
Great question. First one, the FOPI4. I mean, in Spain, we have an outbreak of MDR TB in the 90s, before the treatment of the HIV people, And it was, in HIV, we have an outbreak that killed more than 100 people. We analyzed this strain and was M. bovis. It was a surprise. It was, again, we go to the M. bovis. It was as M. bovis that it was transmitted by aerosol. It was never was found that. And when we look at the insertion sequence 6110, we found that it was upregulated the FOPI region. So we see the increase of this FOPI4 gene. It was in the year 2000. No one's believed in FOPI4 like virulence region, but now everyone knows that it's key. And the second one, we were working in the lab, and when you subcultivated the strain, very soon the first thing that you found that they lost the virulence is when you lost the PDIN, because it's so big the genes that they are regulating that any change that make the strain attenuated. So we decide independently, completely, one a lipid and the other regulation of genes.
Great, thank you.
Thank you.
I have a question here. Can I go ahead? So I'm curious, I'm quite humbled the kind of work your team is doing. So I'm curious about whether you have checked for secondary immune response for people who are vaccinated. Are you following them up and seeing whether they are actually developing TB or not?
Of course. I mean, for example, in the humans vaccinated in Lausanne, they were followed the study for 5 years. Because the beginning was 9 and never once developed any disease. And for the babies, we didn't see any symptoms of the disease, because the attenuation, at least in the animal models, is higher than the attenuation that we have in the BCG. In the animal models, we can see that they live in the lymph node for 6 months. In the humans, we can see that we didn't see any pathology related with TB. And in adults, it was followed 4 or 5 years.
Data in the adult population, right? And
Sorry?
yet to get the data in the adult population, whether they develop complicated cases of TB like TB meningitis, because BCG is not protective in adults. So probably you guys are also following them up. I mean, these kids into adulthood, whether they develop tuberculosis in future or not.
But I mean, in theory, it's, I mean, we will follow, but I mean, in theory, it's very theoretically that they could develop the disease. I mean, it's the same, the principle is the same that the infected, that they don't develop the disease. That is 90% of the people who is infected never develop the disease. And in this case, it's even more, and it's more difficult because there are the 2 deletions in the 2 main genes. So the only way to get the TB will be if you get another strain infected and the recombinant, and never was demonstrated that for BCG in the 100 years of use of thousands of strains. So I don't see, it's an attenuated strain, like the attenuated strain that we use in the Zika virus vaccination, and it's not a single mutation like it's in the polio, it's a deletion of the region. I don't see.
All I'm asking is that BCG is not protective in adults. So
yeah,
your vaccine is, you know, are you speculating that it is going to protect adults with TB meningitis?
Respiratory, I mean, we have to test. I mean, in India, when the trials they start, we could not, no, I was thinking that you were asking for the reversion of virulence, sorry. No, the protection, of course, in the animal models, we have the non-human primates, and the idea is this, we have the 2 models. We have the babies vaccinated with BCG and 4 years later with MTB-Vac, and we have the adults vaccinated with MTB-Vac, and then challenged with the aerosol with tuberculosis, and the protection is good. Against meningitis, we expect if the BCG was protecting, MTB-Vac too. It's alive, attenuated, it's identical, more genes, but not less.
Thank you very much, Carlos. Really wonderful interaction from the audience. Well, we have one more question. If you can make it quick.
Not very well for P. Not very well. Screen.
Amit, and it's not so much of a question, a comment. Why do we not see many more markers of mucosal immunity from the vaccine community, at least from animal models? I would have thought that surface IgA is one of, especially surface IgA in the respiratory tract, would have been one of the crucial things that one would want to induce with a vaccine. And second, although Dr. Sally Sharp has interacted with our group who advocate immunizing by dry powder inhalation, and Bharat Biotech has been with us for the inhaled COVID vaccine, why is there such reluctance to immunize by dry powder inhalation in case of either BCG or MTB-Vac?
Sorry, I don't know if I understand your question. Sorry, it was a lot of noise. The first question, it was the mucosal. Why we don't get any mucosal? I mean, the route of administration is what we are doing the research in the lab. I mean, it's very, very important. We understand that the long distribution of the vaccine, like it was tested in the monkeys with the BCG, is going to give a better protection against the pulmonary. But you go to regulatory via pulmonary, they will take another 40 years, I think. So the idea is to have a vaccine in a time with a goal pattern that is BCG, and then we use the intradermal, we got the same dose, the same distribution, and this is the idea. But our dream is like in polio. First you have the vaccine inactivated, and then the administrative by oral route, and the TB will be great. Your question is good. But the second one, what do you say, BCG and MTB-Vac?
Immunization by pulmonary delivery of vaccines.
Sorry, the way of delivery. Yeah, more or less the same question.
Vaccine instead of injecting. Put it on the.
This will be a dream, but first we need to know if it's working. And this phase 3, they will tell us, and I think this could change the game if it's really protecting. And then I believe I am not going to see that in my lifetime. I suppose in the aerosol, but I really believe that you administrate by an aerosol, because we are working, in asthma, and in asthma is working only by pulmonary route. And we are working too in cancer, for the bladder cancer, and when BCG is not protecting, in the animal models, MTB is protecting. So we are very confident on the vaccine.
Thank you very much for that, Carlos. I have to cut it short. We have our next speaker waiting. Is Dirk online?
Yes, I am.
So it's my pleasure also to introduce to you Dirk Snappinger. I believe many of us know him very well. Dirk Snappinger joined Weill Cornell Medical College in 2001, where he currently holds the position of professor in the Department of Microbiology and Immunology. His current research focuses on the biology of M. tuberculosis and its exploitation for the development of new drugs to treat tuberculosis. He received awards from the German Studienstiftung des Deutschen Volks, Fund of the German Chemical Industry, the German Research Foundation, and the Ellison Medical Foundation. Good morning, Dirk. Please proceed. I'll let you know when you have 5 minutes left over.
Okay, that sounds good. Thanks, Adi, for the kind introduction. I'm sorry I made you pronounce the Studienstiftung des Deutschen Volkes. Also, thanks very much to the organizers for inviting me, and I hope, as Carlos before me said, you accept my apology for not being able to be there in person. Okay, I'm going to try to talk a little bit about what I think are challenges and opportunities for drug development in academia. I'm going to start with the challenges right away. If you look at the of a typical depiction of a drug discovery pipeline, it's clear that you need a lot of different expertises. And I like the citation from Joseph Bohlen, who once said that making new medicines is the most complex team sport on the face of the earth. So that very nicely, I think, illustrates the requirement for expertises from vastly different fields. And there's no academic lab in the world that possesses the all the expertise that's required to develop a drug. And for, at least for biologists, more so than for chemists, the requirements for drug discovery and development are not so easily aligned with the requirements of a career in academia. In essence, the only way to tackle drug development is as in a large team. And in academia, we all need first, last authorship papers, and that's just very difficult to combine with the need for large teams. So I'm going to pick a few cases where I think an academic lab can facilitate drug development in general, and of course focus on TB. I will point out that I learned a lot from being part of the so-called TB Drug Accelerator. That's a consortium that's funded by the Bill and Melinda Gates Foundation. And the main point I want to make is that it includes several pharmaceutical companies, and they bring expertise to our work that nobody in at least my lab, and probably not on the floor here, which includes 4 different TB labs, has. So I learned a lot from that interaction, and I think that's one of the best ways to actually approach TB drug development. As you probably know, one can distinguish 2 principal strategies for drug development. One is compound first, or also referred to as phenotypic screening, and the other is a target first. I'm going to pick 2 examples how I think we can facilitate both of those approaches. So the way a typical drug first approach works is a primary screen, usually for inhibition of growth. You get some hit. This in this case is a famous one. As part of, usually the primary hits you get from any screen are not suitable for work in animals or demonstrating their potential. So as part of the initial work, there will be hit expansion, SAR, which is coupled with in vitro potency and in vitro liability assays. And then at some point, you need to see in vivo proof of principle. If that has been achieved, one can go on to lead optimization and clinical development. Integrated in this at some point are analysis for the mechanism of action of the compound that one inhibited, but it's not a necessity to actually develop a drug. As you can see from some of the drugs that are still in clinical use, I would argue we still don't understand well how pyrazinamide works. So this is certainly helpful, but not crucial in this kind of approach. But of course, having a target simplifies a lot of steps that one has to go through as one pushes compounds from the left to right in that drug development pipeline. Fortunately, now there are a number of mechanism of action assays that are available, and I'm going to focus on one a little bit, which is the use of over and under expression to elucidate mechanism of actions of a compound that inhibits growth of MTB. And I'm going to use MMP-L3 as an example. I'm sure you're aware of the function of this transporter for localization of mycolic acids into the mycomembrane. That transporter is famous for the large number of molecules that have been shown to A, inhibit growth, and B, engage MMP-L3. And a while ago, we developed a strain to help with the identification of MMP-L3 engaging compounds, in part because there's a large fraction of compounds that comes out of phenotypic screens that actually engage MMP-L3. And it would be good to know which compounds have an MMP-L3 component to the MOA early during development. So we developed a strain, and this is primarily the work of a postdoc, now an instructor, Shipra Grover. She developed a strain in which we can deplete expression of MMP-L3 or silence expression of MMP-L3. This is a so-called TET-on mutant. So with ATC, we actually see higher levels of MMP-L3, and then without ATC, when we introduce or start to silence transcription, there's about a 4- to 5-fold depletion of the protein. And this bidirectional expression allows us to check if compounds have the expected phenotype when you analyze them against the strain, and if they are supposed to engage MMP-L3. Without ATC, the strain is more susceptible to compounds that inhibit MMP-L3, and with ATC, where you overexpress, the MIC shifts to the right, which indicates increased resistance. So that's the pattern you want to see for compounds that are on target and engage MMP-L3, and there are several others that fulfill these criteria known before, and they all come out as bidirectional shift compounds in this assay. It's important to note that as is true for most essential genes, if you silence them, you will increase susceptibility to compounds that directly engage the protein that's encoded by the silenced gene. But it also has indirect effects. So as you can see here, there are 3 off-target compounds that all become more potent when you silence MMP-L3. But these indirect effects can be identified by the fact that if you overexpress MMP-L3, all these indirect or these compounds that are indirectly becoming more potent don't show the MIC shift to the right. So this is a way to directly see if something is engaging MMP-L3 directly, and if that engagement is driving potency of growth inhibition. This is a single strain experiment, and it would obviously be nice if one could at least the underexpression part do on a genome-wide basis. This is a project that was initially initiated by Nadine Rücker, who worked with Sabina and me at the time. And when Nadine left for a job in industry, this project was pursued in close collaboration and actually led by Jeremy Rock's lab with Shuchi, a former postdoc, and Nick, a graduate student, leading the project. So the approach here is we take a CRISPR-I library that targets about 98% of all genes in MTB. This CRISPR-I library is activated with ATC. So we start growing with ATC for a short period of time. And then we compare the fitness defects we have for individual sgRNAs, leading to silencing of individual genes with and without drug pressure. And then you can generate these heat maps that tell you which genes will, upon silencing, sensitize MTB, or which genes upon silencing will cause resistance. So this allows for a genome-wide description of intrinsic resistance determinants in the initial publication that is referenced up here. We did that for 9 different TB drugs. And it was also done for at least one clinical isolate. And one of the main findings that I didn't anticipate was that mutations regarding an intrinsic resistance factor, in this case YB7, is actually inactivated in a MTB sublineage that's endemic to Southeast Asia. And that mutation actually makes that lineage more susceptible to macrolides, which suggests that something like clarithromycin that usually cannot be used, but could actually be used in infections with that specific MTB sublineage. We now use this assay a lot in early stages of TB drug development in the TB drug accelerator to gain an early understanding on how compounds interfere with growth of MTB. We do that over a fairly broad range of concentrations. And if you want to look at genes that sensitize MTB, we look at low concentration of the compounds, and then at the other end of the dose response curve, you can identify genes that upon silencing make MTB slightly more resistant to that compound. And this is just one example. This is for a very low concentration. This is a CRISPR-I profile for 98% of the MTB genome treated with a DPRE1 inhibitor. And then the first gene that comes up as a sensitizer is actually the gene that encodes DPRE1. And this slide is here to demonstrate that for a number of different drugs, you can relatively easily spot the primary target, because these are the genes encoding those primary targets here. For example, ATP synthase operon 4 beta-acqualin come up as sensitizers at the lowest drug concentration. That doesn't always work. There are compounds that have a very diffuse and not so defined mechanism of action, and they will not have informative CRISPR-I profiles. That's actually true for a number of different compounds early in drug development. And this also can be checked if one starts with actually a biochemical assay first to check if the compound is actually on target. And one often finds that compounds that were initially found in the biochemical assays that are then still, that also show whole cell activity. The whole cell activity is often not due to engagement of the target against which the biochemical screen was run. But in essence, the main niche or the main step in TB or generally drug development that people that have my skill set can help with is the establishment of mechanism of action. And this pooled CRISPR-I profiling is one way to do that in a relatively straightforward manner. For switching to biochemical screens, the main difference is that the primary screen now is not one for growth inhibition, but one for inhibition of the activity of a specific protein. And the advantage is now you only need to confirm mechanism of action, less so identify a new mechanism of action. But of course, one of the key questions when one engages in this kind of activity is, okay, what's a good target? What is actually worthwhile screening for inhibitors. And we initially tried to identify targets that we thought would be good for drug development by making conditional knockdown mutants that allow us to silence the targeted gene very efficiently. We use at the time and still do that for certain experiments, so-called dual control mutants, in which we both repress transcription of the targeted gene and degrade the encoded protein with a controlled proteolysis step. And the advantage of this dual control system is that you get much more efficient knockdown. And since doxycycline shows remarkably good PK in a number of different tissues, one can use that then both in vitro and in vivo. And we initially looked for genes that, when you silence them, have an impact on non-replicating bacteria. And this is a mutant for the NAD synthesase. If you deplete MTB of that protein, and when it's non-replicating in PBS, it will die. And depleting MTB of the ability to synthesize NAD also has a drastic impact on viability of MTB in hypoxia, one of those non-replicating conditions that induce a wide spectrum of antibiotic tolerance. And then we, of course, look at, okay, what would happen if we had a inhibitor of NAD synthase, and we mimic this by depleting the corresponding gene product during mouse infection. You see it here, regardless of the stage of the infection, when you deplete NAD-E, there's a drastic impact on CFUs, and the same is true if you look at spleens. We then went forward and utilized these kind of knockdown mutants to ask if the lack of CFUs that you observe in later stages of these infection after treatment are indicative of sterilization. And the answer is sometimes they are and sometimes they are not. So here is one mutant that we used in this animal model. It silences the biotin protein ligase. That's the enzyme that covalently attaches biotin to biotin-dependent enzymes. And you can see if we let the infection progress, the bacteria grow nicely. We add doxycycline that induces depletion of BPL, biotin protein ligase. CFUs go down, and then we take the mice off doxycycline and we wait and ask, is there any relapse? And when we kill MTB via depletion of BPL, then there rarely is relapse. And these are three entirely independent experiments that all gave this result. And these are long experiments, they take about a year. But each of these indicated that silencing of BPL-DUC is sufficient to sterilize MTB infections in most mice. That's not true for all genes. So here's another mutant that actually targets the thioredoxin reductase. You see, if you look at the initial sequence of the infection, it looks very similar. You actually have a rapid depletion of CFUs. But then when you wait, there is actually a relatively high frequency of relapse. So this indicates that lack of CFU alone is not sufficient to claim sterilization. There is in some, in response to silencing certain genes, the bacteria are able to establish what we call a paucibacillary infection in mice. Here, there are no CFUs that we can recover from any organ that we monitored, and we looked pretty much at every organ that you could monitor. And if you outgrow them in different ways, there are just no viable MTB to be recovered. But if you give them time, they actually come back and cause relapse. So unfortunately, this is a very low throughput assay, and it's a long assay with each experiment taking about a year. But if you had to pick, you of course would like to inhibit a target or inactivate a target that prevents relapse. And the only way to find this out is to do these experiments. And I should point out that this is a group effort. You can see each of these points are individual mice. That was a lot of work, and it took a team to get that kind of work done. So what I've mentioned so far is the criterion for target evaluation, where we ask the question, if we strongly inactivate this target, what are the consequences in vitro and during an infection? That ignores a little bit the fact that it's often very difficult to find small molecules that achieve the level of inhibition that we can achieve genetically. So it seems relevant to ask, okay, how much do we actually have to deplete the targeted gene to have a phenotype, either in vitro or in vivo? And we got alerted to that when we worked on the phenotype of biotin auxotrophs. If you look at Chris Sassetti's and Eric Rubin's T-and-seq data in vivo, those are the oldest in vivo T-and-seq data there are. Biotin auxotrophs are actually among the most strongly attenuated mutants there are. But
did you hear that? 5 minutes?
Say it again?
5 minutes?
Yeah, that's fine. So it actually requires more than 99% of inhibition to actually achieve a phenotype in vivo, and that might actually not be feasible to achieve by chemical inhibition. So we, together with Jeremy's lab, developed a system in which we use these genome-wide CRISPR-I libraries, passage them, and then follow the fate of each individual sgRNA over time. And the important part of this is that from the sequence, we can predict how strong an sgRNA is. And that's indicated in the color here. The red sgRNAs are strong, the blue are weak. And when you take these data and plot the predicted sgRNA strength against the impact on fitness, then you can get these vulnerability curves that tell you how much do you need to inhibit a certain target before the bacteria actually have a fitness defect. And if you look at different in vitro essential genes, these curves look quite different. So MMP03 is one where you need the lowest level of inhibition to have the strongest defect on growth. And there are other in vitro essential genes where this curve is much more shallow, which means that you need a much stronger inhibition to achieve an impact on growth. And we now use this kind of vulnerability to prioritize targets for drug development. And if you compare these vulnerability curves for different strains, you can sometimes see differences in genes for which we have inhibitors, and then you can show that strains that are more vulnerable to partial genetic inhibition are also more vulnerable to chemical inhibition of the same target. This, of course, is the BC oxidase. That doesn't always have to be the case, but it is when the compound actually is well mimicked by genetic depletion. Not surprisingly, these kinds of vulnerability curves and vulnerability relationships can be profoundly conditional if you look at genes that are involved, for example, in carbon metabolism. It's known malate synthase is essential with gluconeogenic carbon sources, but not essential with glycolytic carbon sources, and that's exactly what you see in these vulnerability curves. Okay. That brings me to the point of thanking the people who did the work, who I mostly mentioned during the talk, and then finally to announce funding, which of course, or acknowledge funding, which of course is crucial for the work we do. And I'd be happy to take any questions.
Thank you very much, Dirk. Questions from the audience?
Hi, Dirk. Sheetal here.
Hi. Hey, Sheetal, nice to see you.
Nice to see you too. So I had a question regarding the vulnerabilities that you've, so the unpublished data in the different carbon sources. So does that, so can you get inferences from the mixture of carbon sources versus single carbon sources to susceptibilities that you find in macrophages? So what kind of carbon sources are important in a macrophage? Can you derive inferences on that, or have you actually done the screen in macrophages as well?
Yeah, so we have not done the screen in macrophages, and we have not done, well, we have done a lot of in mice, but there are technical problems with the CRISPR-I system that make it quite difficult to, at least so far, to look in macrophages and mice. So macrophages we haven't attempted, and mice we know we have a lot of data, but they are all confounded by certain technical problems that the CRISPR-I system has, one of which is instability of the mutants. So you are absolutely correct that we would really like to know what is the vulnerability profile of MTB's genome in an infection, and macrophages would be a great first step. It is a fairly involved experiment because due to the size of the library, you have to have a large population so that you don't introduce bottlenecks at the beginning of the infection. If you take too small of an aliquot and have, let's say, less than 200 copies per sgRNA, you just run into random seeding effects that just mess up the statistics. So short answer is we don't know that yet. It's one of the things we are most interested in doing, but it requires technical improvements of the CRISPR system.
Thanks, great talk.
Thank you. All right.
Thank you very much, Dirk. Much appreciated. Have a wonderful day.
Same to you. Take care.
Hello. Thank you, Dr. Carlos, Dr. Dirk, and Dr. Stein. Can I request Dr. Amit Kumar Pandey to felicitate Dr. Stein? Dr. Stein, can we have you on stage? Thank you, sir. Moving on to the later part of this session. This part of session will be chaired by Dr. Sudhanshu Varti and Professor G.P. Talwar. Dr. Sudhanshu Varti is currently the Executive Director of Regional Center for Biotechnology, RCB. He served as the Founder Dean at THSTI before moving to RCB. He has made immense contribution to understanding of the biology of Japanese encephalitis and dengue viruses, and on the development of vaccine candidates and antivirals. In addition to several awards to his credit, including the Tata Innovation Fellowship, he is the fellow of Indian National Science Academy, the National Academy of Sciences India, and the Indian Academy of Sciences. I request Professor Talwar and Professor Vrati to begin with the session.
Good evening. Very warm welcome to the last session for the day. Lot of scientific talks for last 2 days, and we really look forward to this last session for the day. The first speaker, there's a small change. Dhananjay Kapse is going to be the first speaker because he has to leave. So may I invite Dr. Dhananjay Kapse. Dhananjay Kapse is an experienced pharmacologist and clinical researcher who has worked in various capacities across different organizations. In Serum Institute, he has been involved in planning, reviewing, approving, and supervising animal toxicology phase 1, 2, 3, and phase 4 clinical trials of vaccines and drugs. He has successfully conducted clinical trials on various vaccines and drugs, including COVID-19 vaccines, the world's first pentavalent meningococcal conjugate vaccine, latent TB infection diagnostic test, and needle-free injection device. Dr. Kapse has contributed significantly towards the development of WHO guidance document on the development of RSV vaccine. He has authored several research articles in peer-reviewed international journal of repute. I have great pleasure in inviting Dr. Kapse to deliver his talk on global TB eradication, disease burden reduction, and elimination strategy. Dr. Kapse.
Thank you, sir, for the kind introduction, and I'm kindly for organizing committee for accepting my last minute request to change the agenda so that I can leave early. I'll be talking on the global TB eradication, certain strategy that Serum is working on for reduction of the disease burden and the TB elimination. So in this presentation, I'll be talking on two. Basically, this is also a part of a previous session on TB diagnostics, because I will be talking on one of the product which is being licensed and now used for the detection of latent TB infection, and a new recombinant BCG vaccine that is under advanced clinical development. So as we know, Mycobacterium tuberculosis is very strange and difficult pathogenesis that is involved in this. And to eliminate TB, that is a goal for the WHO as well as the India is also looking for the end TB by 2030. That is a target that Honorable Prime Minister is looking for. So SIPL Serum Institute is working on strategy towards NTB and to make the products, maybe the diagnostics and the vaccine available, affordable at low cost. And the solution that we are looking at, so the TB infection, the persistence of TB infection, or we call disease persistence, is one of the things that WHO is also focusing on if we really want to eliminate TB. So for that, we need to detect TB infection, treat it, and prevent the recurrent infections. So to detect it by the new next generation diagnostic technique, that is CyTB test, and treat it with the TB preventive treatment, and prevent once they have completed their treatment, then we can give effective vaccine. And similarly in TB patients who complete their treatment, once they complete their treatment, there are chances of TB recurrence. So we can vaccinate such subjects with an effective vaccine. So this is the strategy we are looking at. So in this flowchart, I just want to show that. And so definitely if there are symptomatic suspected TB cases, we diagnose. Once they are positive, they are treated with 6 or 9 months treatment, depending on their category. And once they complete their treatment, as around 10 to 15% of treated TB patients, there is a TB recurrence, that is true TB relapse, or it could be the reinfection that happens in 10 to 50% treated patients within 1 year after completion of treatment. So such patients can be, after completion of treatment, can be vaccinated with the effective vaccine. And when there are close contacts or the high-risk populations, there we can screen this population for the latent TB infections. And we are working on the contact tracing that we have seen effectively for the COVID testing and tracing. Similarly, whenever we do the TB infection diagnostic test, we should have some process in place where we can contact these patients and trace them, so that if they are not coming back for the reading the results, we can trace them, and also if they are positive, we can follow up them for the TB preventive treatment. So once they are seen that as if they are negative, then these are high-risk patients or the close contacts, we can give them effective BCG vaccine. If they are positive, then they take the TB preventive treatment, that is And after completion of treatment, maybe around 1 or 2 months that we can see. And after that, they can be again vaccinated so that they are not again reinfected with the TB bacilli. So this is the strategy we are looking at. Based on this, I will be presenting these two products. So the CyTB, or during clinical development, we called it as a CTB. This is a next generation skin test for diagnosis of TB infection. So this was developed by Statin Serum Institute in Denmark, and they conducted the clinical development of this new product. And after technology transfer, the Serum Institute is exclusive right for the marketing and manufacturing of this vaccine, this product. So as I will skip this back, around 25%, and in developing country, around 50% of the population is TB infected. And they around 5 to 10% of this population, they during their lifetime, they progress to active TB disease. And if we target this population, if we diagnose early and treat them, we will prevent them from progression to the latent to active TB disease. And also we will stop these patients once they become active TB, ultimately we will stop the spread of the disease. And so the CyTB, this is easy to use. So this is similar to the administration, and everything is similar to tuberculin test. So this is easy and can be implemented in the field as a point of care. And it is specific. So it has an advantage over the PPD or tuberculin test because it is based on the specific antigen RD-ESAT6 and RCFP10, which are specific to Mycobacterium tuberculosis. So it is specific as IGRA or quantiferon test. So again, as these specific, because of the specific antigens, it is not affected by the previous BCG vaccination status, which is one of the limitations for the PPD test. So this diagram shows that, see the PPD, so PPD has the various antigens of the Mycobacterium tuberculosis, non-tuberculous mycobacteria, as well as the BCG vaccine. That's why it affected and it's not specific. And that's the challenges currently we face with the PPD. But the CyTB, it has the specific antigens and these are similar to IGRA. That's why it is more specific. Again, administration is same. It is given intradermally, and it is the results are read after 48 to 72 hours, and the induration is measured at the site of injection. And one important, there is a universal cutoff of 5 millimeter for the CyTB to say whether this is positive or negative. That is again the limitation with the PPD, with the different, with the BCG vaccination status or the HIV status, the cutoff changes with the PPD. But for the CyTB, this is the universal cutoff that has been found in the clinical trial for all the patients. So this is available. It is a single dose of 0.1 ml. It contains 50 nanogram of each of these two antigens. It is ready to use liquid formulation. And though in the clinical trials that has been conducted, the population as low as 28 days infants were enrolled, and the upper without any upper age limit. But currently this CyTB is licensed in India for adults 18 years and above. So it is available as 10 dose vial. So these are the certain advantages that I already told you. The cost is also an advantage. There's a robust, well-defined, completely characterized manufacturing. And this is also robust in children. And as in the clinical trial, it has been found that it is robust even in HIV with CD4 count up to 100. The results are same with the healthy adults. Coming to the clinical development, so the various clinical trials, phase 1 clinical trials were conducted in Denmark, UK, and 2 phase 2 clinical trials where this cutoff was decided were conducted in UK and South Africa, TB endemic country, and 3 large phase 3 clinical studies were conducted. So in these 7 clinical trials, more than 3,000 participants were enrolled, out of which around 700 were children and around 500 were HIV infected individuals. So these all 7 clinical trials, these are published. And as I was saying about the BCG status, so this was how it is unaffected was tested in T606 phase 3 clinical trial. So here on the left-hand side, you can see the BCG vaccinated individuals, those who are unexposed. So the red one is CyTB. So those who are unexposed to the Mycobacterium tuberculosis, we can see it detects, so it is the more specificity. But in case of blue, that is PPD, it detects these individuals as a latent TB infection. When we see for the close or the close contacts or truly infected, we can say, so this difference is less. On right-hand side, BCG unvaccinated, you can see in BCG unvaccinated, both almost show the same specificity. So just to summarize, CTP correlates with the exposure of Mycobacterium tuberculosis. It has a similar positivity rate with the QFT, and it has a single cutoff independent of endemic setting, vaccination status, or the HIV status, and it delivers the results, IGRA-like results. And as you know that IGRA need special labs, special techniques, and that may not be possible in remote field areas where definitely the CyTB could be an option. Apart from these 7 clinical studies, ICMR initiated a validation study in India with this product. And in adults, more than 2,500 adults, this study was conducted, it was validated. So along with the 7 clinical trials and the validation study conducted by ICMR in adults, this product is licensed in India for adults. Apart from this, ICMR is also conducting additional the extension of this study in children up to 2 years of age, and that they are enrolling around 500 participants, and that results I think recently they completed the enrollment, and we should get the results for from that study. So the current status, so we have got the marketing authorization for adults, and also this data has also been reviewed by the WHO, and they endorse the use of this new TB antigen-based skin test for diagnosis of TB infection. And recently in January, we have also submitted our dossier to European regulator, EMA, and this has been accepted and under review at their end. And also that we would be applying for the WHO PQ, but at WHO at this point of time, they are evaluating whether this to be considered as a diagnostic test or the drug, because it is given in vivo. So EMA accepted this under the category of drugs. So most probably WHO will also be accepting this dossier on that category. So this is another important thing in the elimination strategy that we are working with MyLab to develop a patch. Okay, so this is a patch when we administered the CyTB in the forearm. The patch would be applied at the injection site with this open circle and this QR code, so that we can trace and we can have all the information of the participant, those who received the CyTB, so that they can be followed for the TB preventive treatment and further actions. So apart from this, we are also working on the new vaccine. This is recombinant BCG vaccine, also called as VPM 1002. And we are evaluating this in infants as well as in adults. And as you must be aware that the routine conventional BCG is a treatment of choice for the non-muscle invasive bladder cancer. So the recombinant BCG has also been tried in phase 1 and phase 2 clinical trial for this indication. So this is a genetically modified BCG vaccine, the same Mycobacterium bovis strain. This is a BCG Danish subtype Prague strain. There are 2 modifications done. The listerolysine gene is incorporated into urease gene, And because of this modification, the antigen presentation is a little bit different as compared to the conventional BCG vaccine. So this is the diagram that predicts the mechanism of action. So this is a recombinant BCG vaccine bacilli. So it enters into macrophage cells and then into the phagosome. As the uracil is replaced by the listerolysine, URAC gene is not active. So the phagosome, the pH becomes less than 6 and it is an optimum pH around 5 to 5.5. Because of this optimum pH, the listerolysine gene gets activated and the listerolysine then makes the pores in the membrane of the phagosome. And that's why then this bacilli is also transported to the cytoplasm. This is the difference between the conventional BCG or the wild Mycobacterium tuberculosis. Wild Mycobacterium tuberculosis, they remain in the phagosome. They are not passed on to the cytosol. So that's how the more antigens are presented into the cytosol, and it is expected that, speculated that this will induce both MSC1 class pathway and MSC2 class pathway, and inducing CD4 as well as CD8 plus T cell responses. Apart from this, these antigens also induce the apoptotic pathway, and because of that, this leads to apoptosis and formation of apoptotic bodies, and these apoptotic bodies then presented to dendritic cells, and this is another mechanism by which the immune response is induced. And as we know that, though it is very rare, but with the conventional BCG, there are some serious side effects that are reported as osteomyelitis or disseminated TB. But as this bacilli, recombinant BCG vaccine, once infected, it causes autoptosis. So it induces immune response and it gets cleared from the body. So the vaccine does not remain in the body for long term. So these serious side effects, though rare, would be another advantage with the conventional BCG. So this slide, sorry for, this is a busy slide which shows the clinical development. So this vaccine was developed by, all of you must be aware, by Stefan Kaufmann in Max Planck Institute. So the Dr. Leander Grode, he was a PhD student and it was his thesis topic, and Dr. Stefan Kaufmann was his guide. So they developed this vaccine and they did various animal studies and it found to be effective, and then it was taken into the clinical development. So the phase 1 clinical trial was conducted in Germany in healthy adults. Another phase 1 clinical trials in TB endemic area, South Africa, was conducted. So the vaccine was found safe and effective in both these studies. Then phase 2 clinical trials were conducted in newborn babies. In phase 2B clinical study, both HIV exposed and HIV unexposed newborn babies were enrolled, and the vaccine was found to be safe and immunogenic in both this population. All apart from this, we are doing 3 large phase 3 clinical trials. One as a TB post exposure, as I said, the prevention of TB recurrence. I will show you in short next slides. And prevention of TB disease in household context. This is again initiated by the ICMR. We are just giving the vaccine. And another trial in newborn babies in 5 countries in South Africa as a head-to-head comparison with BCG for prevention of TB infection. These are the publications for phase 1, phase 2 study. This is, I will just tell, as you know, that the non-specific effects of the BCG during early COVID era, that no COVID vaccine was available or was in early development. We also conducted a efficacy trials with recombinant BCG in India as well as Germany. In Germany, the phase 3 trial was basically to see the hospitalization with the severe COVID infection or the respiratory disease. So in that trial, VPM was well tolerated and showed that the prophylactic effect against severe respiratory disease in elderly. So this is the trial that I was talking about, the household contacts. So this is the prevention of TB disease in close household contacts. Once the index case is detected, the household contacts are screened and then they are enrolled. So total 12,000 participants are enrolled in 1:1. And I would like to tell Dr. Talwar sir that MIP is also one of the arm in this trial initiated by ICMR. So the 4,000 will receive the vaccine, VPM, and 4,000 Imivac, and 4,000 placebo. So more than 6 years. So they, once they are vaccinated, they are being followed for the 3 years. So the enrollment in this trial is completed, and very soon we expect that the ICMR would be doing unblinding and getting analysis done. So this is a POR trial, prevention of TB recurrence. Again, in 2000, this is also based on the animal model, which showed the promising results. So here, 2000 pulmonary TB patient, newly diagnosed pulmonary TB patients, once they complete their treatment, they are screened and they are vaccinated, and they are followed for 1 year to see the TB recurrence case. So this enrollment in the trial is also completed December last year, and we are expecting to complete follow-up by December this year, and maybe by Q1, Q2, we will have the results from this trial. Similarly, as I said, in newborn babies in South Africa, in 5 countries, we are conducting trial in 7,000 newborn babies. Again, the enrollment is completed, and they are being followed for the variable follow-up period of 2 to 3 years after vaccination. And we are looking for the prevention of TB infection in this trial. And we are expecting meeting primary endpoint and the results Q2 next year. Yeah, just again, this is a slide just to show the TB elimination strategies that we discussed. Thank you. Any questions?
So are we going to take questions now, or? Yeah, so we can have, because he has to leave, so we can have some small question answer session. Yeah,
So
Amit, go ahead. Yes.
I have questions so sort of unrelated to your presentation here, but why don't we have an inhaled Covaxin, whereas at least Bharat Biotech has an intranasal installation of Govin or whatever they call it. The second is, why is Serum Institute of India Private Limited willing to listen to key opinion leaders when they know very well that MSC class 2 is loaded in the phagosome lysosome and MSC class 1 is loaded in the endoplasmic reticulum? Neither of these sets of molecules are loaded in the cytoplasm. So what is the rationale for believing that a cytosolic bacterium intact would have its antigens processed and presented on both pathways?
So yeah, so I think Dr. Stefan Kaufman has published, yes, he has published a lot of data on this. And just to answer your question, in the clinical trials, as we speculated that this will induce both CD4 and CD8 T cell response. That I think, yes, Dr. Stefan Kaufman will do that.
Yeah, here is Govardhan Das from JNU. So you told us that clinical trial been phase 1 or phase 2 done in South Africa. And all of a sudden you were doing phase 3 clinical trial in India. Why not in South Africa?
So we are doing a phase 3 clinical trial in South Africa.
But in newborn babies, but not in adults. Is there any problem in there? Why not in phase 3 clinical trial in adults? When you did the safety efficacy study in the initial studies, phase 1 and phase 2, which are done in adults, as a number one question. Number 2 is what you were talking about the particular strain, which is a recombinant strain, it doesn't come out, it come out of the phagolysosome, and you are commenting the MTB doesn't, but truly there are several papers is there in there, the MTB also comes out of the phagolysosome. So, but one differential is not really true.
So the phase one were conducted in adults and phase 2 were in newborn babies. And the phase 3 that we are conducting here in India, basically prevention of TB recurrence, that is in collaboration with the Report Consortium. And I would say that 3 of the principal investigators are also in the audience. And Yeah, and to answer in newborn babies, we would also be planning once we do finish the phase 3 in South Africa to get the license in India. We would also need to do the phase 3 clinical trial in newborn babies in India. That is again the regulatory requirement.
We close, and since he has to leave, we'll take his, yeah.
Thank you, sir. Now I request Dr. Vati to felicitate Dr. Kapse. Thank you, sir. Just a small announcement. All the poster participants are requested to collect their poster back. And now we can continue with the session, sir.
Well, the next speaker is Dr. Vidya Mave. And she's director and clinical research site leader of the BJGMC, Johns Hopkins University in Pune, which is a part of Baltimore, India Clinical Trials Unit. She is coordinator of the Johns Hopkins Center for Infectious Diseases in India and associate professor at the Johns Hopkins School of Medicine. Dr. Mave now leads and coordinates all clinical research activities of the GSU-BICTU, which conducts phase 1, 2, 3 clinical trials of therapeutic drug interventions for HIV and comorbid infections, including TB and hepatitis. My God, a whole lot of Her research interests include TB clinical trials of vaccines and new and reproposed drugs to optimize treatment outcomes, comorbidities, and the use of novel tools to study TB treatment outcomes, and assessing best implementation strategies in programming settings. Dr. Mave has more than 20 years of experience in clinical practice, education, and research in infectious diseases. She has mentored more than 20 pre- and postdoctoral trainees from Johns Hopkins and published more than 100 peer-reviewed research articles. Well, you are quite a bit. Dr. Mave, go ahead.
Thank you for the kind introduction, and I would like to thank the organizers for inviting me to give an overview of a therapeutic TB vaccines. So I will be covering the background, why there is a need for a therapeutic vaccines in terms of how it can alleviate or mitigate adverse TB treatment outcomes, and TB vaccine landscape and therapeutic TB vaccines with focus on characteristic biological feasibility, mechanism of action, opportunities and risks, trial design consideration, and ongoing and impending studies. As a way of introductions, with the latest global TB report from the WHO, we know that approximately 15 to 20% of participants or patients who are getting treated for TB have some kind of adverse TB treatment outcome. That could be either they have a TB treatment failure, or they died, or lost to follow-up, or were not evaluated. And this adverse TB treatment outcome proportion is even higher in HIV infected or persons living with HIV. So what are the factors associated with the poor TB treatment outcomes? In this patient-level pooled analysis, which was published in Nature Medicine, that included 3 different fluoroquinolone-based clinical trials for drug-susceptible TB, they categorized these participants into either low risk, moderate risk, and high risk based on the smear status and cavity status at baseline. And they found that patients who are on TB treatment, for even if they miss 1 doses, for example, could have can translate into poor adherence, can be a determining factor for adverse TB treatment outcomes. And even if the continuation phase, the adherence to TB treatment for all doses of TB treatment is very critical. So the WHO TB report really doesn't account for recurrence, TB recurrence. So we know that TB recurrence is pretty common. It occurs in 8% of the participants who successfully complete TB treatment with first-line treatment. And this proportion is even higher in India at 10 to 12%, and it may constitute up to 5 to 30% of the total TB burden. It could be because of reinfection or relapse. And in our own patient cohorts in Pune, India, we have followed about 889 participants who are followed for at least 12 months after completion of TB treatment. Our recurrences were up to 10%. And another issue with the TB recurrence is that there could be a possibility of a subclinical TB recurrence. And in our cohort, we have noted that the number of subclinical TB recurrence, that means that patients are asymptomatic, but they have smear or cultures positive in the follow-up visits. So this was as high or as common as clinical TB. In addition to the known adverse treatment events, post-TB mortality and morbidity could be also because of pulmonary impairment, which could be due to fibrosis or bronchiectasis, and can be one of the reasons for more than threefold increased risk of mortality after patients are declared cured of TB treatment outcomes. So we know that over the last 2 decades or so, the TB case notifications have been coming down, but they are not coming down at the rate which would be sufficient to reach the target, to achieve the target of TB elimination by 2030. So therefore, in addition to what we already have, the potent TB drugs and also the newer repurposed innovative TB drugs, we also need TB vaccines as a tool, and it is an important tool. And in this modeling analysis, it was found that a TB vaccine, which is 40% effective, could have durational protection up to 5 years. As the vaccine effectiveness goes higher to up to 80%, the vaccination efficacy duration of protection could be as high as lifelong. So what are the potential vaccination strategies then? There are 2 different vaccination strategies, pre-exposure prophylaxis and post-exposure prophylaxis. And the patient target populations are different in both of these cases. Pre-exposure prophylaxis, what I mean by that is the target patient population is MTB uninfected. And the target indication is prevention of infection or prevention of disease. A post-exposure strategy is basically among those who already are MTB infected and or had TB disease or have already been completed their TB treatment, and the indication could be different here. It could be prevention of disease, therapeutic TB vaccines, and or prevention of recurrence indication. So this slide you have seen already in the past 2 hours or so. There are at least 15 vaccines which are in various stages of development, and I'm not going to belabor that. And so for the therapeutic TB vaccines, so what is the indications or what it does really do? So the idea is that these vaccines can be given to the persons with TB disease adjunctively with the TB treatment, which we're supposed to induce, at least we should induce appropriate immune responses, and primary aim is to achieve individual benefit, but it ultimately could have a population level benefit. So the basic characteristics of therapeutic TB vaccines could be as per the WHO characteristics. The primary target would be it should improve the cure rate and decrease the incidence of recurrence and should have a favorable safety profile. And the target population could be either drug sensitive or drug resistant TB disease and should be able to cover or have induced immunity among HIV, persons living with HIV, and should have an essential long-term targets. And there are some evidence with the biological feasibility. We already heard about MIP vaccine earlier, preclinical evidence showing that it is protective along with when it's given along with the TB treatment. Similarly, ID93 vaccine also shown that when it is given along with the INH rifampicin, the survival rates are much higher among mice. So biological feasibility of VPM1002 was also published very recently, which also showed that compared to BCG vaccine, the recombinant BCG vaccine had a lower bacterial burden in the lungs in mice. There are some clinical evidence we already know about the POD trials, prevention of disease trial with the M72 vaccine, which showed that it prevented TB among TB infected adults by 50% ratio. And there were some very smaller studies which are clubbed together in the meta-analysis, about 25 studies involving 2,281 patients with MDR-TB. The pooled data showed that smear conversion rate was decreased by over almost fourfold, and absorption of TB foci was also improved by almost 4.4-fold, and cavity closures were improved or optimized by over 3.5-fold. So, however, these kind of data are limited by smaller patient population. So the mechanism action for therapeutic TB vaccines they are to provide host-directed therapy to modulate the immune response. I'm not an immunologist, so please bear that in mind. From immune response from a Th2 response to Th1 response to reduce the extracellular growth of bacilli. So the vaccine then can be administered at the start of treatment or soon afterwards. In case of the second mechanism, the killing of the bacilli by boosting the immune system, their vaccine would be administered after the significant reduction of the initial bacterial load has been achieved by giving the pharmacotherapy. So some of the examples which could be MIP vaccine, ID93 vaccine, H56 vaccines, and Ruti vaccines, which could be potentially could be a target vaccine for this indication. There are many drugs and various stages of development for therapeutic TB vaccination indication, as shown here, and many of them are subunit vaccine. There are VPM vaccine, which is a live vaccine, and there are a couple of whole cell vaccines which are also currently under consideration. So what are the opportunities with the therapeutic TB vaccines? So the potential therapeutic TB vaccine can be used as a prevention of recurrence indication. So ID93 vaccine, GLA-SE vaccine, was evaluated in a phase 2A trial in HIV uninfected adults with drug-sensitive TB and showed good safety indication. There was no safety signal. H56 IC9031 vaccine currently completed its enrollment as well as follow-up in drug-sensitive TB on a phase 2 trial. And there's the M72 vaccines. We already heard about that. That is a drug-sensitive pulmonary TB participants has been evaluated, and VPM1003, you'll hear a little bit more later on. And improving cure is another opportunity with therapeutic TB vaccines, and there are several vaccines as shown here have been proposed for this indication, including MVACI, Mbindicus prani, Ruti, H56, and ID93. And finally, could these therapeutic TB vaccines be used to simplify TB treatment? By that, what I mean is to shorten the duration of total TB vaccine, TB treatment duration. So there are several considerations to be given for trial designs, because when we are giving a therapeutic TB vaccines, we are also giving TB treatment, which itself could have adverse events or side effects. So therefore, we have to be mindful of safety, balancing safety, efficacy, and also ensuring that we also evaluate certain other aspects which generally do not include, like, for example, lung inflammation or impaired lung disease. So what is a possible design of TB therapeutic vaccine trial? So So we generally start everybody on TB treatment at the TB diagnosis, and then we follow them up. At least current standard of care is 2 months of intensive TB treatment. Could we then give the vaccination at the end of intensive phase of TB treatment? Or could we give it at the end of TB treatment? So potentially the idea for giving it at the end of TB treatment is for prevention of recurrence then. Then we do follow them up for at least a minimum of 12 months to see whether there are any adverse events or adverse outcomes, TB treatment outcomes, which needs to be evaluated for these trials. So one consideration to ensure safety by giving a TB vaccine during TB treatment is to do a time de-escalation strategy, where you start with giving, say, an ID93. I'm just giving a hypothetical example. It's ID93 vaccine. So there are trials which have been completed where the trial vaccine was given at the end of TB treatment. It was considered to be safe. Now, if you want to give it at end of intensive phase, then we need to de-escalate on time and ensure that there are no adverse events based on the sample size which has been evaluated. And lastly, improving cure among persons living HIV is very critical for many reasons, because their proportion of persons living with HIV with the adverse TB treatment adverse TB treatment outcomes are really very high, over close to 30%. That is unacceptable, and that they are also on ART regimen while they are getting TB treatment, so their adverse event profiles would be much higher. So therefore, while we are administering TB vaccine in a trial status or trial during clinical trial, we should be considering when to include these patient population in these trials, and what would be the CD4 eligibility criteria, so that we safeguard the vaccine for further development, so that the number of failures from the vaccines are bound to be lower at that time. But of course, there would be a requirement for a proof of concept trials before moving it forward. And finally, one of the considerations should be whether therapeutic TB vaccines can also have any efficacy or efficacy against TB-associated lung function impairment. Of course, there are several RNA signatures which are currently being studied for TB disease progression and decrease in TB treatment adverse outcomes, and potentially whether these vaccines also can show those kind of signals through those biomarkers. So what are the preferred product characteristics for therapeutic TB vaccine for the consideration is that there should be an indication where we know what is the duration of protection from recurrence by therapeutic TB vaccines, and that would that be suitable for the program, and would they have very good safety or immunogenicity profiles. I'm happy to report that there are a really crowded landscape of therapeutic TB vaccines or POR vaccines right now. You just heard about BPM-1002. And another trial which is completed enrolling as well as follow-up is H56. ID93 and Ruti vaccines are in advanced stages of development as we speak. So VPM-1002 is currently in a follow-up phase, recruited all of their 2,000 participants, where participants were between the age of 18 to 65 years, were enrolled into either the vaccine group or the placebo group. And then at the end of TB treatment, and then they are being followed for at least 12 months. And along the way, there are several points where blood is collected for PAX gene as well as PBMC, so that we can do the further evaluation of this trial. And this trial is currently being conducted under Report TB Consortium, along with the Report TB Consortium, which Dr. Sonal Sarkar earlier mentioned, and Report India, which is a joint collaborative project between Indian institutions and US institutions, have initiated the study along with approximately 15 sites total in India and Bangladesh, and have proposed to enroll 25% of the participants and enrolled over 40% of the participants, and follow-up is going to be continued until the end of 2023. And currently several immunological pilot studies are ongoing collaboratively. The second study, which has completed actually enrollments as well as follow-up, is H56, basically a protein subunit vaccine, which has 3 different protein subunits as shown here: AG85B, ESAT6, and RB2660C. And then the difference between this trial and our VPM trial is that the patients are enrolled at the beginning of TB treatment and then are followed for almost 18 months beyond that. A third trial, which is going to be proposed under ACTG, ACE Clinical Trial Group, which is an NIH network, National Institute of Health Network trial, along with ACTG and HIV Vaccine Trial Network. We'll also look for ID93 as a therapeutic TB vaccines, where time de-escalation may occur. As many of you know, it is similar to M72 vaccine, except that it has 2 additional antigens and adjuvant is different. So what is our guiding questions for therapeutic TB vaccine? So what TB vaccine candidates favor risk versus benefit balance, and that whether we have enough confidence based on our phase 1-2 trial, 2 trials to progress or progress them to other trials, including phase 3 trials for its ultimate rollout. So in conclusion, TB treatment outcomes for all forms of TB is suboptimal, especially for people living with HIV. There are opportunities to develop TB vaccines that may reduce the risk of recurrence and post-TB morbidity and improve cure, as well as simplify TB treatment, including shortening of TB treatment. But we must consider novel trial designs. And I strongly believe that the TB vaccines, therapeutic TB vaccines, may contribute to population level impact once its safety and efficacy has been confirmed. And these are my acknowledgements. Dr. Gavin Churchyard, Mark Cattrall provided some of these slides, and we have several funders, including Department of Biotechnology for our Report India Consortium. And this is our study team in Pune, where we have a large clinical trial unit conducting multiple clinical trials as well as TB observation cohorts. Thank you.
Any questions? Yes, please. Judy, micro.
Dr. Mawe.
Yes, sir.
It is correctly stated that one of the important limitation of vaccine development in TB is our lack of understanding about correlates of protection.
That's right.
And it is also said that we will understand the knowledge or information about the correlate of protection only when a vaccine works. Now, BCG works extremely well in children, and it protects millions of children's life every year and works with very high efficiency.
Right.
Is there any information about what we have learned about correlates of protection with this very high protection of BCG that it provides in children?
Okay, so I actually don't have that data with me right now, although I can get back to you and
But it has been
consult
working
with
for a very long time,
Right,
and
sir.
successfully.
Right, right.
And if not, why do we think another successful vaccine will provide that information?
Right. So I think based on at least the preclinical data and the early phase clinical trial data, and that is when I think the investigators, I mean, whoever the investigators are, feel confident in the immunogenetic profile and also safety profile to move it forward to a phase 2 trials. Right now, similarly, what we have done for VPM1002, obviously, the mechanism of or immunogenetic profile of the early trials were comparable to BCG. So at least compared to BCG. So I think that is the bar which WHO right now says is minimally required. The bar for any vaccines to move forward is to be at least as good as or as effective as BCG vaccine. I think that is probably is the consideration, although I'm not going to have the correct answer to your question at this time. So that is the consideration that's been given.
Okay, thanks. Any other question? If there is none more, thank you very much. May I say that today is the World Tuberculosis Day. It so happens that it is today. And in today's one of the newspapers, our Prime Minister Narendra Modi has said that India is going to bring tuberculosis to zero, to eliminate, to eradicate tuberculosis. So I think what I'm hearing, maybe everybody's working towards that. Please continue doing so. Let us get rid of this disease, awful disease. Now, again, my friend will, and my former colleague will take the last speaker.
Thank you, Professor Talwar. The last speaker for the session is Dr. Deepak Kaushal. Dr. Kaushal is the director of the Southwest National Primate Research Center, hosted by an independent research institute, that is Texas Biomed. Under Dr. Kaushal's leadership, the SNPRC became funded by a $50 million research center, and the only such center which harbors BSL-2, BSL-3, and BSL-4 facilities for NHP research. The SNPRC is currently amid a tremendous growth phase and is committed to providing NHP resources to all NIH-funded and other investigators. This center was one of the first to develop NHP models of COVID-19 and led the testing of Pfizer, Novavax, Regeneron, and several other vaccines or therapeutics against this pandemic. Dr. Kaushal's lab focuses on several different aspects of MTB infections. Many of these areas utilize the non-human primate models of TB and TB AIDS. Based on the natural route of MTB infection, which has been optimized to understand the response of MTB to in vivo stress, development of attenuated vaccines against TB, and find host-directed approaches for better treatment of TB. It's my pleasure to invite Dr. Kaushal to deliver his talk, which is related to prevention of tuberculosis in a non-human primate. Dr. Kaushal.
Thank you very much, esteemed co-chairs, for those kind words. I don't know if I deserve them, but thank you. Thank you also to the organizers for two things. One, for giving me a talk. And then secondly, for more importantly, for providing such a wonderful and enriching program. I've quite enjoyed it. I know many other people have. In fact, it's late in the day. I don't know how many people will be back tomorrow. So would you please join me in thanking all the organizers with a round of applause. Thank you. I also know it's late in the day, and thank you all who have stayed here for my talk, although I suspect it might be dinner that you've stayed for. So I work at a primate center, and so I'll be talking mostly about the primate work that we do. So about 15 years ago, my lab, and I'll try to go about as fast as possible because it's late in the day. So my lab set up these models for monkey TB using Indian rhesus macaques that are aerosolically infected with MTB. We believe it's important to infect TB, monkeys with TB via the natural route because that's how it happens in humans. And so you can see here we have a chamber in which everything is contained, and we can use a nebulizer and a class 3 environment and infect these monkeys via aerosol. The reason why we infect monkeys is because we get disparate responses to MTB infection. So here I'm showing you two different lungs from two different animals that were infected exactly the same day, exactly the same time, with exactly the same strain and exactly the same dose of MTB, and yet the monkey on the right ended up developing latent TB, no signs of disease, a few granulomas 6 months later when we euthanized the monkey. And the monkey on the left ended up developing 2 months later lobe level consolidation that you can see on the lung, and huge granulomatous inflammation. And this way, we are able to get monkeys to recapitulate the wide spectrum of disease that we see in human TB and everything in between. And nowadays, we don't even have to actually euthanize the monkeys. We can use PET CT scans to know which monkey is going to have extensive lobe level or lung level disease versus which animal is going to have 1 or 2 or 3 discrete poorly avid granulomas as seen on this image. Another reason why we use monkeys in TB research is because they can very well recapitulate MTB HIV co-infection. That's a program in the lab that I won't have the time to talk about today. So when we infect monkeys with MTB, granulomas form, and these granulomas look very much like human granulomas. They evolved very much like human granulomas. They have features of central caseation and necrosis, and then beautifully layered first myeloid, then lymphoid, and then fibrotic layers as shown earlier by Dr. Mehra. And as shown by Dr. Stein, we can see exotic presentations also. I won't have the time to show you all of those, but we've published those. And we believe that it's really important to study TB in the context of the natural granuloma because of the pressures that these environments put on the bacilli, and the resulting immune response is irreversibly changed as a result. And if we study these, both the immunity as well as the pathogen in a context of a classical mouse lesion, which does not have this ordered structure, then results could be inaccurate. I'm going to skip the slide. This is just the techniques that we'll be using today. I think these slides have shown that MTB infection of macaques recapitulates key aspects of the human TB syndrome. So coming to the actual story today, and this story started when I left Professor Tyagi's lab, where there was a focus on gene regulation in MTB. Unfortunately, Professor Tyagi is here, and joined Professor Bishai's lab at Johns Hopkins 20 plus years ago, where his focus was to study alternative transcription factors of MTB, particularly sigma factors. And he and I developed a project to study sigma H, one of these alternative sigma factors. And we discovered that sigma H actually responded to redox stress and induced thyroidoxins and thyroidoxin reductase regulon. This was a really important result because through the chemical reaction that I'm showing you on the screen over here, thyroidoxin can scavenge oxidative stress, particularly peroxide stress, and thyroidoxin reductases can then re-reduce thyroidoxins so that they can scavenge more oxidative stress. So if you can imagine a situation where an MTB bacilli is phagocytosed into a macrophage, it comes under the assault of at least 2 different important enzymes, INOS and FOX, both of which produce oxidative stress via superoxide and peroxide ion production. And SIGH is an important virulence detoxification pathway regulon that scavenges the stress, allowing MTB to survive this initial innate assault and therefore persist and replicate in future. There are consequences. So I think we all agree that SIGH is a master regulator of stress that exists in the macrophage. Several labs, including mine, have since discovered that SIGH is induced in other stresses as well, INOS, nitric oxide phagocytosis, oxidative stress, heat shock, and even cigarette smoke induce this transcription factor. There are consequences of not having SIGH because of the inability to induce thyroidoxin reductase. So in a model that you can see here, where 100% of monkeys infected with TB develop TB and have to be euthanized within 2 to 3 months, the monkeys that are infected with the isogenic SIGH mutant are completely devoid of disease, and they have 100 to 1,000-fold less bacilli, as you can see here. And by pathology, you can see here in the lower panel, animals infected with the sigma H mutant have no disease, beautiful red-pink lungs, an occasional discrete granuloma, but look at the animals, the lungs of the animals infected with wild type MTB, miliary presentation, hundreds to maybe thousands of granulomas. And this result can be recapitulated in just ex vivo in activated macrophages. So SIGH infected non-human primate macrophages, there's an attenuation phenotype, and the same is true. Now we've shown in human activated macrophages. These human activated macrophages actually have a very strong hyperimmune phenotype. So this is RNA sequencing of macrophages infected with SIGH versus wild type, inducing in the hot color, in red color, immune genes only in SIGH infected macrophages, and also trained immunity phenotype inducing all of these genes involved in glycolysis just in SIGH. And the mutant also fails to inhibit phage lysosomal fusion. We are showing you this data with LC3 co-localization assay, and as you can see, mutant infected macrophages have a lot more phage lysosomal fusion. So we hypothesized that maybe antigen presentation with this particular mutant may also be altered via class 1 cross presentation, because this mutant is allowing phagolysosomal fusion that is prevented or inhibited by MTB. And we generated an ex vivo antigen presentation assay where macrophages, human macrophages are infected with one or the other strain of MTB, either the wild type or the mutant. And there's a T cell hybridoma that we have that only recognizes the antigen 85B, actually one epitope of antigen 85B. And the readout is IL-2 production. As you can see here, macrophages that are infected with the SIGH mutant present much more efficiently to the T cell hybridoma, resulting in presentation of secretion of much more IL-2. And this is a classical autophagy antigen presentation cycle, which can be reversed by silencing Beclin-1. So the data that I showed you in the last 2 or 3 minutes indicates that SIGH is 1, attenuated in NHPs, and 2, induces immune responses that far exceed those are elicited by MTB itself. But wait, aren't these the 2 attributes that we would want in a vaccine? So that led us to thinking about using this particular mutant as a live attenuated vaccine for TB. Carlos Martin talked about MTB-Vax, so I don't need to give you all the details about the advantages of these vaccines, but I'd like to reiterate the point that these candidates would, one, offer an almost complete complement of MTB antigens to the immune system, and therefore result in a broad spectrum elicitation of immune responses that are natural, that are very close to natural. We do understand that there are some people that may not want to take TB-based vaccines, and in order to comply with Geneva consensus, it may be necessary to generate multiple knockouts. But nonetheless, we embarked a few years ago on a series of experiments to study if aerosol vaccination with Delta SIGH would protect non-human primates against lethal TB. So we took a cohort of rhesus macaques and divided them into 3 groups, either unvaccinated in red or vaccinated in the current India cricket blue jersey, aerosol BCG, and vaccinated with Delta SIGH mutant, the old India blue color cricket jersey. And as you can see here, 3 to 4 log reduction in the SIGH vaccinated animals as compared to unvaccinated, about 3 logs better result as compared to BCG. This is data in the lungs. Here's data in the lung draining lymph nodes, and we have published data in extrathoracic compartment as well. There was a huge vaccine effect, but the best data actually is in this top panel, which is a sub-gross pathology panel, where you can clearly see for 1 block per animal, and we've published this data for multiple blocks, the amount of disease that's there in this particular, the amount of granulomatous disease that's there in this particular animal, this particular block that's unvaccinated, about 50–60% of the lung is involved. There is some reduction, maybe 2-fold reduction by BCG vaccination, and almost complete blockade of granuloma formation by SIGECH vaccination, and the granulomas that form are much smaller and rarely necrotic. And the lungs of these animals that are vaccinated with Delta SIGECH are characterized by strong localized T cell responses. So if you can see here, same color code, these bars, these Delta SIGECH vaccinated bars, these are bowel samples, bronchoalveolar lavage samples that we've pulled out of these monkeys at different time points after vaccination. So there's no TB effect here, just post-vaccination. You can see influx of both CD4 and CD8 cells into the alveolus of these monkeys after SIGECH vaccination. And these T cells have all the right attributes. They are activated, they're tissue, they are expressing markers of tissue residence. So they're going to go into the alveolus where we are measuring them, but they're going to go through the interstitium and into the lung because they're expressing CD69 and so on and so forth. But most interestedly, these animals also had strong eyeball bronchus-associated lymphoid tissue responses. These are ectopic B cell structures that Shabana Khader and I have shown are associated with protection, and we see them a lot in the SIGECH vaccinated animals. Whatever granulomas we see in SIGECH vaccinated animals have strong eyeball. Here we are staining for eyeball with a B cell antibody in pink, and you can see them clearly. And in fact, the response is so strong that you don't need confocal staining to see them. You can actually see them right through the H&E. And these you don't see in BCG vaccinated or unvaccinated animals. As you can see over there, there's a very strong statistical effect. And we've since gone on and showed that it's not just a correlation with eyeball or GRALT, but these structures are actually responsible for the protective efficacy of SIGECH. This work has just been published in Nature Immunology, co-corresponded by both Shabana and I. The paper is actually supposed to come out today, TB Day, but unfortunately it hasn't yet come out. So what we did here was we depleted B cells from these animals using an anti-CD20 antibody. Joanne Flynn's actually used this antibody before, but it causes serum sickness in macaques. So we semionize this antibody so that it doesn't do that. And we found out that if we treat monkeys every 3 weeks with this particular antibody, then you can deplete and keep depleted B cells from blood, from alveolus, from lungs, from lymph nodes, from every other tissue. And when we vaccinated these animals with Delta SIGECH and then challenged them with MTB, just like the experiment that I showed you earlier, but then we took a subset of monkeys and depleted B cells, and we took a subset of monkeys and they just had isotype control antibody. Then the B cell depleting antibody animals completely were devoid in their lungs of this eyeball structure that I showed you earlier. And in these monkeys, disease came back, as you can see here, CFU per granuloma in the specific antibody treated group versus isotype treated group. And the T cells of these monkeys are actually depressed for activation. So we actually believe that these B cell structures inform T cells or educate T cells that rush to the site of the infection and are responsible for their activation. So I think thus far I've shown you that aerosol vaccination with Delta SIGECH protects against lethal TB in our rhesus monkey model by inducing the right kind of immunity. I won't have the time to show you data, but we've published separately that SIGECH is actually safe in our model in the setting of HIV co-infection. And so we thought at this point of time that we were in a safe zone, and human testing of Delta SIGECH or derivatives of it was very much in order, but not so fast. Funding agencies wanted us to test the same result either in a different lab or in a different monkey model. So we chose a different monkey model, which is the cynomolgus macaque model that some other people in the field use. We don't have that much experience with it. We use rhesus macaques, but we did a second experiment. It's actually a large experiment. The previous experiment was N of 6, so a total of 18 animals. Here I'm showing you data from 27 animals, cynomolgus, but exactly the same result, significant effect with SIGECH aerosol vaccination over BCG vaccination. And in fact, if you look at the number of sterile lobes, clean lobes that have no MTB growing out of them, and then you only find them about 15% of the time in unvaccinated animals, 40% of the time in BCG vaccinated animals, and about 80% of the time in SIGECH vaccinated animals. And again, with gross or subgross pathology, you can see the effect of total number of granulomas in unvaccinated animals in that one block relative to BCG and SIGECH, and there's a progressive and significant effect there. I've earlier shown you immune responses. We now know that these immune responses, these T cell responses that rush to the lungs of these animals that have experienced SIGECH vaccination are actually antigen specific. So these are cell wall peptide specific. And we see this effect with CD4s at week 3, and then at week 5, as expected, a little later, we see this effect with CD8s because they require CD4 help. So you've got CD8s expressing gamma and polyfunctional CD8s expressing gamma and alpha, and actually granzyme B, so loaded and licensed to kill CD8 cells, are coming to the lungs of these animals. We've also done single cell RNA sequencing on the BAL and the lungs of these animals. And I won't show you all the detailed data. We've isolated all the clusters, but you can just appreciate the total number of cells that are present 3 weeks after SIGECH vaccination as compared to BCG vaccination over here. It's a huge difference, and compared to no vaccination over here. And this effect is retained after TB infection. So 3 weeks after TB infection, you still have a number of clusters and number of cells significantly more than BCG vaccination or no vaccination. And there are multiple effects here. So there are some clusters of T cells that are SIGECH specific. So here the data is no vaccination, BCG vaccination, SIGECH vaccination, and then no vaccination with TB challenge, no BCG vaccination, TB challenge, SIGECH vaccination, TB challenge. So there are certain clusters and genes that are being expressed in them, both canonical as well as novel, that are just SIGECH specific. You don't see them in any other vaccination. And then there are some clusters which are specific to inflammation. You see them in SIGECH vaccination as well as TB infection. So we do believe that these will allow us to determine correlates of protection, at least in this setting. So I think what happens is when MTB infects the macrophage, SIGECH and antioxidants are induced, and they protect the bacilli, leading to its survival, leading to the formation of a granuloma. And then this granuloma sort of becomes centrally necrotic and assumes a immunosuppressive phenotype, which is high IDO, high IL-10, high PD-L1, high TGF-beta of the type that Dr. Mehra talked about earlier. When SIGECH comes in contact with macrophages, there's no antioxidants, and 3 things happen. Either a granuloma doesn't form because bacilli are hammered into submission by the host's innate immune response, or even if a granuloma forms, it's non-necrotic and very small. And there's no blockade of phagolysomal fusion, leading to a more efficient class 1 presentation. And the third thing that happens that I didn't have time to show you today, but is included in the Nature Immunology paper, is there's a IFN IRF4 BCL6 nexus that is responsible for bringing these B cell follicles into the lung, which we've shown very elegantly in that paper. And the combination of the 3 things that I've shown you here result in a much more balanced response, where there's some TH1 CD4 response and a huge amount of CD8 response. And we believe that's what's responsible for the excellent protection with this vaccine that we're seeing. I'd like to end by thanking the people that did the work. Some of my former members of the lab, Ali, Taylor, and Smriti, started this work, but much of the work that I showed you today was done by Dheeraj Singh, who is a former grad student of Dr. Gobardhan Das, and Garima Arora, who's a former grad student of Dr. Ramandeep Singh. Here they are pictured at a Holi celebration in my home 2 weeks ago, and all the other collaborators and the funders are also acknowledged. And also to let you know shamelessly that we are recruiting a few postdocs. Those of you who are interested in postdocing in the lab, these are some of the pictures of San Antonio from my home. It's a beautiful city. It's very rich in culture, and we can talk more about it later if you're interested. I'll be happy to take any questions.
Thank you, Dr. Kaushal. I think we can have 1 or 2 questions because we're running short of time. Yeah, go ahead.
Hi, excellent talk, very illuminating. Quick question, about IBALT and SIGEDGE story, what you mentioned. You had written a review in 2019 whether IBALT was a friend or foe in Journal of Immunology, and where you clearly mentioned that MTB, in case of MTB, it regulates the inflammation. However, in case of chronic antigen stimulation, IBALT can be a foe. But in case of co-infection, that could be a scenario where there could be a chronic. So just I would like to know your views on that.
Yeah, you're spot on there. And in fact, Lou Picker's shown that some B cell follicles actually protect T follicular helper cells, and these are a site of SIV, possibly also HIV infection. So that's a worry. We are actually funded on an NIH grant, which is was listed in my last slide, to actually study that. And we are going to be able to effectively address that question in the setting of SIGEDGE vaccination, MTB infection, and then SIV co-infection, as to whether IBALT is a friend or a foe over there. I have the same worry. But in this particular case, in the case of solitary MTB infection, the strength of the data is just too strong to ignore for us.
Deepak, very nice talk. I'm here. Excellent work. So I think now you have a wonderful model to analyze all the correlates of protection, right? So you have a single gene knockout which efficiently protects against MTB. So have you started looking at what is the role of CD4s or CD8s or antibodies in your model?
Yes, so that's the next step we want to, well, you can't see my slides now, sorry, but I do have a slide that I didn't intend to show, but to identify specific mechanisms of protection from delta SIGEDGE, we are going to deplete B cells, which we've actually done, and deplete CD4s and CD8s in the context of protection, and see what happens to protection, and then what happens to immune responses, and what happens to our single cell clusters. So that's planned, that's funded.
Hi, sir. This side. Here, sir.
Hi.
Hi. So do we also do survival studies in monkeys for pronouncing the worth of a vaccine in terms of its efficacy?
Yes, except we don't do time to death, we do time to humane euthanasia, because there are pre-specified conditions. Once a monkey meets that, then we have to send it to humane euthanasia. The initial data that was published in Nature Communications, we use rhesus macaques. Rhesus are susceptible to TB. So if we hit them with 100 CFU of MTB CDC 1551, which is actually a low virulence strain, but all of the monkeys progress. And so they meet the specified criteria, pre-specified criteria. So there we can do very easily do a survival study like that. The second study that I showed you, cynomolgus are much more resistant, especially with CDC 1551 infection. They're much more resistant to TB. So none of the monkeys, even in the control groups, met pre-specified criteria. So over there, the correlates are 1, CFUs in the lung or the granuloma, 2, the extent of pathology, morphometric pathology, and 3, PET scan scores.
And so how do you envisage this, that if you have an intention of taking this vaccine further, you would have to delete another gene somewhere?
Yeah, I wish you could still see my slides. So so what we've done is we've made 10 different double or triple knockout mutants derived from SIGEDGE. So they are in MC1A, COBM, FBPA, SAPM, SEC2, SODA1, LUD, PEN, CD, MET-A, MC3, MC4. So that's going to be a triple mutant, FAD-D29, HED-C, and RB3683. So these 10 combinations will now be tested in our monkey model. So these are chosen on the basis of the fact that these additional genes that I've mentioned, mutations and all of those we've already shown in my lab, are attenuated in the monkey model. So there's no danger of those double or triple mutants actually living through the monkey, and they should have the same amount of time that they persist in the monkey lung. And in many of those, we've seen there are strong localized responses, including eyeball responses. So that's why those were chosen. So these, the testing of these 10 double and triple mutants is slated for this fall through the fall of 2024. And so around that time, we should have this data. Thank you, Vivek.
Thank you very much. I think in the interest of time, we are really running
Thank you,
late,
sir.
and Dr. Kaushal is going to be around, so people can interact with him. I would like to close this session over here, and I would like to thank all the three speakers for their wonderful talks, and the audience for their active participation in discussions and deliberations. Thank you very much.
I request Dr. Varti to felicitate Dr. Margaret. I request Dr. GP Talwar to felicitate Dr. Kaushal. I request Dr. Krish Mohan Atmakuri to felicitate the session chairs, Dr. Varti, and Dr. Talwar. Thank you, everyone. One important announcement: if somebody has lost his airport, you can collect it from the organizing committee. Yeah, no. Okay. Okay. Now we have a dance performance.